those of you who attended we have the bottom in sight. So I guess we'll go ahead and
scan
be quiet here, and at least for a moment, and make a few notes,
Still
and then we'll go ahead and start the dive. Yeah, thank you, Lindsay. Appreciate it.
Okay.
Thank you.
Thank you.
You can ease off.
Yep. Bridge, this is Nav.
Slowing down.
Hey, Bridge, I just want to let you know that we just got to bottom.
Copy.
And when I acquire,
if I
come
could
down
give
to
a
15
bailout,
your sonar and
looks
an
like
altimeter allows.
pretty much just east would
So
be
very rocky
said
bottom.
best bailout
Once I get on
position if that
bottom,
works for you.
come down to 15 if your sonar and altimeter allow.
Copy.
Yeah, so as we were sort of looking at on our map, we were sort of slightly in the shadow of where the sediments might slide down the side of this hill, and so that is somewhat expected. The surface looks very hard and rocky.
Yeah, understood. That's not a problem at all.
The dark colors that we've seen earlier. This is does not look like it's heavily coated in manganese like we've seen most other rocks down here. This is likely limestone.
Yeah, and right out the bat, I can see some corals already. They may not be that obvious to those of you who are looking from at home right now or from one of the exploration command centers. And by the way, I meant to mention that Bruce Mundy is operating the exploration command center.
Can you find it on the other cams?
Inouye Regional Center on Ford Island.
I'm coming up.
This yellow coral is, of course, pretty obvious, and we'll get a zoom on that in a moment. But there's black cnidarians that appear to be black corals as well that are sort of all over the place.
Possibly on your head.
So we're seeing a lot of stuff already on the bottom. Smaller things, not nearly the huge
on your head.
colonies that we saw yesterday. But we've definitely got animals
here,
down
though.
here, so I'm anticipating a good dive.
Right here. And need to figure out which side of the 6-8 that is on.
And there's a fish, too, that just sort of
came
shot
left
down over
to
the
come up,
lower
right? But
right
it doesn't
corner.
guarantee that.
We're still way up because the ROV team needs to
when I
do
turned
some
to
additional
Yeah.
prep work.
Yeah,
And I'm going to take a guess on
try
this
to
fish
take it
from
off
this
your head
distance,
there.
believe it or
See
not. I think they're
that will drop it.
polymyxia, but I'm not positive.
You're not going to see it.
Yeah.
You can see it's.
Yeah, Chris,
look at
I
your tail
agree.
cam.
That's what
It's
it looks
up
like
on
to
your
me.
tail.
It's called a beard fish, or in Hawaii, deep sea moi. Unusual fish. It's probably only found in the moderate depths of the ocean, like where we are now.
Okay. Are you still in auto?
Yeah,
Yeah,
thank
I'm
you,
in
Bruce.
Heading? Okay, good.
Well, Bruce and I are extremely
hold
familiar
there.
with the species that occurs in Hawaii, and so we can actually spot these fish from a distance. Not really sure, what mental mechanism that is, except for a gestalt, I guess you could call it. And we got another fish down there, and it looks like it's another threadfin as well. I just got a quick glimpse of it, and I thought I saw the little whiskers on them, whatever they're called.
Yeah, and Chris, I thought I saw a fish go by in one of the lower corners on the left that looked like a mirror dory, a granicolepis.
Oh, I missed that. Okay, maybe we'll get another look at it in a bit. So they're looking in the water column because they do some process which I don't understand very well. I've seen them do this before.
Yes.
And so they've got to take care of a little bit of business before we really begin the dive. And it looks like they're also looking at the Sirius vehicle that's on top of us right now. That's the camera sled that's part of this whole system. There's the tether going off to the upper right that's between the Sirius vehicle. Roger, pilot, go ahead. Oh, you're not? Oh, okay. Okay. All right, just been informed by the pilot that you're not seeing what we're seeing here. They're doing a few things, so I won't describe what you're missing.
Mostly because we're not sure what's going on anyway. Doing some check. They do lots of checks. point out, normally we have at least twice the time in descent, and so there's a number of things that they check on the way down to the bottom while we're transiting down through the water column that they didn't have time for today, because we are at such a shallow depth compared to what we have seen.
So while they're doing some of their final checks, I'll do our standard introduction. I'm Chris Kelly. I'm with the University of Hawaii at Manoa and in the Oceanography Department as well as the Hawaii Undersea Research Laboratory. I'm serving as the biology lead on this cruise, and next to me is
Jasper Conter. I am the geology lead on this cruise. I am also from the University of Hawaii and in the Department of Geology and Geophysics.
So you are looking at a live broadcast, or you will be, I should say, you will be looking at a live broadcast from the NOAA ship Okeanos Explorer. We are currently on the east side of Wake Island in the Wake unit of the Pacific Remote Islands Marine National Monument. We've been here for a couple weeks exploring the deep water resources in this monument. And if you were fortunate enough to see our dive yesterday, you saw some of the most amazing coral colonies that I certainly have ever seen. Huge, huge colonies of corallium and other species of coral, some of the biggest I've ever seen. A very high density, gorgeous site. It was on a seamount south of Wake Island and was actually a seamount that was included in the monument after the expansion of the monument to the US EEZ that was signed by President Obama a few years ago.
That's right. And when you say huge corals, we're talking corals as tall as a person and wider than you can reach your arms pretty much, right?
Yeah, absolutely. Meters in height.
Yeah. So,
pilot is watch lead. Are they seeing the video feed yet or not yet?
Yep.
They are, okay.
Just came back, Chris.
Great.
An urchin.
So we've got a lot of stuff right here on the drop site. I think you can now see it a little more clearly. And what we're going to do is, like we do most dives, we're going to spend a few moments in documenting the different types of animals at the drop site. And then once we've kind of finished that process, then we'll start moving up slope. So the yellow colony you see on the right-hand side, I'm pretty sure this is a hard coral. It's called Analopsamia rostrata. So a pretty common species that is pan-Pacific as far as we know, at least Central Pacific to the Western Pacific. We saw them in the Marianas on the previous cruise leg. And we've got a lot of black corals here too. These are these very dark, smaller colonies that are on the rocks. Saw some anemones and a squat lobster a few moments ago.
Now also seeing a number of small fish hovering up over the bottom here, so I think these are probably midwater fish.
Yeah, copy that. I'll just do 10 meters at 200.
To the bottom of the, kind of gotten stuck there. There is a larger fish swimming in the view.
Yeah, Bruce, we just got a better look at that. We just got a better look at that polynexia, by the way.
Bridge, this nav. Hey, can I put in a ship move, please?
So they're called beardfish because they have a pair of filaments on
Okay,
the
if you need
chin,
to
like
finish
whiskers.
too, I can wait a little bit. Okay, copy that. Yeah, I'll just drop it off then. 10 meters at 200 degrees
I'll
at
just
0.2 knots.
fire off a few names of the researchers that are currently in the chat room helping us out with this dive. We've got Diva Amon.
Perfect, thank you.
Was at the University of Hawaii. She was working in Craig Smith's lab, and she's now back in Trinidad.
That move's pending.
We also have Asako
rotation.
Matsumoto from the Chiba Institute in Japan. I've already mentioned Bruce Mundy. We have Charles Wall from the Marine Protected Divisions, NOAA. We have Deborah Glickson, who's a geologist at Harbor Branch, and she was the science lead in the first leg of the Marianas cruise. Those of you who saw that leg may recall. We have Nicole Morgan, and she is with, I think it's South Carolina, if I'm not mistaken. But we also have Tina Molitsova. She's with the Shirshov Institute of Oceanology in Moscow. So we got a lot of help already, and the chat room typically just increases in size as the dive gets started.
Sounds great. Thanks, Rich.
I'm just backing and dropping so that Tyler's not. You can come down with me.
Okay.
All right. Actually, we're just getting situated here. I think we're on bottom and we're in a comfortable position. We landed on on this cliff face, so it's a little interesting.
Another little fish going across the screen. Of course, lots of corals on the bottom.
You can't hear me.
They're just very dark colored, and there's really a polymixia over on the other side. So we've got a fair number of fish and corals today. We haven't seen any sponges yet, but this is pretty shallow for most of the glass sponges. That's not particularly surprising. We haven't seen anything yet. Jasper, what is this? What's this substrate? Again, does this confirm your understanding of this carbonate?
Can you tell him he's muted? Jasper's muted.
They have, usually these seamounts have hundreds of meters of sediment on top, and so this is probably part of an older section of the reef, and that was potentially weathered when this volcano was near the surface. And it is now exposed underwater, coming out of, sticking out of the sand that's being shed from the atoll above. You sort of see the smooth sides to this. That's suggesting that this is, it's kind of bumpy, but it's not super sharp and jagged, and so chances are this has been weathered for some extent of time to sort of get these round shapes.
Watch lead pilot.
Roger, pilot, go ahead.
We're here and we're ready to accept any zooms or.
Okay,
direction.
we've got a laundry list for you right now.
Okay.
All right, so let's start with one of these dark brown ones, and perhaps the slightly larger one to the upper left center.
Okay.
We'll begin with
Get
that.
lasers on, please.
Lasers coming on.
And Chris, if you have a chance to zoom in on one of those elongate fish, that would be good. I know they're moving pretty fast, though, so it may not be possible.
All right.
Thanks, Bridge.
Yeah, Bruce,
Come in and frame the.
I saw those,
lasers.
and we'll definitely do that. There's none in view right now, but I was wondering whether that could be a trichiurid. I don't know what you thought about it.
Can you bring
Yeah,
that
that's
drawer
another
in?
possibility. I was thinking they might be Barracudinas, but that's why it would be good to zoom in so we can actually see them better.
Sure.
So
we're waiting for Tina Molitsova to comment on this colony. It's Antipitherian, I'm pretty certain, and Tina is one of the world's authorities on Antipitherians or black corals, and she's on the chat. She doesn't sound like she thinks it's a a black coral. I was thinking, Tina, it could be Stauropathes starocrata or something like that.
Why is it so
as a
dark?
typo. She definitely believes it's an Antipatherian. Got it?
Swing arms are in, but I don't.
Okay.
And
I can check the upper.
we can get a little
It's
closer
down.
look, or
No,
is that
it's
it? Just to give Tina the best chance of...
Yeah, we can get a closer look. I'm just getting situated here. Come on in, video.
Okay. So that's a full zoom.
Perfect.
That's a nice shot. All right, Tina thinks it's Stauropathes. So I think one of the things she's looking at is secondary branches coming off of these primary branches.
Okay.
And she's asking whether Stauropathes goes shallow in Hawaii, and
That refers to the fact that we've seen this at a much greater depth as well in the last few dives, right?
yeah,
I
so
think we saw it yesterday at like 1,500 meters, 1,400 meters.
right, and I've got records from the old Hurl
Can come in a
animal
little more
guide
if you
here
have
that
That's as far in
this
as I
one
can go.
species,
Okay.
Stauropathes starocrata, is between 600 to 800 meters, so that's kind of where we are right now at the upper end of that range. So I guess the answer would be yes.
Okay. I'm set when you are.
All right, I'm good.
Stauropathes it is.
Okay.
Come on out.
And then when you're finished with that, let's see number 2.
So we're full wide.
I think it's the same thing, but no, let's go with this bright red one right on the top. So this is definitely different.
The smaller or the large
The
one?
larger one.
Okay.
And it's actually got a whole bunch of commensals on it too. We've got a feather
come on
star, which is that purple flower-like thing with the long arms right on the end.
So that's a partial.
We've got gooseneck barnacles over on the right-hand side as well.
Those have funny shapes. And she says Leopathes.
Yep,
go
that's
in
what,
tight.
That's what
actually
you
read
showed
it.
me earlier
Okay.
too. So you guys agree, it looks like?
The reason we agree is that Tina has taught me a lot of this stuff. Tina and Dennis Apresco is another one of the black coral experts, and Daniel Wagner. All of these I have attempted to leech off of all of their knowledge as much as I could. Yeah, so Leopathes is what I guess we would consider to be an intermediate depth black coral. The actual skeleton has commercial value, but they don't occur in the abundance that some of the much shallower water black corals occur at, and therefore it's not really harvested, to my knowledge, anywhere in the world. I don't know if that's your impression too, Tina, but I'm not really going to call this one a precious coral species.
Is this also the genus that lives for a very long time, if I remember correctly?
You remember correctly, Bruce. And you want to tell that story, or did you want me to describe it?
Well, I don't know it as well as you do, but in brief, what little I know is that by using the hard skeleton and looking at isotopes in the skeleton that include markers set down from atomic bomb tests in the 1950s that put certain unique isotopes into the water that were then taken up by the hard skeleton. It's been shown that corals in this genus can live for a couple of thousand years, I believe, maybe even several thousand years. I'm not sure what the latest update on that is. So people were thinking that, these corals might live for decades or maybe a century or so, but it's turning out that they live for many centuries and even a few thousand years.
Yeah, so Tina Molitsova basically has put in the chat that they now know that they can live to 5,000 years old. That is really amazing. I know some of the people who are working on dating these corals and looking at their skeletons to try to give us some information about past climate change. And this is Rob Dunbar at Stanford and Tom Gildeson at the Energy Laboratory. It escapes me the full name of his
All right, to
facility.
zoom out a little bit.
And Brendan Rourke down in Texas A&M. So thank you very much for that view. That's beautiful.
That one just retracted a little. Oh, and he's coming out again.
Moving a little bit, yeah.
The one in the top.
So this camatulid feather star has extremely long naked ends to the actual arms, and that'll actually be a character, I think, that'll help in its identification.
All right.
So when you say that for the folks at home, that means a lot of people
like
are
they
going
might
to
wanna
be
see
looking
the
at this
crinoid a little
footage
bit.
and still
okay.
images from
Okay,
this
I've got
footage
it.
to try and pin these
come
animals down to a more precise level.
Right, and I'll take a guess at the
I always get
gooseneck
excited for
barnacles
crinoids,
too. These
but I
could be in
just-
the genus
Yeah, they're
Herolepis.
really cool.
I just assumed that.
This is
I'm the
just
only
from
one.
some of the things that I've been able to find out from Bob Van Sciok at the California Academy of Sciences, who's a barnacle expert. Pilot, we're good from science
good.
whenever video's
Okay.
ready.
All right.
All right,
Thanks.
come out. So we're full wide.
Okay, I think all of these black corals are the same, so we're going to look for one of the yellow ones
Okay,
that we
I
saw
think I see
coming
one
down.
coming up over
Yeah,
here.
there's
Up
one.
on the left. Yeah.
And there's a purple one in the top, almost right
oh
corner.
yeah, right there.
How about the purple one since it's closer, and then we'll get the yellow one.
Okay.
Good spot, Jeff. I didn't see that one. Good.
So how many purples that aren't Victigorgias, right?
Yeah, so I have an idea what group this belongs into, but not really sure until we get a little bit of a closer view.
Come on in, video.
Okay. So that's a partial.
Thanks.
Right, so let's see if we can get some opinions in the chat room about this one. They have a couple second delay, so we're going to
Come in a little farther if you'd like.
Okay.
give them a few minutes to comment. There's some commensal on it too. Looks like an anemone. So Asako Matsumoto has gone to a purple octocoral so far. Sako
right to zoom
and
more?
Yep.
Tina,
That's
I
full
think
zoom.
this is a plexaurid. Think I'm seeing scales along the branches as well, and there are a number of very colorful plexaurids that we're aware of. Anthomuracea, for example, is one of the genera that has a purple colony. Polyps are a little bit bigger than the species that we know in Hawaii, Anthomuracea tenuospina, but I don't know if anyone agrees with me on the chat, but I think plexaurid purple is a reasonable good field ID. Family Plexauridae are a sort of a variety of soft
clear one.
corals. And Asako's pointing
Right,
out that the
video's
polyps
clear.
are really
Go for
big,
the
and I
yellow.
agree, but
Guy.
I'm not sure that
Alright, we're full wide.
anthophelids have got those kind of scales
It might
on
just
the
be the
stem,
terrain
but
we're
maybe
on,
I'm
but it looks
mistaken.
like we're lighting up stuff in
I mean,
a
that's
different
a
way
beautiful
today.
ID.
It's
We're
like
obviously
it's
going to have to study that a
the
little
lights
bit
are aimed
more.
further
And
out
then
or
now we're
something.
coming up on one of these yellow colonies.
Okay.
I don't know enough to know what
we
to.
can bring the upper swing arm lights down a little and see if that
Tina
makes it
saying
any better.
perhaps Victigorgia.
Yeah, except Tina didn't have the typical pink base on the stem, and then again I was
Better.
noting that Victigorgia tends to
yeah,
have
I
a
can't
smooth
tell.
stem, and
What
this
video
clearly
it
has scales
Yeah, that-
on the stem.
think
That's
that
why I
looks
went
better.
to
You like it?
plexaurid.
Okay, I'll go all the
But
way up and I'll come
I
down.
don't know.
You let me know.
We're
Okay.
going to need to get a
Where you
bunch
like
of
it.
opinions.
Okay, you can come in on the yellow one here. This yellow one,
That
there's a
was the
couple
whole-
different kinds.
That was everything.
This is weird. Oh,
I
it's
couldn't
over.
actually hear you, Copilot.
Open down there.
Yeah, we're kind of looking down on it a
Doesn't
little
appear to
bit,
make a
and
big
this is
difference,
clearly Analops samia with the stair-step coral
Yeah, I can't tell.
arrangement here. This is a scleractinian
Come in,
or
partial.
hard coral.
Are those on? Start there. Those lights on?
Are the lights on? Yes. I
Yeah,
did
okay.
move it and I could see us
Oh,
sitting
okay.
over it, but it didn't sound like it got
Tina
any better or worse.
Molitsova agrees that this is Analops
We're on a
samia.
Bit
and
of a
she
cliff for the
saw an
lowers. We could
anemone.
bring the port out. I keep swinging on my starboard because of the current, so
Want to bring it out very slightly?
you can bring the port down slightly and see, or even around a little bit if it makes it better.
I can
Yeah,
try
here.
inboard first.
She's also commented that this is another star of
That might be
to the
glary.
left, which
I might
is
want to
good.
bring out and then inboard a little.
Copy that.
That's a Antipatherian coral to the left, on the left side of the video, and this yellow one is a hard coral that's in a completely different group. This yellow one is more closely related to your big mounding typical shallow coral reef corals. If you're a scuba diver or a snorkeler in tropical waters, you'll see a lot of those. This is
Working on
sort
the
of-
lights video, hopefully they
This is sort of a
Gets a
deeper
little better.
relative.
Oh,
Further
thank
than
you
that.
guys.
a little bit.
Okay.
Just as long as you don't have the shelling, right? That's all. That's the only
Yeah.
thing that.
Yeah, that does look better.
Okay. Yeah, we hadn't
sci-
deployed the lowers because we're on a cliff face right now.
Right.
So science is good with this
Right.
whenever video
Okay.
is.
Okay.
Yeah, we were just adjusting lighting so that things look a little better. But I can go to the next thing.
All right, I'll come full wide.
Thanks.
Yeah, that's way
Right,
better.
let's
than
take
it
a
was.
look at this anemone that's over to the left, if it's possible to go over there. And looks like a flytrap anemone. It's right on the
Yep,
edge of the
right on the edge. Well, there's
rock.
might be two of them even. There's one.
Looks like there's one
Yeah.
even closer behind
Maybe
the
this
coral.
this one that's coming right to the left of the lasers now, because there's another single polyp scleractinian that you could get in the same view.
Okay.
Nice crab or something over there.
Oh, we only have another 20 minutes.
Yeah?
Oh, jeez. Sorry, man. Should have got you in here.
Or shine.
I thought we were going to 20. I don't know why I thought we were going to another hour and 20. That's not right. Come on in.
So that's a partial.
I have an idea of what this is, but I'll wait to see if we get some suggestions from the chat.
Take
When
it
I
the rest
say
of the way
chat,
when
I'm
you're
referring to our text
Alright.
messaging chat room that other researchers are participating, watching the video and text messaging us their ideas. So our current depth is 635 meters. Our temperatures is about 6 degrees centigrade. That's a little warmer than we've been for a while. And our solved oxygen is still pretty low. It's,
Yeah, it's only 2.
Still at 2. All right, so this anemone, not seeing any other suggestions, I'm fairly certain that this is something in the genus Actinoscyphia. It is a Venus flytrap anemone. And then if we could slide over to that single polyp scleractinian to the left,
right where it
when
is.
video is
Okay.
done.
The polyp here.
Well, actually,
Over
this
to the
snap. Did I hear you call me?
possible cup coral or something.
Nope.
All right, sorry, never mind.
All right. It's basically
Jeff is
centered.
sneaking up on it,
There it is.
creeping up on his quarry, and he's got it.
He's got a nice little vase. Did they retract into that, or is it just hanging out from there?
They can track that a ways into that. I'm not sure they can go completely into it, but I'm not positive. So my guess, based on the striations on the actual cup itself, the calyx, if you will, and also just the appearance of the tentacles and the shape of the calyx. I think this is in the genus Polymyces, and it looks quite close to Polymyces well's eye. There's something we've seen in the central Pacific. You can see all the nematocyst batteries, those little tiny bumps on all of the tentacles. Those are all nematocysts or stinging cells. So this is also related to the yellow coral that we just imaged, if you guys can believe that. And they're related in the sense that they secrete this hard
Happy when you are.
calcium carbonate
All right,
skeleton.
I'm good.
Okay.
So come on out.
Great.
Great.
So we're full wide.
You've got a little squat lobster over there, and I guess what we could do. Are we maybe ready to start a little ship move?
We can continue up.
Yeah.
I don't think that we're quite on the ridge, or close. We're not quite on the ridge crest yet.
Yeah, do you want
Yeah.
to start
I
with
agree. I think we
15
got
meter?
most of the major
See,
animals
see how I'm way out here like this?
right in
Yeah,
the
you
landing
seem
site.
almost a
By
little
looking up a little bit, maybe
off.
we'll
That
catch
shows
some
you
of
I-
these
30
fish
meters
that
away.
Yeah,
Bruce
let's see
Mundy was
Sirius.
interested
So I
in.
could sidle right and try to get centered in Sirius here.
Do you want to do that first, then we
Yeah,
can
let's do
adjust
that.
Sirius?
Let's do that, and then we can adjust Sirius heading and get everything lined up to go up the ridge.
Yeah, copy that.
So for those watching at home, this moving of ship type of
Ooh, I think I see a different kind of coral as I
discussion
float
Yeah,
over.
if you
is
shrink
a very
the
brief
RV,
way
it
of
should,
describing
give
this
us a
sort
little
of
better
careful choreographed
It does
dance
look large,
between the
doesn't
ship and the two slides.
Pilot, there is a white coral that's coming into the center right now. You're seeing it on edge, right
Yeah.
above that red one.
Yeah, that's I'm headed for it right now. I saw it on my way and decided to go that way, thinking you might want to see it.
Yeah, great. Thank you,
Looks
Jeff.
like there's a
Appreciate it.
sea star
There's
down
a
in the
fish,
bottom
fish
right.
shadow
too.
swimming in
there.
the lights.
Oh yeah, he's touching the craft right now. Actually.
Can we swing over and take a look
Yeah,
at
there
it?
he comes.
he's
He's
right
coming
there.
There he is.
and another one.
Polymyxia,
Oh, a whole
3 of them.
school of them.
Yeah. These are the beardfish, right? Bruce?
Yeah,
Is
this
that correct?
guy's slower here. Okay, come on in on that guy.
Okay.
I've got a
He's
little lag
lingering
here,
a
so I
bit.
haven't seen them yet. Okay, now I do. Yep, they are 3 polymyxia. I'm a little confused about the species, so I can't say right now which they are, Burnti or Japonicus. But yeah, they're these beardfish. They have the sensory barbels on the chin that they use to feel along the bottom and find small worms and crustaceans to feed on. So that's a good
All
view.
right.
That should help with the identification.
He's outta
And if you
here.
can linger on the fish
Come
a
on,
little
White.
bit like you do with the corals, that's always helpful to pick out the
Chased him
identifying
back to where we
characters.
started.
The
Hey,
color
pilots, do
pattern
you want
on
your
the
sonar
fin
brought in
is
at all?
important for
We're still at
identifying
1580, respectively.
Perhaps
you can
this-
bring me in. I like
Perhaps this is
Sirius
a
where it is at
stupid
the moment.
question, Bruce,
Copy that.
but do 2 whiskers constitute a beard? I don't know. That's a pretty scrawny beard, if you ask me.
Are you,
Oh,
are you
there's another
judging
one
the
going right
manliness
in
of
the
these fish?
In this case, yes, they do, though, because that's what people have named these fish. So
little
I guess
sea
it's enough.
stars, well, yeah.
Maybe the minimum.
I think I have more of a beard when I wake up in the morning, but whatever. No, it's just-
I was going to say, I think it's going to depend on who you ask.
that's true. Maybe they're curling them. You know how they do like a handlebar mustache? Maybe they're,
A little grease.
yeah, a little bit.
All right,
Sure.
this is kind of the
Now,
best
who
angle,
knows what's
given
going
the
on at night
terrain.
here
You
with
come in
these
on it.
fish? They could
Okay.
be doing all types of interesting things.
Isn't it always kind of night
That's
here?
a partial.
Yeah. Right. And just to warn those of you listening, I am prone to stupid jokes, so you may hear some of those. It's, listen at your own risk. All right, so we're looking at
Coming a
another
little
coral.
closer.
We're getting back to seriousness here. And I think this is a bamboo coral of some kind,
Wow.
because I see part of a dead stalk behind it.
That has a
Yeah.
little anemone on it, doesn't it?
It does. And I actually can see some nodes on the live tissue as well. And
And it's not
unfortunately,
quite
I do
full
not
zoom.
see either of our bamboo coral experts in the chat room yet. Can
What
we
do
see a branch point
A
anywhere,
branch
Jeff?
point. Yeah. Come on in,
Let's-
video.
I think it's internodal. I think I saw one.
That's full zoom.
yeah.
Right there. Yeah.
So when I'm referring to internodal, what that means is that the members of this family, which is called Isididae, they can branch either at that dark sort of band along the stem that you're seeing a really nice image of right now that's called the node, and it gives the family the name bamboo coral because they occur at different intervals. So it can either branch at that black node or it can branch in the white area between nodes. And when it branches between nodes, we call that internodal. And that's informative. That helps narrow down the possible candidates that this species could be. The other thing they're looking at right now is they're looking at the polyps very closely, and I do not see very many sclerites, which are the microscopic parts in the polyps. And that will also be informative to people who know a lot more about these animals than we do. Sako Matsumoto agrees this is an icidid.
To the right and start getting lined up on that so I know where I'm headed. If you don't mind.
I think we're good, pilot, whenever video is. I think that's the best we can do with this.
Okay.
All right. Video's clear.
I'm clear.
Okay.
Tina also concurs the sclerites are invisible, very thick tissue.
So we're
so
full
it's
wide.
an unusual bamboo coral. There is a different animal.
Where?
The pom-pom anemone.
Oh.
To the upper
Do you need
center.
a view?
Yeah, it's
Okay.
it's above the laser, just about now.
Yeah, I see it. I have it in my sights. We'll get it.
Oh, Abby LaPonte is on right now. Sorry, Abby, I didn't
Come on in.
notice you were on the chat room. So Abby knows her bamboo corals. We've got
Oh,
a
it's a
bamboo
partial?
expert on the line. This is great. She thinks it looks something similar to the genus Keratoisis, but she also thinks it looks a bit new. Tina Molitsova has identified this anemone as in the genus Liponema, and common name for that is a pom-pom anemone.
Is it all right to zoom?
Beautiful, delicate animals, extremely hard to collect because they're so fragile. Most of the tentacles there are just water inside, and they come apart very easily. All right, what do you think, pilot? Should we give the fish guys a chance to spot a few more fish? And
All right.
All right. Video's clear.
Thanks.
thank you very much. A gorgeous
I'm looking
close-up.
at 2, 2,
By the way, the red
3,
dots
so
are a
somewhere
laser
around there.
scale. They're 10 centimeters
How does
apart.
that line up with the bathy?
I always say this,
So
but
Bathy, I think, is
even though they're
250.
lasers, they're so weak they certainly can
Yeah,
do
I'm
no
looking at-
damage
I'm looking at
whatsoever to
the
the
Bathy where
animals.
I am,
They're
and
just
those
light
looks- I
spots,
mean,
so
that's-
that
it's
we
out
can
here
understand
somewhere.
the size of
Yeah,
what we're
we
looking
can
at.
still
that
lateral
maybe.
a little bit.
Yeah, I'm gonna lateral right a lot
Okay, I'll keep going.
and come around and.
Yeah, I wouldn't mind seeing what Zero sees at 240. See how that does in sonar.
That's 2, 4,
Yeah.
5.
Oh, looks good.
Yeah. I can
Right.
put you a move in that direction.
Yeah, let's do that.
That's pretty. Do you just want to start with 15 meters?
Sure.
Copy that. 15, 245.
Ooh. I had a fish just bonk into the back camera of the vehicle.
Bridge, this nav.
Any chance we could get it? or
Hey,
is
can I
that
put it in a ship move?
Not
No, and
possible
it was
for
another
it.
one of those bearded.
the beard
1.5
fish?
meters
Okay.
at 245 degrees at 0.2 knots.
I got to see his whiskers in the camera. He looked right into it.
Could we tilt up just slightly?
Yeah.
Just
Of course.
Good copy.
a little bit longer view. Thank you. Jeff, so do
Thank
you
you, Rich.
detect much of a current today? And if so, do you have an idea what direction it's from?
I don't detect much, but it seems along the ridge from the west,
Okay.
possibly. Yeah, I see that guy on the crack there. He's coming in and out of view, but we can zoom in and see if we can catch him.
Yep.
Come on in.
Oh, there is a little guy. Oh, he just swam around the corner.
Yeah, he's been coming in and out of there.
At the end of your tether right now.
Thanks. Yeah, come out wide. Oh,
There he is.
Just
Pan
pokes
up
his
a little
head.
bit. Oops,
Yeah.
almost had him.
And gone again.
Just the nose worth.
How good are you, Bruce? Can you ID the fish from the very tip of his nose?
I think Jeff is now thinking we're.
Sometimes you can get a guess.
There he is. He's sticking out a little bit more. Boy, got long. If it's okay, stick with him just a
Can
little
you
bit
brighten
longer.
him up without...
There
brighten
he
up a bit?
is.
Yeah. Come in a little more, maybe, if you have it.
Oh,
I've
that's
got
his
a
head.
lag, and then I've got screen freeze here. So
He's
it's
a
difficult.
cute little guy.
Yeah, he
You'll
is.
see him. I don't know what this is, Bruce.
I've got a
I
little bit more
I don't
zoom,
recognize
I think.
it.
You can take it.
Oh, that was it.
All right, we'll take it. Take all we can get.
Yeah, if he'd come
Now,
out
how
a
far are we
little
in that
bit
move?
more, we could see if that's a single short dorsal fin or a long dorsal
Move's
fin.
only been going for a minute. The
kind
ship
of acts like a
is
morid
almost 10
of
meters
some
in.
sort of codling.
So yeah, you
I
should
don't
probably
see a
start to feel it
whisker on
actually.
the chin.
Yeah, I am.
But
a
again,
little
he's
bit.
just not quite showing enough of his body to for us to get a good identification.
Yeah, the eye is really big. I don't know if that helps. You're obviously seeing this too.
Yeah, this looks like a morid.
Come out, little
to me.
guy.
Again, I wish he'd just kind of come out a little bit more so we could see if it's a single short dorsal fin followed by a longer
Well,
one. He's
Why don't
not
you
a
come out
recruit.
partial? I might be able to, now that we've got this long shot, I might be able to sneak around and get a little more of his dorsal
like that
fin,
to
because
me,
I think they want to
particularly
see.
with the clear
So
membranes
that's
between
partial.
the fin rays.
Okay.
And I
Thanks.
don't think it's an
Just kind
eel.
of try to sneak
From
left
what we
a
saw
little
before,
here.
it was a shorter body
Or sneak
than
right
fish.
a little
So
here.
I'm
Let's
still
see if I can't
guessing
get
codling the morid.
more of a view.
Bruce, he's going to try to just get you one more look, see if he can position just slightly better. Maybe give you a little more view of the dorsal fin. That's kind of tough because it's in such a little crack here.
Yeah.
Whoop. Whoosh. Okay. Yeah, that'll flush him. Oh, there's a shrimp. Looks like a plesionica. The shrimp.
That was quite the whoosh.
Okay, might get a better view of him. It looks like a little more of his body is exposed.
No,
oh,
his
there's
tail.
his tail. Quick,
Come
zoom
on
in.
out. Come in.
Yeah, if it has
Of
a
course I
distinct
dusted it.
caudal fin and a distinct tail fin that's separate from the fins on top and the bottom, that would make it a morid.
Yeah, Bruce, I think you nailed it. I think it was a morid. And he's coming out again right now. A little bit of sediment in the water, though, but
That fin goes all the way to the back. Or is there a tiny?
the
No, there's a
tail-
tiny one in front of it.
tail fin's a little weird, Bruce. I don't know if it's damaged or whether that's...
Feeding the fish.
But I'm convinced that this is a codling. It is a morid. It's
All right,
genus
thanks.
maybe
I need to come.
Fissiculus or
Yeah,
Thank you,
come out.
Jeff.
Okay.
Yeah, that'll be good.
Yeah, Pies, that ship move.
Yeah, go ahead, Bruce.
Very
I'm sorry to
full
interrupt.
So I think we've got the best
Copy
that
that,
we can
thanks.
get.
Yeah, I think so.
Sorry for the poof. I'm not sure what happened there.
So let's go ahead and start up slope a little bit and see if we can see some more things. Lots and lots of these Stauropathes all over the bottom. This black coral or Antipitherian.
Looks like orchid crinoid right in the very middle.
So Jeff, is the icon for the ROV on HYPAC reversed, or are you really going backwards?
I'm sidling towards the right, but when we're moving on a cliff going up and down, it's hard to know.
Oh,
You know,
okay.
we're going up, not really.
Okay. Yeah, the movement is up, but yeah, that's why I'm trying to register what I'm seeing on HYPAC and what you're doing right now.
And so I'm
uphill towards the...
We landed off towards the south on this, and so I've been sidling towards the north to get centered up on the cliff and coming up at the same time.
All right, I understand. I know where my confusion is. The purple is actually deeper and the blue shallower on this color scheme.
Yes.
Okay,
Yeah.
now I get it. I understand. Thank you.
Yeah. Towards the west is deeper. And we have
There's a little urchin. Yeah, maybe we could do a quick zoom on this.
Okay. Tilt up
Looks
a
like a
little.
spiky guy.
Tilt up a little. Come on in on the urchin as I get it centered in view here.
The spikes are bigger than his whole body.
Oh yeah, thick spikes. Can go in a little farther.
So these are beautiful urchins. We've got the species in Hawaii
you
as
good if we do
well.
another 15-meter move?
Just seeing if anyone in the chat room
Copy
wants
that.
to
I'm just going to keep it at 245.
forward
That works
a
for you.
guess before I throw out my guess.
Bridge, this is Nav.
Chris Ma, actually from the Smithsonian and Echinoderm specialist, might be calling in as well in a little bit. So this is in the family Podinidae, and this is in the genus Cynopodina, and I'm pretty sure that it's Cynopodina.
Hey, can I put in another ship move?
And just by
all right, same thing.
everything,
1.5 meters,
green
245
tone, the
degrees,
different
0.2
sizes
knots.
of the spines, the thickness of the spines, and other factors.
Good
It's a
copy.
dead ringer for that species that we have in Hawaii. Yeah, we're good when video is on, pilot.
Thanks,
This
Bridge.
one.
What's that?
We're good on this whenever video is. It's a pretty common urchin. You got a little squat lobster.
All right, video, I'm clear when you are.
All right. Video's clear too.
Thanks.
All right, we're full wide.
And we're going to have a little brief pause. I think we're going to have a front row change out in a few seconds here. See some other guys have moved into the control room.
So, Chris, I have a guess about the genus on that morid. I think it's probably a Gadella. When we zoomed in, I didn't see a chin barbel, and it looked like we could have seen it if it was there. So that would make it in the genus Gadella. And I just typed in that I'm not aware of any records of that genus from Wake Atoll, but then nobody's ever looked or sampled at the depths that we're at before at Wake, so it's no surprise. We'll probably have just about every fish that we see at this depth be a new record for the island.
That's right. We're setting records left and right.
Yeah, thank you, Bruce. I did see your note. And when you first mentioned Physiculus or Gadella, that's what I was wondering, because Physiculus is going to have that barbel and Gadella doesn't, just as you explained. And so, yeah, I did not see that barbel either. I think we would have seen it just from the views that we had. So given that that had a barbel too, Bruce, given that that had a barbel too, why don't we call that a beardfish as well? Although it needs two barbels to become a beard.
What it is
that
yeah. Maybe one you could call
it. It didn't have the barbel.
That's right, it didn't.
Maybe we could call it a goatee fish.
Hey, pause, just so you're aware, we're just finishing up a move.
Sorry, I meant the other members of the family, the Physiculus. You're right.
Up and out here.
I sort of blew my joke. Sorry.
Co-pilot, I'm going to come up a bit and push out.
Copy
that
that.
can
Coming out.
get to be confusing because there's a shallow water family of fishes that have two barbels on the chin, slightly different structure morphologically, but quite popular by fishermen, and they're called goatfish. So we've got to create confusion by having goatfish and goatee fish.
Okay.
But you actually
I'll try to come up with a better name.
You know, goats are known to have beards as well, so maybe that's they're going with the same beard theme by calling it a goatfish.
Yeah.
All right. Do you want to point your camera down at like, 40, 45? It's fine.
41.
Right there is perfect.
Okay, we're moving up slope right now. Our current depth is, what is it? That's hard display to read
That
here.
should move
595 meters.
Yeah.
So we've come shallower than.
Looks like you got the port swing arm
no,
out.
according to this, we're not. We're at
Starboard swing
610
arm.
meters.
Yeah, just the port one is
Yeah,
reading-
595 is the other vehicle.
Yeah, I was reading the wrong numbers.
All right, Fernando, Levi. So the plan here is we're going to keep making our way up this steeper face, and then once we kind of see what we think is top of this slope, we're going to
Turn right.
angle. We're going to still kind of face up slope, but angle Sirius in our part way in our direction of motion and part way up slope and kind of skirt along. So
Yeah,
Copy.
just as
Be a
another
little
comment,
different today.
I
Follow
don't
the
think there are
route.
any records of the beardfish that we've
All right,
seen
that
of
sounds
that
good.
genus from Lake Atoll either. And again, no surprise because nobody sampled or looked here.
Yeah, thank you, Bruce. Pilot, so now it's Dan Rogers, I believe. Is that correct?
yeah, so we just had a shift change, and now we've got Fernando Aragon and Nav. I'm Dan Rogers as the pilot. We got Levi Unima as co-pilot, Annie White on video, and Emily Narrow back in the clipping chair. And we're all with the Global Foundation for Ocean Exploration.
Great. Welcome, Dan. So maybe we'll get a quick snap zoom of this anemone. This is the first one we've seen of this type.
Sure.
And this is different than the Venus flytrap anemone we imaged earlier that I identified to Actinoscyphia, to that genus. This is the other type of Venus flytrap anemone. It's in a different family.
All right, video. I'm going to go in.
Okay.
Hold there.
So this is in the family Hormathiidae, and this looks to me
Going a little tighter.
similar to something we've identified again
Right
in
there.
Hawaii as being in the genus Philiactis. That actually ID came from Dr. Michelle Rodriguez
Co-pilot,
in
are
New
you
York,
happy with the
and
settings
she's
in
one
your
of the
sonar, or do
anemone
you want to
experts
come
in the
in
world,
250?
and she kindly identified a
Yeah, we should
specimen
come
of
in
this
50.
that we
Yeah,
sent
50
her.
would be good.
Again,
Alright.
something that we collected in Hawaii, but it looks
You want to
looks
zoom out of
quite
here
similar.
and frame it up?
So
Okay.
is this one all closed up because it's just done eating or eating something?
Could be. Yeah,
can go
not
in a
really
little further
sure.
than
We're
that.
kind of
Just
looking
went
right down on it.
What was
So
that?
it does
You
have
can
a
go in a little
stalk.
further than that.
Okay.
Yeah, hold it like right there. It's just going to frame it.
But are you ready for me to move a little bit?
Yeah. All right, let's zoom out video.
So we're full wide.
Okay.
So, Dan, if we can keep a little bit wider view just to spot fish.
Sure. Well, are we full wide video?
Yes, we are.
All right, we're full wide right now.
And then when we spot some good invertebrates, we'll zoom in. But it's a little hard when you're coming up a steep slope, obviously, to see too far ahead.
You see how the rocky formation has these whole scoops missing. It's definitely looking
I'm ready
like
for a
it
move,
got
Bob.
Yep, let's go ahead and put it in.
weathered out.
They've
Dissolved.
been doing 15-meter moves. Does that sound good?
I think we can do a 20.
Yep.
Okay.
And it actually looks like it's going to start getting flatter here. So
Cap it.
That's
0.2 knots.
More substantial sandy patch
Copy
here.
that,
With some,
putting that
some
move right now.
ripples in there, probably from the current driving over
Bridge,
the,
this is Nav.
over the top of this ridge. Oh, there's another one of your beardfish.
Yeah, and this might. If
Let's
he's
get a
gonna
move in.
cooperate,
That's
it
going to
might
be
an
20
opportunity to get a
meters,
little closer view of
245 bearing,
the previous
0.2
ones we
video.
looked
knots.
at were not very cooperative.
Good copy, Bridge.
Going further. Hold there.
Big eyes.
Oh, it's a marvelous shot of this fish.
Gonna kill lasers. You got it.
Now we're getting into depths where there might be some very dim light downwelling from the surface, so
Going
eyes
tighter.
would be more useful down here
Hold there.
than they would in the deeper waters where we usually survey. But again, they're probably just seeing silhouettes or shadows or fishes and other prey.
Good copy, Bridge. Thank you.
You want to zoom out a bit? Just a little bit. Yeah, hold it right there. Nice.
Bonk.
It's moving quick.
So an absolutely marvelous shot of that fish. That's the best I've ever seen. That was beautiful.
All right, let's
Thanks,
zoom out.
pilot.
And he was out of there.
Yeah, he's
Just
Careful,
a
Wyatt.
big
Can't see
anemone.
today.
It's one of those anemones we can't go any further than an anemone on. The big brown ones.
Ooh.
Oh, what is that? Oh, just
Some of
a
those.
little bit of junk.
No. I don't think so.
that was junk?
Starting to
think
get
it
a
was
lot
one of
flatter
those.
here.
It looks like the
Yeah.
Go ahead,
So
Bruce.
I think we'll let this move happen and then we'll start.
okay, if I
kinda
saw
making
that
our way
correctly, it
lateral.
looked like the snout was
Copy.
rounded and protruding above the lower jaw on that beardfish.
Let's just
and
look
that
off in the distance here.
probably
Is
makes
that
the
another
species
fish?
Polymyxia japonica, unless there's something undescribed
Lens
in
flare.
the area. But Polymyxia japonica is fairly widespread in the western Pacific and over to the Hawaiian Islands.
Yeah, thank you, Bruce. So I actually didn't know that the upper jaw extends a little bit more than the lower jaw. Is that what you're saying?
Just get a quick characterizing.
Once again, I
shot.
got it wrong. The snout extends over the upper jaw, protrudes out beyond the upper jaw. So I'm sorry, it's not the lower jaw, but oh, now I'm getting really confused. I'm not reading this correct. I have to go back and look at the images. Actually, it's Polymyxia burnti that has the snout protruding over the end of the upper jaw. So I could have reversed my identification. I should read these things more carefully before I speak. It could be Polymyxia burnti. So I'll retract everything I just said and have to go back and look at the photographs that we'll get from this.
All right, you want to get that starboard lower swing arm out
Just keep them
at a similar angle.
Okay, thanks, Bruce.
Absolutely.
Of course, we've never done that, have we?
No, we get it right the
never.
first
We
time.
get it right off the bat, and from great distances
That's
too,
right.
don't we? Yeah.
Like being able to say, look, that white stuff there is
Yeah.
sand.
Bruce, of course, just nailed that moored. Like he had.
He got
you
to
know,
see the
a couple
nose and
meters
he
too.
did, he get it right.
There's a
Co-pilot.
little fish right here. I don't know
Let's
if you
come
can
up a
get
couple
him.
meters here.
He's going really fast.
All right, pilot, we're almost at the end of that move.
There's something in the water
I think
column
we're gonna get him.
up in the upper
nice.
left. Or
Upper
upper right.
left? Upper
Upper
right.
right.
Okay. You want to zoom in? What do we got here?
So partial.
It's the jellyfish.
Okay. We can zoom
Hey,
out.
don't hurt his feelings.
Well, it's a gelatinous fish rather than a scaly fish.
Okay.
That's what we were.
But you
So
said
that move
just.
is finished up now?
Yeah, would we
we
be able
have
to
probably
image this
about
sea-pen
2
here?
meters.
And you can see
Do
the
you think we
substrate
already
has
reached the
changed
top?
considerably
The
as
flat.
we came up over the top of.
Sorry.
We're on top of this thing now, yeah.
All right, can you repeat now? Sorry.
Do you think we already
Okay.
Take a pause in
reached
the fish
the-
action
the
just to
flat?
get another
Yeah, I think
vertebrate
we're
we
going to
haven't
want to
seen.
start doing lateral here.
All right, let me know where do you think lateral is, and I'll
A lot of
see
sediment
the,
here.
I'll just the heading.
So the ship is stopped. So co-pilot, why don't you turn your head a bit?
10-4. Coming over.
And why don't you come up a couple meters here? Actually, they're looking better. But and if you bring your camera angle down to 35 degrees. Pretty good right there, yeah.
35.
All right.
Yeah, we've clearly gotten on top of this this big carbonate
Yeah, I can
block
see
or
30 meters
protrusion, whatever you want to call it, and we're now seeing all the sediment that's being shed from the atoll.
Yeah, Dan, before we leave
Yeah.
the sediment park, could you grab one of these sea pens? I mean, not grab it, but get a picture. It's
Oh,
over to the
well,
left.
we can grab it
Or
too.
down there.
No, I don't.
Okay.
Oh, which one do you want?
Yeah.
Either one of
Well,
them.
let's. I'm gonna work with my co-pilot just for a second to get a bearing on the next move, and then.
What's that?
It's like a piece of debris.
Yeah, that is not a natural. Oh, there's a fish in the corner there too.
Yeah, there is.
That's one of the beardfish.
Do you want to zoom
again.
fish first?
How about the sea pen? I think it's,
Okay.
well,
You
let's
wanna
give this
snap
fish
zoom?
a moment to
Snap
come
zoom.
up here.
Okay.
Yeah, it's another beardfish, so we
Same
can
thing.
go ahead and pass on that one.
Okay. Let's go ahead and zoom out. All right. So what's our general bearing toward that?
That would be
If you want to go zoom out to waypoint 2.
Copy that. We are. That's 313.
313. Okay, and I'm looking at like 305.
Nice.
Let's
yeah,
do.
That's as good as we'll get on that, and I think that probably spending a lot of time with other polynixia would be less productive than looking for
Go ahead,
other
Winch.
types of organisms.
Right, he's working with his navigator right now to just get squared away on the direction we're going to
Hold on a
make
second.
the next move on. That's why they're
What is
puzzled
the
for
workshop
a
saying?
Thanks,
Oh, Winch.
Chris. Yeah, I'd be interested in seeing what's a little further upslope and seeing if there's a greater diversity of
Yeah, I'm
fishes.
looking at like a.
All right, pilot.
Yeah,
so
we happen to be
that
in
would
kind of
be
a little
310. Do you want to
terrace
Let's
area
do
right
a
now. That's why it looks
300.
copy that?
Go ahead, Winch.
a little flat, but it's really not that flat.
Hearing a little noise coming from
Yeah,
the
it's going
area
to
of
quickly
the overboarding ship.
rise again.
In-
Creaking when we pitch forward.
Pilot,
back.
aside from the sea
Run
pens there,
Sirius
it
up
might
and
be a
down
good idea to
couple
take a
meters.
look
I'll
at what
make
this
sure.
piece of
I
debris is
suspect
here
the
that's on the
noise
bottom.
It's the
One of
shackles.
Yeah.
Yeah.
against
Tile.
each other, but I don't
Whatever that is.
bearing.
Yeah, Dan's thinking just
The
shackles
brick.
too here. Oh, come on.
Could be cement. Looks like
But
could
that
be
was
some
a
cement.
good observation.
Yeah, we're good. It's,
Deck, yeah.
It's just the shackles.
Yep. Thanks, Deck.
Thanks, Dave.
co-pilot.
All right, co-pilot. We're looking at like a 300. See if you want to line it up.
Yeah.
And we'll get a move in, and then we'll look at this C pen.
And that square object, maybe, if it's possible.
Okay. Yeah, let's get a.
Probably zoom
Let's
that
just
from
do
here.
a
30-minute.
30 meter.
Oh, okay.
at
30 meter at
300.
0.2.
Yes, please.
Copy that.
Yeah, I can see
Bridge,
up there,
there's
so
a
it's
nav.
Is that just a rock?
Let's get another
Very beveled
move in.
edge
That's
to
going to be 30
Very
meters.
Okay. You want to zoom in?
Bearing 300 at 0.2 knots.
Hold there.
Oh, it's got a little stoloniferin on it.
That is a strange thing.
Good copy, Bridge.
All right. Are you satisfied with that?
Yeah, I am, although I'm not satisfied with what we've figured out it is.
It doesn't
Okay.
look
Weird.
like the brick or
It
the metal piece
could
or
be.
whatever I thought it was.
Could be a piece of cement or something.
And it looks like the edges kind of broke off and make it look like it has this square pattern.
All right, well, thank you,
This
thank you,
sort of
pilot.
continues up there. That looks more natural.
Copy that, thank you.
The purple coral that's growing along it, I think, is a stoloniferin. And
Want to
it's,
zoom in? Hold there. Oop. Sorry.
these are really pretty.
Yeah.
They can,
The purple's in the middle.
yeah. So these, actually, the polyps arise
Yeah,
from, I
zoom
guess
out.
you could call it, well, it's called a stolon, but it's sort of a runner. And the
That's
polyps sort of
Okay, thank you.
arise as sort of
All right, let's find
a
one of these
line.
C-pens. I think I saw one over here.
Closed up Venus trap anemone.
All right, I see a C-pen just below us that I'll take a look at.
Okay.
And there's a shell fragment in the left bottom corner that looks like one of those pectin shells again, maybe.
See that lens smudge down there? See that, Levi?
Copy.
Try to notice that earlier this season. Do something about that in the off season. All right. All right, let's zoom in video.
Okay. So it's a partial.
Yeah, Nav, if you could right-click select waypoint 2 for me.
Copy that.
Okay, so what I think we're looking at is in the genus Panatula. It is a sea plant, pen, in the family Panatulidae. And I think it's something very similar to, if not the same species as Panatula inflata.
All right, you want to go tighter?
No.
It's probably a tough one to focus on.
Yeah, it's beautiful detail right there. Not very good at the anatomy of these things. It's too bad Scott France is not on the line right now.
That's my mistake to make.
There's the peduncle, right?
Yeah, that's the peduncle.
Really?
Yeah.
And
Let's go.
that actually allows this particular species to either live in the sand or it can live on top of the substrate or on rocks.
Anchor,
even.
holding it down.
Yep.
And let's see.
Top here.
Charles Wall saw a tube anemone somewhere. I don't think I saw it. Somewhere in the background there.
Fish is going to
Are
follow.
you fine with this view?
Yes, we
Oh,
are.
we got a
Thank you,
fish
Ben.
coming in.
Okay, let's
Zoom out a bit. Yeah, there he is.
another
Another
beardfish. So there was a. Let's see what Charles was looking at.
Should I come wider?
Yeah, let's go full wide.
Okay. That's full wide.
Thank you.
Getting sort of a mixed substrate zone. Sort of sediment on rocks and cobbles and shell debris.
Any interest here? Oh, there's
No,
something
we got
floating.
yeah, we got both of those. If you want to get the floater,
You want to zoom in?
that'd be good. 'Cause don't know
It's
what-
probably not the best lighting. See what we can do.
I think it's too close to focus on.
Oh, really?
Yep.
Ah. Yep. Oh, nope. Okay.
It bounced up against the lens.
Well, it was a stick figure, wasn't it?
Uh-huh.
So, that's what it looked like. I don't know what that was.
All right, co-pilot, we should have a few meters left on that move. Just 2 or 3 meters.
Yeah, pilot directly below us. There is
Okay,
a very
probably
strange animal.
30 meters to it, but we'll wait
let's
just a second.
wait.
Oral
yeah.
thing.
Yeah.
There's another
okay.
fish swimming towards us as well.
Oh, yeah,
Can you
there's
come up a
fish
few
coming
meters?
down there.
Yeah, another beardfish. See his little whiskers out. Bruce, you did order some other species of fish today than just the beardfish, right? I think he did.
He said he was going to put a smorgasbord in, I thought.
did, yeah. Yeah, I ordered some serranids, some basslets, and some dories, and some other things. So, but it's interesting. These
All right, you
beardfish
wanna
are
zoom
one
in?
of the most primitive of the spiny rayed fishes. So now we're at depths where we're picking up spiny rayed fishes instead of just the cusk eels and true eels and rat tails that we saw in deeper depths.
Okay, I was just checking, so that's good. You've got a little more
Get
sorted
lasers
order.
on just for a second.
Hey. So I'm waiting
You weren't
for the
happy with
chat.
the moored?
Yeah, the moored was good. That was good. I'm
Okay,
waiting
you
for
can
the
zoom in
chat.
tighter.
I have an
Okay.
idea what this is. I'm going to see if
So that move is finished up.
We can.
If anybody will confirm your idea.
It hasn't,
That's
but
not quite full zoom.
yeah, it is done.
Okay.
Yep. Looks like the
It's interesting
Ship's
how it's
vector's
much more
going around now,
three-dimensional
so.
than a lot of the ones we've been looking at.
Come on, Abby. You're in Les's lab. You should be able to fire off, and I guess at this one. I'm putting the pressure on her.
got you at 45. I can look down a little bit.
Okay.
Asako has identified it to family already. It's a chrysogorgid.
There's a little creature in there. You want to zoom in?
Yeah.
A little shrimp or something.
Yeah, it looks like a shrimp. Giving Abby a few more seconds. The clock is ticking down. It's getting close.
We need one of those sound effect panels so you can
All right, we can
dial
zoom
in
out.
all the sound effects.
Okay.
Click, clock, click, clock, click, clock.
Yeah. All right, time's up.
Very
This, I
wide.
believe, is in the genus
Thank you.
Rodaniridogorgia. And Nicole Morgan suggested Pseudochrysogorgia, but not really. Oh, we've got a different fish. Finally, there is.
To the right, to the
Yeah.
right.
To the right.
Yeah, to the
Yeah.
right. It's
Oh,
coming right
there
at
he
us.
is.
Here we are.
Ooh, that's a funny looking one. Look at that tail.
Yeah, you ought to wait till you hear the name. If you think he looks funny, you wait till
okay,
you hear
zoom
the
in.
name.
It's going downhill. Can you watch?
Oh, he did not like that.
All right, he's well off to the side now. Let's zoom out.
Okay, this is Bruce. Just for Chris's benefit here, this is a Gramicolepis brachyusculus. I'm not going to type that in. You have to type that in yourself. It's called a tinsel fish because the sides look like aluminum foil. It is a type of dory. A
Got you at,
xenophorm
38.
fish. There's
Think
not
about 'em a
dory like
Yeah.
Excuse
Dory in the movie Finding Dory.
There's a
Another
That's
30
a flat
meters
fish,
sounds good.
right?
Oh yeah, hold on. There's a
they're
fish
mostly
here.
in the deep sea, although there's some in colder waters called
Is that
John
goosefish?
Dories
Or-
up around Europe or off of
Zoom in.
Canada, that it can be found in somewhat shallower waters. So
Here.
I actually will type that name in. I just wanted to give you a
kill
hard time
lasers,
there for a
got it.
second.
You might like the next one too, Bruce.
Yeah, Bruce, we got another fish that's on screen right now. You'll see it any second.
So this is a goosefish in the family Lophiidae. It's an angler, type of anglerfish. You can see it's got a long whisker-like thing coming off of the nose just in front of the mouth. It'll actually lift that up, and there's probably a little flap on top of that that acts like a lure. A very broad mouth full of sharp teeth. You can start to see those now. They blend in. They sit on the bottom.
Go tighter.
Their pectoral fins, the fins on the side behind the head, are kind of modified so they're
That's
almost
not quite
like
full.
hands or short arms, and they can walk along the bottom. This is an ambush predator.
Yeah, was there
It
any
relies on
waypoints after
luring
waypoint
other
2?
fish in,
There is.
Okay.
With that
That's what I was
lure
looking
on the
for,
head.
but I cannot
They're
see
related
it.
to the shallow water
Hold on, maybe
frogfish
it's
that divers see. There you have a
I
good
don't think-
view
I don't know
of
if
the lure.
There it
There's
is,
actually
okay.
a couple
Yeah,
of,
there you go.
We're just going to follow this
modified
bridge. Okay.
spines.
Yeah.
There's several
Sounds good.
modified spines along the
So
back.
we're just kind of following the top of
And
this.
you can also see that it's got
yeah.
fringes all over the body and
Sounds
around
good.
the mouth that help it blend in.
Can you
with
go
the
tighter?
bottom.
We're going on to
Well,
what I think are teeth or something, whatever they are.
That's full zoom.
Shallower
Okay.
parts, they rely on the camouflage.
Yeah, it's about
To blend
400
in,
meters from
lure their
waypoint 2.
prey in, and then
Okay.
all of those sharp teeth in the
I think our
mouth,
average pace only had to be like
nasty
0.05
traps for small
knots today.
fishes
So
and shrimps and things.
So Charles Wallace pointing out, you can see the reflection of the ROV in the fish's eye.
well, that's cool.
Is that a fisheye view of the ROV then?
Ta-dum-tsh.
Unfortunately,
That
folks,
was
he
a-
will not be here all week anymore.
All right, let's
You
zoom
could-
out.
Okay.
Yeah, that's a beautiful shot. You can actually see the teeth behind all of those little
Yeah.
Or
fleshy
full wide.
All right,
projections.
let's do a
on
move. 30 meters.
Yeah.
Copy that.
This is related to the monkfish. that's
Bridge,
caught
this is
by,
Nav.
Commercial fisheries in the North Atlantic. And
Let's get another move
fish. The ones that we
in,
have
same
here
as
in the tropical
before.
islands
30
are
meters,
smaller
300 bearing,
that
0.2
distributed
knots.
support any sort of fishery. We actually don't know, if they're good to eat or not. They probably are, but they're smaller species. There are a couple of genera
Good copy.
that they could be, and I won't hazard a guess about that. It's just in the family Lochiidae, the goosefishes.
Are we full wide?
Yes, we are.
Copy bridge. Thank you.
I think we could just look up look up here.
Got a good delta.
There we are. There's the edge.
You're looking for fish, right?
We're looking for fish and corals.
Coral and sponges.
And sponges, yeah.
This one's
That's
swimming
not
over all the way on the
something
left
out there.
It's going to be another beardfish, I think.
Zoom in.
Oh, don't be so negative.
Bridge, can you give me an update on
That's
the
a
conditions,
different
please?
fish.
I
Yeah.
don't know.
That's
It's
different.
pretty far away.
It's a beardfish.
Oh,
It's a
okay.
beardfish.
Sorry.
Let's zoom out.
I tried, Bruce.
That's good. Keep on trying. And we'll hope to find more corals, too. And maybe we'll even get a token sponge for Chris.
I don't know. It's not looking like a very spongy place to me
Copy,
so far.
thank you.
It's too much sand.
Yeah, actually, there are sponges at this depth, but there's not that many glass sponges. There
Is
might
that
be
a
some.
giant clam on that rock? Or is that something else? Over on the left?
On the left.
Yeah, what is that? Or is that a crab?
You want to zoom in?
Yeah, quick zoom. Probably
Good
just a
video.
rock.
That looks like a. Well,
that has a
it's
crack
a
down
rock.
the middle.
It's a rock.
Okay.
It's a rock.
Okay.
Dan's not completely convinced. Okay,
we'll
fine.
We'll just make sure.
Go ahead and prove it's a rock.
Well, in the end,
All
I
right.
can still be right because all the rocks here are made out of shell and coral
All right,
fragments
zoom out.
anyway.
That's true.
Our full wide.
Thank you.
I'm fully covered today.
Looks like there's another fish in the center off in the
Oh,
distance.
yeah. Go ahead.
Beardfish. And that's not beer fish, it's beard fish. So
Yep.
I guess it is
All
still
right, zoom
Friday
out.
evening, isn't
Yeah.
it, in mainland U.S.?
yeah. So we're still coming up on a little less part of the wall. We haven't quite made our sort of northern turn.
We actually have. We're heading toward. What do we say? 300.
Oh, 300. Okay, I
yeah.
see now.
So we're starting to skirt up to waypoint 2. Still, we'll slowly come uphill, but it's pretty flat.
Reminded me, it is Friday here in Hawaii and on the mainland, so you have to find an octopus on this dive too.
Okay, we'll look. Yeah. You're now looking at the.
Nav, I would say let's keep a close eye on the ship and just keep the moves going. I mean, actually, let's range out to 50 meters on D2, and I imagine we're going to be able to keep moving here.
Copy that.
Yeah, it looks like a pretty good angle. Now that I see the track, that's a kind of nice intersection point. I can see
Right.
waypoint 2 now on the
Ship
map.
is
Okay.
underway.
So we're making a beeline, or we're heading right toward it.
Looks like another anemone on that little cliff edge.
Yeah, we got a little crab there too, right on the sediment, right in front of the ledge.
Crab.
All right,
down.
so if you zoom
No,
up,
on the
zoom up a little bit. He's backing up a little bit now toward the ledge to the
up
upper
on the
left.
dark ledge.
Up on the dark ledge. Oh, I see him. Okay.
Yeah, I don't think there's a hole there. I think you got him trapped.
I can move in tighter. The lighting seems pretty harsh here. Can adjust that after this. Let's zoom in partial. There it is.
Yeah.
Go ahead. Zoom in. Hold there.
I could take a guess at this, sort of. He's
I'm
missing one claw.
Yep. I'm going to go with crab.
What I'm looking at is the spines on the side of the carapace to
Come out
the
partial. I can get in
either
closer.
side of
Yeah,
the eye.
that's
There's
other.
a single spine.
So that's
And
partial.
a starfish too.
Oh,
On the bottom
yeah.
right of the screen.
Let's zoom in on the sea star.
Are you done with
Two
the
after-
crab?
Yeah, that's about.
Okay.
I think I got an idea for the crab. I just can't remember which is which. I'm looking
Okay.
it up right now.
Go ahead and zoom in. And hold there.
Yeah,
That one's like cookie cutter perfect.
Yeah, it is. In fact, that's what they're called. Is they're called cookie stars?
Copy that.
Believe it or not.
Is it really?
Yes, it is.
There's a little crab crawling
I
out of
don't
a
know
hole
if
below
Chris
it,
Ma is going to
really?
call in on this,
right
but
at the edge of the screen.
Yeah.
be one of two. This
Can I
could be
zoom in on
something
it?
called spheroidiscus.
That's full zoom.
Is it?
And there's a little crab underneath the rock there, yeah. Or is that even a crab?
Okay, let's zoom out.
All right, pilot. As soon as you're finished with this, I'm gonna put in
Still
another
trying
That's
to
movement.
full wide.
look up my notes on that
Okay.
crab
Yeah.
we just imaged.
What is
Looks
that?
like there might be
Oh, that's another
sea
sea star. This would be a good one to
just
image
up
too.
Okay.
from
Right.
Let's
Center.
hold off on that
where the
now.
lasers
Yeah.
would be.
Yeah.
You want lasers?
Sure.
The
Thank you.
crab could be something that we're calling progerion moose. Sort of sounds like shampoo or something, doesn't
Oh, I
it?
thought I was focusing on the moose part and thinking,
There
where's
you
the
go.
antlers?
Okay. Well, it's moose, M-U-S.
Oh,
Let's see if I can find a picture of the guy. Yeah, so that's the one that's got just very simple spination on the carapace next to the
Alright,
eye.
go ahead, video. That's a sea star, huh?
Yep, it is.
Go ahead and zoom in.
could also call it a sea lump. That does have sea star qualities.
Yeah, and it's a star that doesn't occur in Hawaii, and that's why I'm having trouble
That
remembering
looks very
it.
Zoom
similar.
out just a hair.
Yep.
Right there. That's good.
So I don't know if Chris Ma is going to be able to call in if he's on the line right now. I'll throw out an idea. It looks like it's Tree Master to me, Tree Master mirabilis.
And I was saying you're looking this up. What are you looking this up in, Chris?
I'm looking this. Well, actually, in this particular case, I'm looking at the Hawaii Undersea Research Laboratory Animal Guide, which has got an image of it. But we're going to include the sea star in the Okeanos Animal Guide this year because we got some in the Marianas.
That's right.
So this will be a new
Call
edition.
and move.
Okay, copy that. Bridge, this is nav.
So we'll
Same
see
move,
if Chris
please.
concurs on
30
that if he's
meters,
watching right
300 bearing, 0.2.
That's Chris Ma at the Smithsonian, our sea star expert.
Good copy, bridge.
Oh, what just happened? Oh, okay.
Sorry.
Oh, okay.
I don't know why I did that.
Sometimes the iris sticks, and you have to adjust it pretty far to get it to come back. I'm not sure why that happens. It's a controller issue. Oh, there's a pretty big coral above us.
And we're moving up to a slightly harder. Oh, that's a nice one.
Wow.
Moving up to a slightly harder surface, and immediately we see some corals again.
All right. This is a slightly larger version of something that we looked at down below.
Want to zoom in a little bit and frame it up?
Yep.
Okay,
How's
perfect.
that?
That's good.
So if I'm remembering this correctly, a version that we've got of this
Just a little
particular
bit
coral
heavier.
in Hawaii, it's got a lot of these lateral branches
fish.
coming. Oh, fish! Yeah, maybe we could. Any chance we can nail that fish now that he's quiet?
Oh,
Nope.
go ahead and
That's a
zoom
little
in.
one.
Do my best here. Hold there.
Oh, excellent.
And zoom in tighter. Yeah, there we go.
This is for you, Bruce. Dan said it's for you.
He's got something on his chin there.
I'm coming up a little bit for pilot, so just keep
Yeah,
an eye on it.
Oh, look at those teeth.
16 meter delta.
Got more zoom?
Of the fins on this thing, this is an
Look at those
unusual
teeth.
shot of something called a snaggle tooth. They're also called star eaters, an unusual name. That might have something
He's
to do with
drifting
those
away from me, video.
rings of dots
That
on
helps
the
with your
belly that
focusing.
are actually
It does.
bioluminescent photophores. Big fangs, you can see an impressive set of
Looks like he's got like a
large
little stick
canine
on his
teeth.
barbell.
It
Yeah.
has
Little
a blister on
red
the
down
chin,
the end of
a
it.
little white bulb at the end.
It's
and
not
closed.
backing down.
And
Yeah.
that
No,
uses
he's
that
Coming right at you.
he's not
in the
scared.
family Astronestidae. So I think that
Showing
you
off
may
for
have
you.
one of the few videos of a live fish in this family, but don't hold me to that because there are lots of other ROV and efforts on this.
Nice job, video.
It's it's a relation of the dragonfish, but unlike the dragonfish, it has a larger fin on
any
the
more
middle
zoom, or
of
is
the
that
back,
full?
and then a smaller fleshy
That's full.
fin back
Okay.
toward the tail on the top of the back over the anal fin. There are lots of species. They're identified by the pattern of those light organs and other characters that we probably can't see. Family Astronestidae, snaggletooth.
Trying to move in a little tighter.
Bruce, could you punch that into the chat? Because this is certainly the first time I've ever
And
seen
he's
that
gone.
fish.
All right. Zoom out.
This is a midwater fish, so it's
full
not
wide.
surprising.
Thank you.
Pilot bathymetry shows
Typing it in now,
the
you're
terrain
right,
not
it
being
is
as
a
flat
midwater
as
fish,
before.
and
Do you
that
want to
kind
keep
of,
going along the ridge, or
corroborates
do you
my
want to
idea
just
when we were
go
going down, we saw
uphill
lots of little
towards
fish in the
waypoint
water
2?
column. I think there's a scattering
How much left in
layer
this
of,
last movie called in?
of these fishes that move up
Should
and
be
down up to the sea surface at night,
about
down
14
during the
meters.
day. And I think what's happened
Okay.
is they normally move into deeper
Right
water,
now,
but a lot of them drifted
Waypoint
over
2 is
this
at
island slope and
318
then are blocked from their
Okay.
downward
Well,
movement.
we'll,
so they've
let's
accumulated
we'll let
here
this
on the island
move
floor.
finish up,
And
and we'll
it's
see where
thought
we're
that
at.
these
Copy that.
midwater fish that kind of
We
get
can
trapped
adjust.
on islands
Probably
provide a food base
Bring
for
it closer
some
to like
larger organisms,
310 or
larger
315
fish,
here.
and all on the deep
Copy
beach
that.
slopes of islands.
Yeah, that sounds good.
Great, thank you, Bruce. Any chance we can get these corals down on the left?
Sure.
And we passed one by, I don't know what it was, pretty big coral.
Yeah, we can look at it.
Astronectidae, I know that I've heard of the family. Okay.
All right, why don't you come up a few meters here, co-pilot?
Yeah. I got you at 48
I'll
right now.
try to do this quick. All right, sitting down. Okay, go ahead.
Co-pilot, about 2 meters left on this
I
move.
think this
Oh,
could
perfect,
be
thanks.
a bamboo. Could even be the
Zoom in tighter.
similar to the one that we imaged earlier.
So that's full zoom.
Okay.
Seems to be a bamboo.
Yeah.
Yeah, it is. I can see the nodes. I think it's branching at the inner nodes.
Some interesting anemones.
Yeah.
It certainly looks like it's branching at the inner node. And there's some sea
There's the-
anemones. One's closed, one's open. So,
All right. Is that a good view? Good enough view?
Yeah, that's beautiful.
Okay.
Thanks.
Zoom out.
Appreciate it. And I think that larger colony is probably the same thing as well. They're this
Okay. Ship
squat type of
stationary.
bamboo corals
pushing
down
out.
here at this depth.
What is this?
So these bamboo corals are
Snap zoom.
could be sort of considered to be.
Another sea pin. Okay, zoom out.
Oh, it's a different sea pan.
Oh, it's a different one.
Yeah, it is a different
We'll
one.
look at it.
Sure. A quick snap zoom on it. I think I know. I have an idea.
Sitting on a sphere almost.
Looks like ship move is complete, pilot.
Okay. Go ahead and zoom in.
So it's a partial.
So this is another thing that's referred to as a rock pen, and I think
And
its
zoom in tighter.
best guess would be something in the family Anthoptylidae, and possibly the genus Anthoptylum. And one of the things you'll note that's different about this one, the one we just imaged, is the
tighter?
other one had what are called leaves or branches coming off the main stem. Here, the polyps are coming directly off of the main stem, so it's quite a different morphology. And superficially, they look similar because of the color and the big foot, but they are quite different.
I'm going to zoom out a little bit and frame it up. Hold there.
Abby Laponte has come in, and she said that bamboo may be similar to the last one that we saw. And we'll cruise a little bit further. We'll probably see some more of them, but we're certainly keeping that in mind as a potential collection option, those bamboos.
I think I'm pretty much stopped, pilot. Should we line up towards waypoint 2, maybe a 315?
Yeah. Let me push out a bit here and we'll call it in. Why don't you bring your head over?
10-4.
Another pom-pom anemone that we imaged earlier. In the genus Leponema.
Do a snap zoom. It's up there.
Right.
Just a quick snap zoom. Dead stock
sort of a dead coral branch here. It's got some anemones on it.
Okay, can zoom out.
Oh, you closed
that
right
one,
up.
Yeah, that one's
that's
not today.
That's actually a piece of bamboo coral, maybe the same type we saw earlier. Oh, there's a bunch of stuff in the background, looks like.
Looks like another one of those sea stars.
How about first the one on the left before we get to the big one?
The sea star might run away before
on the left.
Yeah, I mean, sorry, right.
Oh.
That's it. To the
left
right of the laser.
The right part of the screen.
Right. Oh, that's going to be another bamboo. Maybe we can skip that. It's the same one.
Okay.
And we can go to this kind of reddy, larger colony, or there's others actually showing up. Oh, wow, we got a lot of stuff showing up right now.
Yeah, we'll just hold it here for now.
Copy that.
Yeah,
we're actually
we're stopped, right?
not moving.
Yeah, cool. All right, perfect. This is nice. All right, well, we got time. Chris, what do you want to look at?
I'm like a predator circling some school of fish. I'm trying to find a target here that I want to go after first.
Well, we got time.
Let's see. What would be, well, might as well, I think this ratty one down below is the same as the one on the right.
Okay.
So maybe we can go over and look at the one on the right that's a little better condition. And also has a better angle.
Yeah, we hit some real rock and immediately we see some corals again. Though the ones up top there may be having some sediment issues.
Yeah, these may all be the same thing, and even those smaller ones we've imaged might be just smaller colonies of this bamboo. I'm pretty sure it's a bamboo.
All right, go ahead, video.
It is fighting some sedimentation, isn't it? It is a bamboo coral in the family Isididae.
Got any more?
And
white coral on white sediment background, not easy.
Great for color photography, right, Dan?
Just making Annie's job a little tougher.
Well, good luck finding a dark background here.
It's hard to say whether it's nodal or internodal.
There you go. It's a little better.
All right, Abby. We're counting on you. Come on, Abby. Fire something out. An opinion. This the same thing? This is Abby Laponte. She's in the University of Hawaii in Les Watling's lab. And she studies this particular family of corals. One of the authorities on them, along with Les and Scott France.
Okay, want to zoom out a bit? Just shrimp. Okay, let's zoom in here.
Okay. So that's
Beautiful
full
little
zoom.
anemone growing on this dead coral. Really nice shot of the polyps too, of the coral itself in the branch.
All right, is that enough for you, Chris?
Yeah, that's
All right,
enough.
zoom out.
Thank you.
Okay.
And what's next?
We can look at these other white ones. I think they're all the same, but we can just sort of move through them and just check to see if any are different. And these are Hormathiid anemones that are closed up, those sort of pillar-like things. We got a close-up of an open one a little earlier.
We've got something
Pom-pom
flat
anemone.
and different
I'm
colored
not seeing
behind
anything
the open
new right
anemone
in
on
front
the
of us,
left.
Dan, at the moment.
Oh, is that another?
Oh,
Another
another
fish.
One of those
fish.
fish,
Yeah.
yeah.
Got
Pom-pom
something flat
anemone.
and different
I'm
colored
not seeing
behind
anything
the open
new right
anemone
in
on
front
the
of us,
left.
Dan, at the moment.
Oh, is that another?
Oh,
Another
another
fish.
One of those
fish.
fish,
Yeah.
yeah.
Well, Ken Sulek joined us if we want to maybe do another.
Yeah, we'll get a group shot with the anemone, coral, and goosefish.
Oh, there we go.
Nice composition.
The triple crown right
Yep.
here.
Frame it just right.
And actually, that anemone could be different than what we looked at earlier.
Also got a yellow coral under my right toe.
Probably Samia, if it's kind of a hard coral. Does it look?
I'm not sure. It's pretty small. All right, you want to zoom in
All right.
and hold there. For now.
He's standing on his feet.
Oh, he's getting.
No, he's getting ready to jump.
That is he.
And away he goes.
Nope.
Go ahead, Bridge.
Doesn't want to be in the photo.
Yeah, but that's okay. We saw one still. Now we get to see one actually swimming. It's pretty nice to get both.
Zoom in. See his legs.
That's not quite full.
Turning
Yeah, that's interesting.
starboard
Usually
side,
you don't see them
20
quite
degrees
that
to-
active. That's a lot of activity for one of these
40
anglerfish or
degrees.
goosefish. They usually rely on their
Copy that, that's fine.
camouflage to just blend in or not be seen. I mean,
You
he's
say
not
it was
that
winds or
camouflage
current?
with these bright lights on him, but with dim light, he would be. And so they
be
usually
winds.
don't move around that much. So that was a little bit unusual.
winds
Do we have the
shifted
lasers
a
on
little
just to
bit
get
now.
an idea of his size?
Okay.
We can
Oh, lasers on, please.
Lasers coming on.
And zoom out a bit.
Here you can see
There
that
you go.
long
There they are.
lure is lifted up a little bit off of the back, extending back from the snout across the over the top of the fish, which wasn't as apparent in the previous
Okay,
one.
and zoom out.
You can also see the lateral line organs on the
Sort of
side,
stuff.
these little bumps that are vibration sensing that he would use to pick up movements of small animals in the vicinity to find prey.
I'm just going to back up. You want to take a look at this
Thank you,
yellow
Chris.
coral that I see real quick?
maybe a quick look. I think it could be something we already imaged just from your description, but
Okay,
let's just
I'll just
confirm.
back up and we'll
Yeah.
get it into frame. That one there.
Yeah, it's sort of a busted up Samia rostrata. We got it earlier. That's what I was thinking it was.
Okay.
Probably wait for them to shift their head and then we can get a move in.
Yeah, so Andrea Quattrini just
Hello.
joined us as well. Yeah, Andrea, we imaged another one of these fish earlier on and got a marvelous close-up of the lure and eyeball. We actually could see
use the
the
skids
ROV
video?
in the fish's
Oh, is that-
eye to
You
give
can
you an
use the
idea of how
skids.
close it was.
Bridge,
Yeah,
does it
Andrew,
nav?
just reminding me though, it's one of the important things. Is the
Let me
tip
know
of
when
that
you're
lure,
completed
whether
with
it's
that
got an
turn.
expanded flap and what that flap looks like, and also if we're
Good
getting
copy.
to look for that, it'd be kind of a small feature, but that could be important for the identification.
Yeah, I was looking at it, Bruce, and it looked like it had a white, rather small ornamentation. The lure itself was just a very white,
And
kind of
snap zoom on
fluffy
this
tip.
coral.
Okay.
And this is the same stuff we've been seeing, Chris.
Yeah, it's just more of
So
this
that could be a
bamboo
useful
coral.
Okay, let's zoom out.
Okay. Oh, look at this. Looks like we've got some karstic formation
Yeah, we
here.
definitely have some hollowed out formations here. So this is definitely weathered out limestone. So the limestone was formed by carbonate deposits from the ancient reef, and then that got weathered out. That process is called karst formation sometimes.
Anything of interest here?
No, I don't think so. Could we look upslope just the hair?
Sure.
See what the upslope looks like?
Towering.
Lip.
Yeah.
I think we can get a move in now.
A lot of little crustaceans in the water column.
Pilot,
That's for sure. A lot of food for things.
315 as we talked about before.
Yeah, I think 3-1. Let's do 310.
310.
Yeah,
Some more of these tree master
Alright, do you want another
sea stars
30 meters?
on the
Yes,
seafloor. These kind of
Can they move while they're changing heading?
type stars.
Just 95 right now.
Are they still changing heading?
Yeah,
Okay,
they'll get back to me.
Why don't we wait? Yeah.
Yeah, they'll get back to me when they're done.
Okay. Hey, Chris, the ship is making a heading change to equal out the forces more up at the surface, so we're going to wait for them to finish that up before we call another move in. So we got some time here.
Well, I'll just throw this out. It's a bit early to consider collections, but do we have any vote for this bamboo coral?
Come up
There's
some
any interest in this, or shall we just wait for something something else? Since we're sort of holding here. Michael Vecchione, by the way, has joined us, as has Ken Sulek. Michael Vecchione is at the Smithsonian. He's expert on a lot of midwater animals, including midwater fishes. And Ken Sulek is sort of a mainland version of Bruce Mundy, expert in a lot of different fish too, and he's been extremely helpful for the cruise. Let's see. Andrea Quattrini, by the way, I already mentioned. She's at Harvey Mudd College. And I think that's it from the folks that I already... there's a coral straight ahead that's a little different. You just one right on the edge of the rock here.
Okay.
Dead center.
You want to zoom in? Can't really get too much closer.
So that's not quite full.
Just drift in until I get to the end of my tether. Cool urchin there.
So urchin,
yeah,
the one
that's
we saw earlier.
Yeah, the Cynopodina pulchella. This coral is... What is going on with this coral?
Zoom in tighter.
It's got
hold there. Oh, tons of brittle stars.
Yeah, that's what's going on with it. It's got a severe case of brittle star. Wow.
Touch my toe. Let's zoom out a bit. I don't want to run over
If you
these
collect
corals
this, they are
anyway.
not going to be individual commensals, Chris.
Come all the way out?
Quite,
this is fine.
Okay.
Quite frankly, I have no clue what this
Just
coral
drifting
is
over,
either.
but I noticed some corals under my toe, and I didn't want to.
I don't know, Asako, do you have an idea what this coral could be, or Abby, or Tina, maybe? Any one of you have an idea of what type of coral we're looking at?
That's full zoom.
Okay. Almost, boy. Come on, Chad, I need some help here. I'll take even wild guesses that could be completely wrong.
I'll go with coral.
A ship dollar? If I put a ship dollar out there, do we have any takers for a ship dollar? No.
I'm sure there's plenty of people that will come up with moon waffle coral.
Yeah.
Zoom out a bit.
No, we've gotten that far rolling. That's cheating. You know,
Zoom out.
I like Asako's comment, sort of, well, dot.
Got a little bit of tether here.
It's an oak tree.
An oak tree. Okay,
there's
so octocoral it
Oh,
is.
fish in the right.
Oh yeah, let's get the fish.
Looks like an eel, maybe.
Yes, it could be.
All right, let's zoom in.
Okay,
Bring it
pilot,
over.
we're almost at the end of the turn. I'll wait until you're
Okay.
done with the fish, and then I'll put in a move.
Okay, sounds great.
Well, Chris, it looks like this has a flattened snout, long flattened snout. Is that what you're seeing?
Yeah.
The eel, I'm already writing in the name, Bruce, but I want you to go ahead and blast it out.
Zoom in tighter.
Okay, well, this is a
Hold there.
duck-billed eel in the family Netostomatidae. It's a different family. It would be really important to zoom in on the head and also the top of the body just behind the head, because there are a couple of genera of
Trying
these,
to keep
and the
it stable
position
for
of
your
the
video.
nostrils is really important for identifying them.
Zoom out a
The
bit.
posterior nostril,
Hold it there.
some
Yeah,
of them
there we go.
actually have that nostril behind the eye, and some of them even have a posterior nostril on the back of the head, believe it or not. So he's not cooperating. It looks like this is more anterior. I'll have to take a look
Copy
at the
that.
notes
Stand
and
by.
see
Nope,
if
another
it's
fish.
Metastoma parviceps or
Fish
one
zoom
of the
out.
other
Yeah,
species.
let's-
There are more species in the western Pacific than
That'd
there
be
are.
fish.
Yeah, let's stick with the eel,
But a
please.
duck-billed
Okay.
eel, and that name comes from a pretty obvious reason. In the family
As I move
Netostomatidae.
back towards you,
They're the
co-pilot, you might
beardfish
come up a little bit.
polymyxia,
10-4.
photobombing the eel.
So, Bruce, this sure looks like Netostoma parviceps to me. Would you agree based on that tail? Because that's the only one that I'm aware of that's got that really flowing, well-developed dorsal fin, caudal fin, whatever you want to call
I'm
that.
ready for that move.
Should I come
When
full
you are,
wide?
yeah, let's come full
Yeah, I'm
wide.
going to be a little cautious.
That's why.
after,
It's like I thought we were
jumping in too soon with some of my other
zoomed out further than
identifications.
that,
Do you
sorry.
remember parviceps
I was like, why
has
isn't the
those
camera
very
tilting
dark
down?
borders on around the fins?
Yeah, it had very dark borders, right? Let me see the pictures of parviceps we got in the gallery
Ready for a
here
move,
to
Pat?
see if it also
Yeah, let's
has
do
those
it.
borders.
All right, putting in a move 30 meters, 3110. I mean 310.
310.
Bridge, this is an F. Let's get a
Wonder if
move
we
in.
got,
That's gonna be 30 meters at bearing
Netostoma parviceps last
310.
year on the Okeanos.
Speed
I don't
0.2.
think we did. So I'm going to go ahead and look at the Hurl Gallery,
Good copy.
the Hawaii Undersea Research Laboratory Gallery. Let's see here. There we are. So, Bruce, the one that we've got image in the Hawaii Undersea Research Laboratory Animal Guide does have a black margin along the fins, like the one we just saw. It's maybe a little less extensive, not quite as dark,
There's a
but
fish in the upper left.
it's there. I don't know if that matters, but
Go ahead and zoom in.
It's another beard
it's a
fish.
beard
Yeah,
fish.
so the reason I asked to zoom in on the head, the
Go-
reason I asked
Oh, it
to
doesn't
zoom
have
on
a
the
beard.
head was that the genus Nettostomatidae
no,
has
it
a
does.
posterior
Alright, zoom out.
nostril behind the eye, or even back further past the head.
Come up a little
And the
bit.
genus
Down.
Netostoma has the posterior nostril on top of the head, in front of the rear margin of the eye.
Another fish.
That's a different fish. I wonder if we could take a
Zoom
look
in.
at that
Quick.
one.
Ah, sorry.
Really? Yeah,
little fast.
he was zipping. Oh, he's turned.
He's still
Go ahead and zoom in.
going
Yeah, if we
front.
could get him,
Hold there.
he is ripping along.
Is that the same one we saw earlier?
No, that's
No.
a different one.
You don't think that's the one with the fangs?
Nope.
He's cruising.
Well, maybe he
Swimming
is.
towards us now.
I don't see his barbel.
Well, that was a very tiny barbel.
No, it was big actually. It was sticking right out there. It's just-
Alright. A little more maneuverable than I am. Alright, let's go full wide.
Yeah, but I think this is another snaggle
Full
tooth
wide.
in
Thank you.
the overall morphology. We can't see this.
Okay,
clearly
co-pilot,
in the
we're
last
moving.
One, but it looks like
Excellent.
the same
Thanks,
body
Nav.
shape and all. And I made a correction in the chat room after I talked so much about these. There was a clonistic analysis of the group of fish, the dragonfishes that this is in. It's actually placed in the family Stomiidae with all the other dragonfish. Snaggletooths don't have a formal ranking, but they're often considered to be a subfamily now, the Astronestinae in the Stomiidae. A little bit of...
Yeah, pilot, there's a gold coral that's over to the right off screen if we get a
Okay.
chance.
Yeah, we can look at that.
Yeah,
We'll
thanks,
move in
Bruce.
that direction too.
There is.
And there is a subgenus in the genus Astronestes of those snaggletooths, the subgenus Stomianodon, which is thought to have species that do associate with island slopes and vertically migrate up off the slopes and then down back near the bottom and aren't found out in the open ocean. It's they're part of what's called the mesopelagic boundary fauna.
Yeah, thank you, Bruce. So that's the voice of Bruce Mundy at the NOAA Fisheries Science Center.
That's a partial.
And he was referring to, he's doing some follow-up on a fish we just imaged that went speeding off before we got a really good look at it. So this coral is tipping over. It actually has come loose from, its holdfast has actually come loose from the substrate a bit. But he wound up leaning against another coral, so he's still upright. And if we could zoom a little tighter on this, yeah.
It's not quite full.
Okay.
Give it full.
All right, Asako and Tina and others in the chat, you have a chance to redeem yourself after that that one white coral, which we didn't get a whole lot of help on. Yeah, here it is. All's forgiven if you can identify this thing. Asako says it's a plexaurid. Okay, you're redeemed, Asako.
Okay. We can zoom out.
Okay.
And let's see, Diva Monde had noticed that there was an aplecophoran on the side of that, so that's pretty interesting. That's
you want to go back in and take a look?
Sure. Yeah, if we got some time here. This is a type of mollusk that doesn't have a shell. Has a very worm-like shape.
Okay, go ahead.
Let's see, it was down a little ways when you were zoomed in tight. Where are you? Oh, there it is, to the upper left right now, that thing that looks like 3 rings on the branch.
Oh, yeah, I see it. I'm going to zoom in. Hold there.
That's
I thought that was a brittle star arm.
Yeah, and but I think Diva is right. I think this is one of these aplecopherans. There's a brittle star right below it.
Interesting. So it's just a worm?
No, it's actually a mollusk, if you can believe that. It's not a gastropod. It's in its own group, own order.
Okay, co-pilot, we're pretty much at the end of that move.
Okay.
Now the kind of stranger inhabitants down here.
We'll
that's
push out
great,
just a
thank
little
you.
bit, and then we'll call the next one in.
Still looks like a worm.
There you go. We're stationary.
Sounds good.
So now we're cruising right along the steep drop-off of this feature, which we were hoping to do today. So this is great, finding all types of stuff.
That's rather large.
Yeah, maybe the same type of plexaurid we saw earlier.
When our oxygen has gone up to 2.4 now, and our temperature is a balmy nearly 7 degrees Celsius.
If we could zoom up just a hair, pilot.
You want, tilt up?
Yeah, or tilt up, I mean, not zoom up. Yeah, that doesn't make sense, does it? Tilt up. These do look like plexaurids to me. I think they're the same
get
thing.
another move in.
Yeah.
Copy that. It's going to be the same move.
Yep.
Bridge, this is nav. Same move in, 30 meters, bearing 310, speed 0.2. Good copy.
Another pom-pom anemone, more of the tree master sea stars. That's that sort of little white and orange disc that we imaged a little bit earlier that's just above the lasers.
1 and 2.
Yeah, a couple of them. This gold coral is a plexaurid. soft Gorgonian sea fan. Those of you just joining us, perhaps from the Maui Ocean Center on Maui or the Waikiki Aquarium in Waikiki or some other location, this is
Rich,
a live
can you update
broadcast
me
from
on
the
the
NOAA ship
payload you have up there?
Okeanos Explorer. We are diving today on the east side of Wake Island in the Wake Unit of the Pacific Remote Islands Marine
Good copy,
National
Rich.
Monument. This is one of our shallower dives. In fact, it's the shallowest dive that we're doing on this cruise. And we are at about 554 meters below the sea surface. We are on the hunt for precious corals and fish and anything else that we can see, because the fauna at these depths anywhere in the Wake Monument is completely unknown.
Another one of those pin cushions.
Yeah.
That coral on the right
So
looks a little different.
I don't know if you can get to it, Pilot, but there's sort of a yellow thing that just went off to the left.
Yeah. Okay.
And I believe this is something we haven't imaged today yet. That yellow thing right there with the white
below?
above.
Okay.
Yeah, the lasers are slowly getting to it.
See it?
This is different than the Antilopsamia that we saw
Zoom in.
earlier.
Hold there.
That's it.
All right. Do you want me to sit down and get a closer look, or are you happy with it? Oh, look at that.
Oh,
What is that?
that thing,
Zoom in.
that grabber sticking out of the.
Yeah.
What?
Hold there.
It's this gray grabbing thing. It just latched for something.
We've got a little bit more zoom.
Doggone it. Well, I see
Go
it
ahead.
now, but I didn't see the action. I was writing in my notebook.
Zoom in.
It just went
What
for something.
is,
That's
what
full
is this
zoom.
thing?
Slowly dragging back in near the little hole it's coming out of.
Is it
Just
one
set
of these
my toe
spaghetti
down.
worms or something? But it tried to
It
tried to get
it
a
literally
sample.
went
Hold
for
on. Let's come out
something.
video.
Yeah.
Really? Wow.
We're full wide.
Okay. I'm just gonna get a little closer here.
Yeah, let's take a little closer look at that.
I'll see it. I'll try to sit down, and maybe that'll cause them to retract again. All right. Let's zoom in. Partial. Let's go in more. Hold there. Bump. He's not feeling it.
Still pulling back in a little bit.
So Deefa on the chat is commenting that there's only one tentacle, and that spaghetti worms usually have more than one.
We can zoom in.
That's full.
And it's still very, very slowly dragging back.
I'm going to zoom out just a hair. Right there, yeah.
Yeah.
He's not going to give us another show. Here.
There's something swimming out of there.
All right, I just asked Emily, the clipper here, to do a rewind on that. I didn't see what had happened, so I did get to see it. It was hard to say whether it was going for that little crustacean or whether it was retracting when the crustacean came out of the hole. This is completely bizarre. I don't know what we're looking at.
In the rewind, it looked like it came.
Oh, there it goes.
Yeah, it goes
Yeah.
back in.
Look at this. I'm not so sure anyone has seen this before, this type of animal. David commented that the spaghetti worms have more than one tentacle. This
This
just
is
has
full-
one
oh, no.
tentacle coming
That's full
out
zoom.
that bifurcates right at the tip. No, we don't know what it is. Yeah, Brian Kennedy just joined us here.
Ship stopped. I'm looking back at you.
All right, ship is stopped though. It stopped a while ago, right?
It just stopped.
Oh, okay. Yeah, it'll be all right.
It's also these spaghetti worms, if I'm not mistaken, are often in sediment, not
Copy
in
rich.
Rock, and this is just
That's
coming
my
out of a rocky
max
hole.
camera angle.
Okay.
You should see view.
So who knows what
I'll
the
be
rest
able to
of
push
the...
up pretty far.
So
pretty hard.
Andrea Quattini says, we've seen this before. So Andrea, did you ever figure out what it was before?
Can't remember the
Can't
name.
remember. Well, it's a first for me, that's for sure. I have not seen this. Amanda Demopoulos might know. Okay, hopefully somebody can pass her a picture of this.
Deb is pretty sure that it's not a rock.
All right, let's zoom out.
Okay.
So I think I'm going to go with the same
the tether shift
the
a
same
bit.
view here.
So we're full wide.
It's inside one.
Yeah, tether should be pretty far above you. So
I don't even know what to
Footballs
call
to
it
keep
in
it
my
up.
little notes here. Weird bifurcating
Snake tongue.
snake tongue, yeah.
that was interesting.
That was cool.
Bifurcating
Yeah.
blue-green worm.
All right.
I like
Pushing
snake
out.
tongue.
Yeah.
Until somebody comes up with a real name, I'm calling it a snake tongue.
So before we got distracted by whatever that beast was, we were zooming in on what I think was an Iguchi samia, a hard coral, and that would have been the first observation of that we've seen today. Jasper is just headed down to lunch to get a bite to eat, and that leaves me free to say anything about the geology that I wish, whether it's true or not. Maybe I'll refrain, because I guess Deb Glickson is in the chat. She'll probably hold me accountable, so I'm not going to say anything. I'm going to wait till Deb makes some remarks, and I'll pass them on to you. What could we look at this? This looks like some ordnance, frankly. Could take a look at it.
Yeah, let's zoom in. Hold there.
I don't know if it's ordnance, but it's man-made objects. Yeah, I don't think it's ordnance, but it's. Thought I saw some fins there.
What do you think that is?
Here, we can get a side view of it.
I mean, those are fins for
Those
sure.
are fins,
Actually,
yeah.
it could
Yeah.
could be ordnance. Come to think of it,
Yeah.
or some type of a flare or something.
You can zoom in further. Hold there.
Maybe we could drop a mark, a navigator on this location.
Okay, copy that. Watch lead.
Zoom out a bit and frame it.
Zoom in or out?
Zoom out and frame it.
So it's
Yeah,
got a
right
little ring on the tail end of it too, so it could be something like a parachute flare or something like that. So it actually have a parachute, and it almost looked hollow between the fins as well. But I'm not positive. Can And Brian Kennedy has just come in and identified that weird creature as a green spoon worm or something similar. But let's see, let me get the
Zoom out.
name on that if you don't mind me
Or full wide?
scribbling. Spoon worm. And Bonelia viridis.
I'm lined up on 310.
Okay.
Yeah, these are a deep water. Yeah. Oh, I think you nailed it, Brian. Thank you very much for that. Maybe that's what Andrea Quattrini was thinking of, a green spoon worm. There is a sea star to the upper left we haven't seen yet. I don't know if that'd be possible to take a quick zoom of that.
Sure can. All right, this one coming into the lasers now.
That's correct, yeah.
Zoom in. Hold there.
So it's a Gunny asteroid, cookie star.
Am I trying to keep you in view, or
just hold it.
you're gonna come back?
Yeah, we're at 310, right? Is that bearing?
Yep.
Yeah, I'll come back. You can zoom in tighter.
This is our test to see if Chris Ma is watching right now. So they're called cookie stars because they have literally like a Christmas cookie type of shape to them. Those of you who celebrate Christmas and have done some homemade cookies, this looks like one of the star shapes that you typically find in a Christmas cookie cutter set.
Zoom out.
Okay, pilot, let me know when you're ready for the next move.
Okay.
I'd be okay with it. Or you want to saddle over first?
We'll saddle over. Oh, interesting. Let's see what we got here. I think we maybe come up maybe a little bit less of an angle to the contour here. Just because
think you're coming a little more westbound.
Yes.
Okay.
Like, do you mean this way? Or
yeah, let me why don't you come up left. Wow.
Andrea and Amanda Demopoulos both confirmed that this is an Echiurin spoonworm. So yeah, they're all agreeing with
let's bring your heading
Brian's
to the port, co-pilot.
Investigation on the web as well.
And why don't you come up as I back toward you?
10-4.
That was Brian Kennedy again who came up with that. Thanks very much for that. So this is
the
rock.
current really picked up, and you can see stuff whizzing by here now.
Yeah, and the rock looks like it's almost lithified bed forms, but you know that's a sort of a
Good
prompt for
delta,
Deb
facing
Glickson
west.
to maybe put in the chat what we're looking at here.
Yeah, I think let's come. Yeah, we're basically facing west, huh?
You want to do a hop west and then
Yeah, let's do a 270.
then go north? Follow that ridge?
Yeah.
Okay.
okay, copy that. Do you wanna get to the like around here?
Yeah, let's just do 30 meters.
30?
Yeah.
All right.
So we're sort of looking at the shore side of this feature. That's to the left, the port. There's a little bit of a depression in there, and you can see
Bridge.
it drops off
This is nav.
quite a bit on this side.
Another move for you. That's 30 meters, bearing 270, speed 0.2.
There's something odd looking.
Good
up.
copy.
What do you see?
Straight ahead to the left, a little bit up against the.
Oh yeah, up there.
Yeah.
Yeah, there's a fish up ahead,
Is that a fish?
and there's also
Yeah.
something
Yeah.
big in the water
Some more
column,
here.
a jelly or
Yeah,
big
there's something in the
jellyfish
water
in it
column just up above
as well.
the
Chili.
camera right now.
The fish is the
Trying to give you some
Grammacolepus,
more
I'm
leash,
pretty
pilot.
sure,
Okay.
and we'll get a view of it. But this jelly is pretty amazing up here. I don't know which is closer.
Zoom in. They're both pretty far out.
That's
It's a big ctenophore, I'm not really sure.
Is that all you got?
No, I got more zoom, just no more light.
So
Yeah.
Yeah, I don't know what that is. It's larvation, maybe, not sure. While they're zooming out, I want to read to you some information that Diva Amon has provided about that weird worm, the Okeanos worm.
Fish.
Okay.
Go ahead.
Yeah, there's a couple of these fish in there, the Grammacolepus, we believe. So those worms, apparently they all settle out, the larvae, on the seafloor as females. And then as they grow, then if a larvae lands on one of these females, it turns into a tiny male that stays with it for life. So that's pretty fascinating. Thank you, Diva.
I know they're real far away, but...
Now these tinselfish, Grammacolepus, are pretty well known. It's a species that occurs all the way around the world, except maybe for the eastern Pacific. Deep bodies, but very small mouths. If you look, they're planktivorous. They pick plankton out of the water column, things like copepods and other small crustaceans.
Sorry for wide.
They have this highly reflective appearance. The scales are elongate, vertical, and gives the fish a very reflective, almost mirror-like appearance.
Yeah, thank you, Bruce. This little white thing here, this is another animal that we haven't seen today. It's kind of transparent and to the upper left.
Yep.
Alright, co-pilot, distance to waypoint 2 is about 100 meters.
I think all these little stubbles you're seeing all over the seafloor might be bases of this Aguchi-Samnia coral. That there's only a few colonies that are still alive here. That's my guess, but could be wrong.
How far have we moved so far now? Just curious.
We have a sponge. Yay, there's a sponge here.
We still have about 13 meters left on the move.
I think we can
add another 30 or something.
I'm thinking.
Do you want to be south of?
Let's go ahead and hold it. Well, I was just thinking we could go ahead and stop the ship here and then readjust.
Okay, copy that.
That just gave us a little bump up the hill, and then we'll be able to follow the flatter area up to waypoint 2.
Copy that.
Zoom in.
Great, this is nav. Let's hold position here.
Is this what you're talking about, Chris?
That's exactly what I'm talking about. Thank you. So
go ahead and zoom in.
Good copy, thank you.
This is actually, even though it looks like a glass sponge, it is not a glass sponge. I'm pretty sure this is a demasponge.
It's not
and
quite
I think
full.
it's in the genus Poecilastra. It forms these ribbon-like colonies at this depth, but also much deeper as well. And we always thought these were glass sponge or hexactinellids for years until we finally grabbed one and took a look at it, and were utterly surprised to find out that it wasn't. And then behind it, the yellow coral is another one of these Aguchi-Samnia, these hard corals that we were zooming in on before that weird spoonworm showed up.
Just going to change the setting again.
Yeah. Do you want to do it before any move?
Does he need to change the setting now? Did he say? Just how urgent it was.
Lots of texture on the seafloor behind it as well.
Yeah, let him go ahead and put his heading change in, and then we'll move.
Bridge, this is nav. Go ahead with the turn, with the heading change. Let me know when you're done, please.
Yeah, that's good. Thank you, pilot.
Okay, let's zoom out.
All right. We're full wide.
We've seen a lot of different types of animals today. Really, I've got 1.5 pages of notes already.
I'm going to come to your starboard co-pilot.
Copy that.
We'll get readjusted here.
You want me to turn with you?
Yep. Yes, please.
Sounds great.
Teeth and all.
There we go. There's some interesting stuff there.
Something in that hole.
Looks like an anemone.
Yeah, maybe it is an anemone.
Take a look.
Standing by.
Just like to look at these bigger holes because they're often being used as shelter by fish at these depths.
There's not much light in there, but
it's wow. Well, it's either an anemone or a corallimorpharian.
Tilt down just a little bit more.
Or maybe it's a hard coral, a scleractinian. It could be the calyx there that ribbed those ribs on the top part. So it's upside down. It's actually attached to the roof of this
as we
little
get going again now.
cave.
It's all right if we end up slightly west of waypoint 2 to keep us like.
So Asako believes it's a scleractinian, and I actually would agree with that as well. Not sure if anyone else wants to weigh in on what we're looking at.
It's a nice
here.
shot rolling.
Yep.
Basically no light.
Yep, if you can give me a little more tilt down, we can lose that hot spot. I can bring it up some more.
Yeah, it's a beautiful image of the nematocysts on these tentacles. Yeah, Charles Wall also agrees with Sako that it's a scleractinian or a single cup coral. Great, thank you.
All right,
that'll do it. I think that's the best we're going to be able to do with that guy.
All right, pilot, if we want to be about 20 meters east of waypoint 2, that would be...
sorry, west. So we'll be uphill, slightly uphill.
Oh, uphill,
yeah,
okay.
Which is totally fine, if we need to. Yeah, like right there's fine.
All right. That would be bearing 321 from
those of you not
Sirius.
On the chat, a little update. We're at 534 meters. Temperature is at 7 degrees centigrade. Our dissolved oxygen is about 2.7 milligrams per liter. I don't know what that was that just drifted by. How about that, right where the lasers are?
Sure.
This is a different fish.
Go ahead and zoom in.
He's facing, unfortunately, right into the hole.
There.
Okay, I've got an idea, Bruce.
Yeah, well, this is the back 3/4 of a scorpion fish, and it's, could say it's not a pontinus, but there are a lot of species of scorpion fish, and they're very difficult to identify. Although, from what I can see, this could be something called Sitarches, but I'm not sure because we're not seeing the whole fish. Do you have any thoughts, Chris?
You went exactly where I was going because of the size of the pectoral fins, the body shape. I think it looks like, I mean, it's not quite the same as the Sitarches guntheri in Hawaii, but that's where I was going as well, Sitarches.
Yeah, and there are a couple of species of Sitarches over in the western Pacific. We only have one in Hawaii, but I think there's also something called Sitarches longimanus.
Yeah, hold it right there.
Which means, longimanus means long hand, and that's in reference to the prolonged pectoral fins, those long fins on the side that we can see. I'm not saying that this is that species.
Yeah, just turn thrusters a little bit, Pat.
It's a good guess, Bruce.
Yep.
It's still a good guess.
Change still, or is he?
Big head?
I said it's still a good guess, Bruce.
Yeah. Anyway, I'll leave it at
10
that.
knots of wind.
Yeah, thank you. I'm going to sneak downstairs for a couple minutes here and grab a bite to eat, and Jasper's going to take over for a few minutes.
Yeah, let's call up to the bridge and ask if...
Because I already ate.
Should be about 15 degrees more, so it should be about a few minutes more.
Oh, so he's still moving. Okay.
Yeah,
all right.
still turning.
Yeah, we'll leave it then.
Turn his head, okay.
Okay. All right, let's zoom out video. Thank you. All right, let's keep coming.
Copy that. I think the magic number was 322.
Do you want me to push past the skids, or?
I'll tilt up a bit.
Okay, copy.
Yeah, and we're looking for fish, so I'll help with that.
Try to give you a little more tether, but
yeah, let's do a 3. Yeah, that's not too bad.
Appears the bottom has gotten a little bit more sandy on top of
Let's do
the
a 310.
rocky outcrops.
sounds good.
30 meters is fine.
Copy that. As soon as they're done, I'll call it up.
Okay, I'm going to come back a little bit. We got a little bit of time. The ship had to make a heading change, Chris. They're still moving around, so we're going to wait for them before we put in another move.
It's just Jasper right now.
No, Jasper.
Chris went to lunch.
Chris went to lunch.
Oh, we got
okay.
Jasper.
Yeah, it's 11:30.
Oh, is it? Oh. You want to hop in, Levi?
Sure.
Lost track of time there.
And for those watching, wondering what's going on, we're quickly hopping seats here between pilot and co-pilot. They're doing little musical chairs. Looks like there might be something in the water column to the right. Or not.
Come in. What do we got?
No, that's not a fish.
Garbage?
Looks like potential ink from. Yeah, just unable to resolve it.
Yeah.
Translucent? Yeah, we're going to do a 300 now.
If it doesn't focus, we might as well move on.
Okay.
Pull out video, I think.
You can see a nice slope here, pretty steep. See the current ripples in the sand, so water is definitely moving along the side here of this
pretty
seamount.
steep. How steep would you guys estimate that?
Oh boy, well, about 45
Copy
degrees.
that, Bridge. When you're done with this heading change, can we put in a move at 30 meters?
You can still sort of see the little steps in there, so you
0.2.
can still see that there's a solid rock underneath. But the sediment's deep enough,
Copy
deep
that.
enough to really
Let
make
us
some
know
nice
if you need
ripples
to
and
change the heading anytime.
If you have wind.
Not see too much of
current
just
going
the
this way.
underlying.
Yeah, they're at
ground,
opposite
underlying
of each other
rock.
right now.
Turn into the wind, then you can just back your screws against it.
Well, they're the DP experts. I'm not sure. There might be other factors to contend with.
Copy.
Pilot, are we waiting for a ship move?
Yes.
Yeah. Had to change its heading a little bit. So. Not got a whole lot of tether, but.
Copy, Bridge.
You can pan left and right to look at both edges of this slope while we wait.
Yeah, I don't know whether you copied the drone, so if we're waiting, we could just look the other way and see what's over there.
And why don't you come back towards Sirius right now?
Okay. Laterally.
Okay, co-pilot, the ship is moving right now.
Okay.
Yeah, so we're seeing similar ripple marks, and if you're looking up a little bit, you can sort of see a step in the underground. So it looks like this may be some remnant of layering that you're seeing up top in the top of the screen.
Comfortable distance D2 there.
Copy.
So starfish to the lower right of HD2.
Oh, he's way below me, I think. Alright, start moving this way.
I've got a lot of light immediately in front of the vehicle, but not much off into the distance at this slope angle.
Yeah, there's not a whole lot. I mean, we can do the lights are out. I don't want to bring them too far out because we're kind of kitty cornering the slope.
A little bit more.
What about the upper?
We only have one upper, and it's the side that we don't want to deploy. So we can move up this slope.
Floating Tina 4.
There's something up in the middle almost.
Watch your tail end.
Yeah.
So this is a Tina 4. It's a type of gelatinous zooplankton. They don't have stinging cells like jellyfish. They used to be sort of placed together with the jellyfish, but they're a separate type of organism off on their own. They have rows of cilia, little tiny hair-like structures that are in bands. The bands of the cilia look like little combs, and that's what
we can come in and then we'll
comb berries.
this guy.
So they ripple.
Now let's let it go. We're moving.
And
Let's
that's
get you out
what
ahead.
creates that shimmering on the body. It's actually these little tiny hair-like structures
So let's come up and push out.
in rows of bands on the body that are all moving and reflecting the light and creating that shimmering rainbow-like effect. There are a number of species. Some of them are round like this with the teeny, the bands that move. Others are more like bands or things with wings flapping through the water. Some of them are actually predatory on other gelatinous zooplanktons, and some of them feed on the very small types of zooplankton like the copepods. So that's a Tina 4.
All right.
Yeah, let's punch it, pilot.
I was wondering about the little green lights, whether that was within the organism or whether that was just reflections off of the light we were shining. But that explains it.
Yeah.
Okay, pilot and co-pilot, we're at the end
So
of that move. I'll wait until you push out a little bit before putting another one in.
But we weren't seeing bioluminescence there, but we were actually seeing the reflection of the lights from the movement of those little bands of cilia.
Looking good.
I'm seeing something on the chat room that other people aren't hearing what I'm saying. I'm only about 3 or 4 inches away from the microphone. I don't know if this is any better. I can try to speak up, but it also may be some problem with the sound balance.
We hear you very perfectly on the ship. You seem to be balanced really well with the shipside scientists.
Yeah, I can hear you just fine. So it must be on the other side.
So I think we'll have to come a little more westerly on this next move.
more westerly from 300.
Yeah. So we just don't want to get ourselves like too much of an angle to the slope.
Okay. Do you wanna follow this contour right here?
Yeah, like 280 is looking pretty good.
Yeah.
Let's do that. 30 meters.
Copy that.
No, it wouldn't be the video going out because that's what I'm monitoring up here.
Putting in the move. 30 meters at 280. Bridge. This is nav. Let's start
Should
moving
come
at
back a
30
bit,
meters.
drop down.
I mean, yeah, 30-minute range, 280 bearing.
The tendency is to get D2 out too far ahead of Sirius, and then you start stretching the tether.
Bearing 280.
So you're drifting away, and it's getting stretched out. So come to port, come closer to Sirius.
Is that an urchin on the left edge of the screen or an enemy?
The yellow guy here, left to center?
Nope, left just came on the screen.
Oh, in the sand about halfway on the screen.
I'll center him up.
And I don't know what you guys have seen, I'll just point it out to you.
Okay.
Yeah, we might as well look around at it a little bit as we're waiting for the vehicles to catch up.
Okay, video can come in.
That looks like the same one we've seen a couple of times before, actually. I forgot the name for it, but this, I think, is the third one we've seen.
Okay, video looks like they've seen this one. We can come out.
Okay, co-pilot. The ship is moving right now.
Yep.
Can we take a quick look on the bottom right? Just went off the screen. There's a little yellow
bridge. This is
coral,
nav?
maybe?
Absolutely.
The range of that last move was 30 meters, correct? Copy.
Is it this one or the one on my left toe?
Just above your lasers.
Okay. All right, video, come in.
And then maybe after that below.
Partial.
I'll get closer. You can come in more if you want.
That's pretty small. All right, unless I see anybody in the chat room saying that they would really like to spend a lot more time here, I think we might as well move down a little. There was another little colony that looked a little bigger.
If they wanted to see, got a second. This guy.
Almost time to punch out.
Okay. This will be a quick one. Video, you can come in right now. I'll bring you up.
There we go. Not sure what's hanging off of that.
All right, why don't you move out ahead here?
Okay. Everybody can come out. We got time to move.
Oh, we are moving. Yeah, it looked like something like that. Oh. Thanks. Yeah, Roland was pitching in that might be a gastropod. We'll see if anybody else has any opinions on this.
Seems like the number of fishes have significantly reduced.
Yeah, there was a lot earlier. I do feel quite a bit of current here.
Yep.
Port starboard.
Hey Nav, can you zoom out a little bit so I can see where the second waypoint is? Or is that the thing up in the corner
You can
already?
see it right there. Yeah, that's already there.
Oh yeah, okay.
So we're, I don't know, Jasper, if you can hear me or not. We're just kind of following what's comfortable in terms of cutting laterally and upslope.
Okay.
So we may not hit those waypoints exactly.
That's fine. I was just curious whether we were heading in a completely different direction.
Yeah, we're just moving upslope and moving as laterally as we're comfortable.
Sounds perfect. Yeah, and as the folks on the chat are pointing out, Deb's pointing out that the
See a large
ripple marks
fish
are nice.
in your Sirius camera.
Nice and obvious.
Me,
There's a
yep. Take a look at this guy real quick.
somebody seeing a fish, but
Go ahead, video.
where that's not the one.
That moves done.
I think that's one of these. Is this one of these?
All right, you want
rock
to zoom out on high
patterns, it
pack
looks
nav?
like?
Let's get waypoint 3 in view and let's just figure out what we
Pull
can do
video.
here.
A little crab dollar. Okay.
Oh yeah, in the serious view there is a small fish coming up
That is
towards the bottom.
162.
Yeah.
Little star over here.
Do you want to just follow? I mean,
Fish is
we're
Yeah, I
not really
think-
going up
down.
slope.
Yeah, I think we,
The fish is following my tail.
There's a starfish on the ground there.
Oh. Video, it's coming out of the starfish.
It's another one of those cookie cutter perfect starfish.
Is that rings on the right? Is those shells?
Yeah, I think the substrate you're seeing underneath the sand is basically a limestone made out of reef debris, so that's mostly going to be
So
shells
what are you
and
thinking,
corals.
pilot? We should
I'm
Also,
thinking
they're cemented
we
together
just
in layers.
kind of cut.
like straight through. I mean,
Well, we can make
These
as
might
much
be-
of a northwesterly track as we're comfortable. But I don't think.
It's got a bite taken out of the top.
Wow.
I'll
Yeah,
talk
he
to Chris
does
when he
look
gets
like
back.
he's
But
missing a
we'll
little
go
bite
as
there.
northwesterly as we can, as we're comfortable, but still continuing upslope.
okay.
So.
Okay.
Pilot, do you think you could go
Alright, you can
backwards
zoom
and
ba-
up a
back
little
in.
bit so we can see that fish? It
Probably, yeah.
seems like it wants to be hanging out there, so maybe we can catch it.
Oh, the fish behind me?
Yeah, you can see it in the serious view. It's basically on the screen right below it.
See what I can do.
Yeah. He's sort of under us now. I think he's going to follow us.
Watch this. You might be able to do this move here.
Oh, there he is. Big red guy. That is not a little fish. You're right, Bruce.
Zoom on your command.
I'm not going to be able to zoom here.
Watch how much you go off screen on Sirius.
Yeah.
Okay, this is one of the
That's about all we can do,
living
video.
bottom fish. It's something called an
Thanks.
alfonsin or an alfonsino. It's one of the fish that I had expected to see at this depth. The family is Boricidae. The genus is Beryx. There are 3 possible species in this area. 2 of them are slender-bodied, more slender-bodied. One of them is deeper-bodied. I couldn't quite tell from that angle.
I think we do like a
which of the
270
species
here.
it was. They are caught
Chris just came into
by trawl
Oh.
fisheries at seamounts north of the Hawaiian
Chris?
Islands and also seamounts north of
Or do you want to get on comms?
the Mariana Islands in Japanese waters. So they're a popular food fish, one of the commercially important fish. They're distant relatives of the shallow water
Hey,
swamp
Chris?
fishing.
Can you hear me?
Bruce, sorry, I just came back from lunch. What were you describing there, if you don't mind repeating it just briefly?
That was a very nice barracks that swam around the ROV, swam under the ROV.
All right, why don't you push
and they turned the D2
port a little bit.
very nicely, and I do appreciate that, by the way.
These rocks while I
pilots did a
talk with
really
Chris
nice
and see
job
what he
with
wants to do
that.
here.
So it gave us a good view of a barracks, our first barracks for this site. Unfortunately, we were kind of looking down at it, so I couldn't get a good idea of what the body depth was to see if it was Decadactylus versus splendens or molars, but a barrack species.
So, Bruce, do the splendens, do they get big? I'm looking at Art's screen right now. He was doing a rewind of this so I could get a look at it. It looks a little deeper body, but it's a pretty good size. Do you know how big the splendens gets?
Video, zoom in on
Give
this
me a
fish
moment
back
to
here.
look that
It's
up.
Way back here. There's the word current.
Oh, there's a big fish way in the background there. Let's see if he'll turn.
Ooh. Yeah.
Yeah, Chris, I've got you in co-pilot here.
Yeah. Hi, Beck. Okay.
Yeah, there's two.
So, Chris,
Looks.
Barrack splendens is the largest species, getting about 50 centimeters long, about 1.5 feet, while Decadactylus and mollusks get about 30 centimeters. And now we're looking at another.
There's a whole group of them there, yeah.
And I can see this one's deeper bodied. Oh, there's several of them. Yeah. So these look deeper bodied to me. So I think these are Barrack's Decadactylus, the deep-bodied species. Wow, 1, 2, 3, 6, at least 7 of them here.
Yeah, these are.
Can I pull out pilot? Just show the schools. They're coming.
Sure, co-pilot. That's fine. Yeah, I'm looking at it right now. Sorry, I was a little distracting.
He's coming right at you.
Yeah, I think that's fine. If it's do what you need to do there,
Copy,
obviously,
Rich.
to make it work for the vehicles.
These guys are pretty interested in anything.
They're coming to see what we're doing here.
Yeah,
Yeah, they're attracted by the lights and the noise. I think that's amazing.
Oh, everybody's coming now.
That's a good spot, whoever saw those.
Bruce, sorry,
Can we set it
the
up
co-pilot, we
HD2
were
and
discussing
that's
the
for you?
track just as you were speaking there, and you were commenting on Decadactylus versus splendens. So do you have a better idea of what these things are now that you're getting some more looks at them?
Yeah, they look deeper bodied, so I think they're Barrack's Decadactylus. To really pin that down, you have to try to count the fin rays. We almost could do that from these videos. But my impression is that they're deeper bodied than splendens or mollusks. What do you think, Chris?
Yeah, I would agree. They look deeper to me as well, and pretty good sized fish too. So, some of them are. That one's a little bit not as big, the one that's in front of the lasers right now, but
They're swimming up against D2 to the camera and to everything.
Yeah, I can see it on all my cameras.
yeah, you rammed right into the camera lens,
Yeah.
didn't he?
He did.
Yeah, clearly they're terrified of the ROV.
Chris, I was looking up the maximum sizes of these. Barrack splendens is actually the largest one. It gets to 50 centimeters.
Oh.
The other two species, oh, that's a beautiful shot. Anyway, the other two species get to be 30 centimeters, so about a foot long for the Decadactylus and mollusks, and then about a foot and a half for the Barrack splendens. That's definitely Decadactylus.
Great, thank you, Bruce.
So on your third camera view, the quad changed a little to show those different fish coming in from different angles.
Copy.
There's another starfish in the sand there.
Yeah, I know.
Bridge,
a little,
this is
little
Nav.
high. I'm not sure it'd be possible to get a zoom on that, but if it was.
Yeah, we got a minute.
Let's get another move in. That's going to be 30 meters, bearing 270.
Sand's definitely a lot deeper
Speed
here.
0.2.
Look at that jagged limestone there. That's definitely been weathered out.
Yeah, copy.
So
Dev's pointing out this is pretty clearly
Copy,
reef
thank you.
debris sort of sticking out.
So did Chris Ma call in at all yet about the other sea stars we saw, or not yet?
Not while I've been here.
Okay.
David, you can come in just a little bit, not all the way yet. Yeah.
Well, this one is a little. Is this the same one that we saw earlier?
It could be, and he's full.
No, we saw one of these while you were gone.
Well,
He had a little bite missing though, so I don't see the bite missing in this one.
Partial.
Okay.
This might be a different one.
Current's blowing me a little bit here. Try to keep it in frame.
Different individual.
Come on.
Yeah, so I wish Chris was here to talk about this, but I think when they get swollen like that, that's how I would sort of describe it, is they actually have got a lot of food maybe or water that they've inflated with. So just because you see one that's kind of higher like that and one that's a little flatter, that doesn't really necessarily mean anything.
No.
Sorry. Just
look at the tracks in the sand too. You can see there definitely lots of animals moving around here.
Yeah, those could have been his tracks right there.
Maybe.
Where that little rippling.
Oh yeah.
Where the lasers are right now. Yeah, thank you, pilot. Shoot it.
All right, video can come out. Sounds like we're done here. Keep moving.
Charles is asking who's at the end of the track. I think it might not be that much to find there anymore at this point. It ended in a little rock pile.
Big anemone, I'm trying to figure out what that orange thing is. I think it's just a cluster of anemones, but if we could take just a quick peek at that orange cluster and then maybe the larger one too after that.
Absolutely. Okay, video can come in.
Okay, it is a crinoid or feather star that's on in Anelopsamia. There's a mushroom coral too there that's closed up, and it's on a fossil.
I was going to say, did you notice what this is all on top of? This is
Big combination.
a big structure that is clearly was part of a former part of the reef.
So the mushroom coral, I don't know if Tina's can see enough to, we could, after the feather star, just do a quick pan down to that red blob to the directly below the camera. Yeah, he's
the blob.
He's not in terrific shape, but a little squat lobster under the coral as well. Beautiful shot of the fossil coral.
Yeah, you can see the sort of long, stretched out tubes.
Yeah, I see that as well, a dead bamboo right at the, the remnants of what used to be a live bamboo. And you've got some very clear stoloniferans on the left-hand side next to the bamboo as well. So lots of animals in this zoom.
Come out a bit, video, and then we'll see everything in the view, see if there's anything else they want to see here.
And that anemone, I don't know what the anemone is actually. Anemone, maybe?
I'm with you.
Yeah. It seems a bit shallow for Exosalactus. That's the problem I'm having with it.
It's got the little
gastropods are right on the base of it.
It's got the little nubbies at the end of those tentacles.
Yeah. I don't know. Could be in that family at least. Exosalactidae. I'm not really sure.
Okay, video cloud a little bit.
Wait.
Watch these. Anything else here you want to see? Or?
No, thank you. You got a lot in a quick zoom.
Yeah.
That was
combo shot.
Yeah, very, very much a combo.
All right, we'll switch out pilots here real quick for you.
Yeah, so we're going to be pausing for a moment here as our front row is switching out.
So, Chris, what do you think of my fish order now for the day?
You did pretty well, Bruce. You definitely did pretty well. Of course, a nice onaga and Edelus coruscans would be a great addition as dessert, maybe. But
or if one of those sharks would just zoom through while we're actually watching, that would be nice.
Yeah, ship move just completed.
Lots of little bumps in the sand here that are not ripples. Those are all marks from someone walking through the sand here, essentially.
Yeah. Current is port to starboard. Okay, and he's
facing
it looks like
west.
it's coming south. It's coming south and going north.
It was the same yesterday afternoon, I think. Might be tidal.
Yeah. Yeah, it definitely could be. There is a fish scooting along the bottom to the left. Okay, I got my eye on him whenever Josh is just settling into the pilot's chair right now, and I'm trying to keep my eye on him. Although it's hard when he goes off screen. There he is. It's one of those tiny little stubbles that's in the upper left. And
you're going to have to tell them a little more than that,
yeah,
probably.
So keep coming up and more to the left with the lasers. I'll see as we keep coming more to the left with the lasers. He's kind of elongate. There, he wiggled. Or you're getting really close now.
There's two of them, I think.
Yeah. Yeah, there are.
Those are nice and
Partial.
reflective.
Come on in.
This is an easy one for you, Bruce. This isn't much of a challenge, but it's the first of these that we've seen.
Yeah, I don't know if we've seen these at Wake before. We haven't been shallow.
Co-pilot,
these
this
are
is
our
Nav.
old friends, the green eyes.
It's passed to me that we might not want to head directly to waypoint 3. We might want to head more of a direction of 330. Kind of go up slope a little bit.
This genus just means green eye. They're interesting fish because, like the deep water fish that we see, they're simultaneous.
Did you guys just change lights?
They're both male and female at the same time, which is not true for
I
fish,
think
but it is for a few. So they're an ambush predator down here on small crustaceans, small things as well. We've seen them on both soft sediment habitats and on rock. I don't think we can know what species this is because the family still needs some taxonomic work.
Well, if we want to start heading towards waypoint 3,
there's another one sitting right there.
Yeah, usually when you see one, you'll see a whole bunch of them. They're sediment dwellers. In fact, if you look, you can see at least 3 or 4 of them. They're all wiggling
see how
around.
firm of a target that is.
It's a pretty common inhabitant of these depths.
Dan was telling me they want to go up like this.
So it's just good to sort of document their existence here.
Kind of past. I thought that was more for safety, traversing up the slope rather than going along.
I mean, this isn't a very... It's not a lot of... I mean, have you guys been seeing a lot of rocks? I haven't been seeing a lot of rocks.
Yeah. Let me touch base with Watch Lead.
It's taken off quite a ways.
Not like the sun.
Watch Lead, this is Nav.
Roger, Nav, go ahead.
Just it was passed to me on the watch before that we might want to head more west from the waypoint 3 instead of heading directly towards waypoint 3. What would you like to do, Watch Lead?
Well, yeah, so previous navigator, and this is Nick, right?
This is Nick,
yeah.
yep.
Hi, Nick.
Hey.
Previous navigator was making a recommendation based on the vehicles and having them in a efficient location. From my point of view, the closest we can hug the drop-off, the better, because I think we're going to see a lot of sediment if we get too far away from the drop-off, but it's a matter of what you need to do.
So the drop-off over here.
Yeah, to the east side, and that looks like a piece of ordnance there, shell casing.
Yep.
Okay, let me talk to the pilots, and we'll come up with a route.
Yeah, thank you. Appreciate it, Nick.
You turn the lasers
no problem,
on?
Watch lead.
They're on. We just have to zoom out to see them.
Oh, okay. Never mind.
They're still on.
Okay, pilots.
Just curious how big this was, but
it's pretty small caliber. I'm pretty sure. Could be. It may even be small. It may be 50 caliber. 50 cal. And I'm not really certain.
There's the lasers. Yeah. So that's maybe 5-6 centimeters. About this big.
Yeah, could be. Not. It's the casing for a projectile, sort of a medium to small size projectile.
This morning, while we were on the bridge, somebody was pointing out that there's still leftover machine guns and such on the beach on the island right above us. So it's not entirely unexpected to find some traces of this out here.
Spoke with Watch Lead,
this could even
and
be
they would like to
an
hug
airplane. I'm not
drop
sure
off
the
as best they
calibers,
can.
but basically what you're looking at is the remnants of a
once
casing.
you're done viewing
and
this,
the
if you want to
casing,
come up and give us a sonar view looking north.
Fast, so if you were to actually try to touch that, it would just crumble like paper. Just you would be unable to pick it up.
West stop. Let me turn.
They're
turn more northwest.
I don't know whether it's just the bronze or copper or whatever the metal they make the.
Yeah, we can start heading that way and
not
see.
Start heading.
But
yes,
north. It's got a lot of copper in it, I believe, that just erodes very quickly in seawater.
Let me put in a
That
40
would
meter
look like
move
that color,
due north and see what we see.
Brass,
That sounds good.
Good copy.
It's a little weird to seeing a single casing.
You guys ready for the beat now?
Out here,
Yes, I'm
because
ready.
what does that
Copy
mean?
that. Bridge, this is Nav.
Means there's probably more, but in this sand shifts,
put in
I'm
a
sure.
40 meter
so there
move.
may be a whole bunch of them buried too.
Due north
Another sea
at
star right there.
0.2 knots.
Yeah, these sort of puffy sea stars and another Clarpthalmus, sea pens, these rock
Or you
pens.
can turn your heading, but we'll have to keep a very close eye on that sonar.
Good copy. Thank you, Bridge.
Because if you're not looking at it, it becomes easy for you to end up like this.
Right.
Right up against it.
Josh, can I get you and Dave to move your mics a bit closer to your mouths?
Oh,
Video, is that better?
This is a different rock.
It's a little
You
bit.
sure?
I'll have to monitor it for a while.
It's still the similar
No,
type of coral.
Yeah, it's a huge fossil head of coral.
You want to take that one home as our rock sample?
Sure.
Okay, Josh.
It's only about like a meter across.
Good copy,
roughly
Bridge.
west. Monitor you in the Titan.
That was
and
a
watch the
pretty
sonar
spectacular
carefully.
head of coral when it was alive. I think it's a single head,
Move
and probably
initiated.
attached on the right.
On the right, yep. And at least one enemy made its home on it.
So we may
Oh, and there's a little thing on there too.
Yeah. Pilot, I'm going to take a guess here, but I think we have a different type of fish to the right of the fossil rock. It's on the bottom, not the one that just came out, but the one that's next to it, a little further right, directly right of the lasers now.
Very well camouflaged. Come on in, Roland.
I could be wrong, but
Looks the same.
no, it's not.
Oh.
It's different.
That one has a flat snout.
Looks different to me.
Yep.
Looks like a gecko.
Excellent.
Nice shell
So
fragments.
this is a different kind of fish. The green eyes are soft rayed fish. This is a spiny rayed fish. It's in the duckbill family. Looks a little different than the ones we see in Hawaii, but there's something called a green spotted or a yellow spotted duckbill fish. It's in the genus Cryonema. may just be individual variations, so I'm guessing that it's Cryonema chryseries.
Right, come on in. Yep, that's
very similar in habits to the green eye.
Excellent.
Yeah, and Bruce, I think you saw some of these in the Line Islands on your submersible dives, and it also looked a little bit different than the Hawaiian version of the species. I don't really remember exactly. So why do you think they call it green eyes?
Yeah.
That's just a gorgeous shot of the eyes right there. Look at that. They're
emerald green. Yeah, it's very nice.
Yeah. Oh, yeah, you can.
I think that was over the private chat, private channel, so that Roland's noticing that looks like the lens on the left eye is actually in view. Stick it out and see it.
Tons of shell debris here all over the bottom. Urchin spines, I don't know if these are pteropod shells, I'm not really sure. Broken coral debris.
So knock the coral just above the fish.
Urchin plates in here as well. Yeah, and then you've got a few really tiny sea pens kind of just sticking up out of all of this mess.
This is what happens to a reef after the stuff dies and starts getting knocked around. Here's a pin from one of these urchins.
Yeah. So thank you, pilot. Whenever video's finished, we're good.
When you're good, Roland. I'm going to back up a little bit and then start moving north.
Another starfish.
So we're lateral.
You want to see the sea star?
Toward the tether right now, but it's
We
still out ahead of us.
Can probably skip the sea star. We got a really good image of it earlier on. This thing is Cookie Star.
What was that, Dave?
And
we're
this one too,
lateraling
that's a little
toward
fuller.
the tether, but the current is keeping it out ahead of us. I'm watching it.
Let's see if we can find some more corals and fishes as we're pretty close to this ledge. It's a little bit of a little ingress right here that we're on at the moment.
Oh wow!
He's spinning to the ledge right now.
You can sort of see the steep cliff that's sort of just below us. Yeah. Large limestone structure that's totally rounded out, clearly weathered out. There's a fish on the top of it, almost in the middle of the screen.
Yeah, I'm looking at that. Could we take a look at this? Yes. Oh, good one. Okay, it's the first one of these that we've documented today. And the coral behind it is really good too, the sea pen, but let's stick with the fish.
Partial.
Yes, this is another thing that we'd expect to be very abundant in the right sort of habitat at this depth. This is a deep-water cardinal fish in the genus Epigonus, and there are a number of species of these, too, difficult to identify to species. They're often around slightly more vertical relief with undercut areas, little crevices, caverns for shelter, and they're planktivorous, hanging out in the water column. They're called cardinal fish. They used to be classified in the same family as the shallow water cardinal fish, but now they're considered unrelated and in their own family, the Epigonidae.
Yeah, thank you, Bruce. You're just cruising over one of these Iguchi somnia corals, or at least what I think is, that we imaged earlier.
Yeah, just let him go. Come on out, Roland.
We have a sonar target coming up northwest of us.
And I'll also just mention that new species in this genus Epigonus seem like are being found all the time, and every couple of years another new one is described from
Are you
someplace.
seeing that in Sirius?
Sonar, only in sonar.
Are you seeing it in Sirius or in
No,
DT?
not yet. Oh,
I'm
sorry.
trying to think
Sirius
of how many
sonar.
we've got in Hawaii. I think we must have about at least 4 or 5, maybe even 6 species of them.
You can turn to that.
If I'm not
to the
mistaken,
north.
there's
Turn
Athranoides,
northwest.
Devanii, I can't even remember. Glossodontus is another one.
Yeah, I have to start looking that up, but you're right. There are 4 or 5 species in Hawaii, and there are more in the western Pacific.
So we're going to stay on the top of this ridge. We're not going to go down at all.
Copy.
That way if something happens, where we can pull up above it pretty quick, or we'll already be above it.
Sure.
Copy that, watch lead. Is that better, watch lead? The ship in the upper left-hand corner?
Sort of heading along this ridge. Again, we're on a little bit of a flats area, and then it's going to just drop straight off in just a little while. They're very close to the drop-off right now, but this track will probably give a view so we can peek over the edge, see what's hanging on right at the edge. What is that? Is it? That's just a rock, right?
It's a rock and a white line from something else further away.
Oh, okay.
Copy now.
Let's do a quick zoom roll.
It's probably another piece of coral debris.
Yeah, funny looking rock.
Yeah, that's another. You can see this structure. This is another coral head.
Yeah, a little rock pen on top with a little immaculate crinoid.
Co-pilot, how do you feel about a bearing of 330?
Where's that? Go from Sirios.
That's.
I know, but when you click and drag,
right.
Go from Sirios.
Right.
Yeah, that's fine.
Yeah, I think we
almost looks like there's some manganese crusting there.
On the left, maybe.
Prefer to go due north.
Pretty shallow for manganese crusting, but
yeah.
I'm not sure how.
Well, what's also surprising is that we haven't seen it on anything else.
Seeing a little bit on your sonar. Almost a little bit left of you, almost dead ahead. That's the only reason I said 330, but we could, if pilots want to go due north, we could do that too.
Let's go north.
Yeah, the drop off, I'll bet.
Due north, copy that.
How big of a move do you want to do?
30 meters.
Yeah, that's fine.
Bridges, Nav. I'd like to put in another ship move, 30 meters, bearing 000 at 0.2 knots.
That's Grammacolepus again. We got this is the Grammacolepus brachiaculus, the tinsel fish.
Ah, yes.
Yeah, we got a couple shots of that already. Is that another one in the background, or is it something different? Sort of over to the
about
right.
a third of the top on the right. Yeah, he kind of disappeared now.
Could be the same fish. No, it's not. Maybe it's a barracks or something. Might be worth taking a zoom at that.
You have him in the middle up.
And yeah, it's another one of the barracks, the Alfonsinos. I think Bruce mentioned this when you first came across them, and I was down at lunch, but these are a commercially valuable fish species, so it's really great to image their presence here. They're harvested in other places of the world.
Come on up.
Tino 4?
Yep, come on in.
Something rather, pretty jellyfish of some kind. Tina 4. Yeah, and again, you can see those iridescent rippling rows of the combs of cilia, just like
it's really out there.
They can be very pretty.
Yeah, we're pretty stretched out. Now, what speed are we moving?
Thank you, Bruce.
It's 0.2 knots.
Okay.
You are listening and looking at a live broadcast from the NOAA ship Okeanos Explorer. We are on the east side of Wake Island. We are currently at a depth of 505 meters below the surface. We're inside the Wake unit of the Pacific Remote Islands Marine National Monument.
Where's that move going to put us?
We've got a bunch of people on chat right now, including Diva Amon, who's at UH and is now down in Trinidad. Abby Lapointe, who's at UH.
I care about Sirius. I don't care about
coral
the ship.
expert. Sako Matsumoto, another coral expert from Chiba Institute in Japan. Bruce Mundy, that's his voice you've heard a couple times on the fish. He's in the NOAA Fisheries Science Center on Ford Island, on the island of Oahu. Charles Wall with the NOAA Protected Marine Protected Areas Division. Deb Glickson from Harbor Branch down in Florida. We also have Ken Sulak, another one of the premier fish experts, and Michael Vecchione from the Smithsonian, Nicole Morgan, and Tina Molitsova from the Shirshov Institute in Moscow. And my name's Chris Kelly. I'm with the University of Hawaii, and I'm serving as a biology lead. And next to me is
Jasper Conter, and I am with the University of Hawaii, and I am the geology lead for this trip.
And if you're watching this broadcast from one of the aquariums that are doing the live feed, such as the Maui Ocean Center on the island of Maui in Hawaii, or the Waikiki Aquarium, welcome.
Okay.
Hope you're enjoying your visit to these two beautiful aquariums.
So on the bottom corner, you can sort of see how you have these yellow blocks sitting on top of these black ones. It looks like some of the more recent reef debris has been falling down into this field of these black-coated rocks. We're still a little puzzled why they would have manganese coating on them, but that's what it looks like they do.
Small blackfish left of lasers.
But Deb was wondering about the upper limit for manganese coating.
I don't know.
I don't know either.
Yeah, I'm not seeing them, Merlin.
Supposedly, the optimal depth range for manganese crusting is typically about 800 to about 2,000, 2,500, and we're at 500.
Yeah.
So we're
He's
several
top
hundred meters above
top left
the
third.
lower limit of that range, but that doesn't mean it can't occur.
If you look at the bottom of these rocks, right where they border on the sand, you can see the yellow carbonate color shining through. And then you look a little bit further up, and it turns completely black. So I think it is a
to the right.
It is a coating
right next to the edge of the screen.
That little guy?
Yeah.
Oh, yeah, we've seen these.
Okay.
Come on in.
From that distance,
I
couldn't tell what
spotted
he
another
was.
little fish. Aha!
He's way out there now.
Yeah, I mean, I can't get to him.
I think Roland actually saw that one.
Okay. I think this is a different variety, Bruce, but I'll let you weigh in on what you think this is.
I'm not getting a clear look at this yet. It's so indistinct against the dark background. I can't tell if it's another epigonus or if it's something else. If it is an epigonus, it's more slender than the previous one, more like the epigonus atherinoides.
Yeah, come on out.
Atherinoides means like a silverside. There are a group of shallow water fish called silversides that are unrelated that are slender. So, Chris, you could probably see this one. Do you think it was an epigonus?
I was actually, it looked to me like it had adipose tissue on its nose, and I was wondering whether it could be Glossanodon or something like that.
What's that?
Ship move just completed. You want me to keep going?
Yeah, that's the other possibility.
Go more to the
I
west.
couldn't tell.
Let's go
The fish
uphill a
that
little
Chris
bit.
just mentioned, Glossanodon, is, I don't know, if I describe it as kind of a deep sea smelt, I guess, or it's a soft rayed fish that has a single dorsal fin. The epigonus have two dorsal fins. But I couldn't really see the fins against the dark background and at the distance. So we'll have to leave this one as unidentified.
Excuse me just for 1 second. This is a really important cluster of corals here, as are the ones on the ridge. And first of all, we can look at the sea star on the coral, but we absolutely need to zoom in on that gold-colored coral to the left. This, I think, is what we are really looking for today.
Yeah, standing off on the move.
Yeah.
Is that a tinselfish?
Yes, it is.
All right, go ahead.
And I'm pretty sure I'm right about this because I think I'm seeing the early phases of the overgrowth of this particular parazoanthid on one of the bamboo corals to the right. So this has overgrown a bamboo coral, and it's hard to say for sure, but my best guess is this is a gold coral, and it is one of the precious corals that we were hoping to see today. This is in the genus Kulamanamana, and the actual species in Hawaii is Kulamanamana haumei, which is a mouthful, but we can have some people who really know these corals take a look at these images, but quite frankly, I'm absolutely delighted to see this.
Fighting a bit of current here, Chris. Sorry for the shaky picture.
Let's go for a full of the two.
And then when we're finished with this colony, if we could look at the colony that's in a partial state of being overgrown. This is as soon as we finish the zooms, I'll explain to you what I'm talking about. Those of you who are not familiar with how this all works. And the bamboo coral actually is different. It's a different species than what we've seen today as well. So you can see the yellow Kulamanamana is overgrowing. It's actually attacking and killing this bamboo coral.
Come on out, Roland.
I think I'd be in a better position if I were maybe 5 meters to the west.
I think he's going to swing around and look at it from the side.
Yeah, and there's even some more up on that other...
You want to be 5 meters?
Or maybe 10 meters to the west, more behind you.
I'm coming around.
Yeah, see there's another gold up here on the very top of the ledge as well. So this is terrific.
And then we should come up this.
Yes, I'm coming out already.
So Dr. Frank Parrish at the NOAA Fisheries Science Center, along with Amy Bacco-Taylor and a bunch of other people are studying this coral. Frank in particular is really interested in this group and understanding their growth rates and the environments that they need to live in.
To me also.
And this is a species that can live for thousands of years as well. He's described it as being in 2 phases. He calls the phase where this parazoanthid is overgrowing the bamboo coral, but it hasn't started secreting its own matrix yet. It's just overgrowing and killing the live coral. He calls that as the Midas phase because he's turning one coral into gold, and that's why he called it that. And then as soon as the colony is fairly subsumed, then this parazoanthid is one of the unique parazoanthids that actually secretes its own matrix, and it's a proteinaceous skeleton that's quite valuable in the jewelry trade. I'm certainly not advocating the deep sea coral jewelry trade. I'm just explaining to you what the process is. And you've got a very unusual sea star there, but this is what would be called the Midas phase of the formation of a gold coral colony, and you can clearly see that the parazoanthid has overgrown the skeleton of the bamboo.
Copy.
This bamboo is branching from the nodes, so I think it's a different type of bamboo than we saw earlier.
Yeah, that's better, thank you.
And that's typical of Kulamanamana as well. It seems to attack nodal branching bamboo corals primarily, such as Acanella, and this could be a species of Acanella, I'm not sure. That sea star may be something like Hipposteria too, and this poor coral is being attacked by a couple different things. Not only this other cnidarian that's overgrowing it, but the sea star is eating it. So it's struggling here.
Gonna pull out partial.
Oh, that's beautiful. Thank you very much.
Full specimen.
There's a Bathopathes. I think it's the first Bathopathes that we've seen, and some Plexaurids next to that.
Anything else here, Watchley?
No, thank you. And then there was some right up on that ledge. You could take a peek at those too, perhaps. There's another barracks. Yeah, that's the barracks, and you can see these things from a ways off because they swim with their pectoral fins.
Yeah, he's got big fins.
Yeah. This is a colony that's in a further state of being overgrown. So you've got a gold that used to be a bamboo like the other ones on either side of it, but now it's completely overgrown by the gold coral.
Is that a fish in the top one?
Yeah, another barracks as well. A lot of these are around. Oh, this is fantastic.
Yeah, come on in, Roland.
Put a little squat lobster on it. So this colony is, I think it is completely overgrown, and it's this particular gold coral is probably entered into the phase where it's producing its own matrix. Some of these thin branches that you see on the right here, it probably created that skeleton itself, this parazoanthid. Wow, this is terrific. Yeah, thank you. So beautiful shot of the polyps.
So this thing overgrows first and then starts producing more branches itself?
Yeah, well, it does do that, and it starts laying down a matrix, a skeleton itself, on top of the bamboo.
Oh, okay.
So if you were to cut the main stem of this coral after it's matured and looked at it, you'd see all of this black type of matrix that, there's a little scorpion fish there on the left. You would see this black matrix that they use to make the jewelry, and in the very center you would see the remnants of the bamboo coral that it overgrew.
A little down, Josh.
You want to see the scorpion fish?
Sure. Yeah, let's take a look at them because we have a better view of them here.
Okay, come on in, Roland. I'm going to come down a little bit.
I don't know, Bruce sure looks like what we were talking about earlier.
Yeah, it looks like the Satarchy species again. I don't know if we could zoom in on the head. There are spines on the front part of the gill cover, and the nature of those spines can distinguish the two species, Longimanus and Guntherite.
Tight.
So, again, I don't know if the distance allows that, but getting a really tight zoom on the head.
They're doing that now, Bruce. You'll see it in just a couple seconds.
While we're waiting for him, you can see very nicely how the coating of the black stuff on the rocks starts a little bit up from the actual sand. So the actual rocks are these same carbonates still.
Definitely looks like crust, doesn't it?
That looks like manganese, but very thin. We saw this before on the other side of Wake Island, too.
Back.
It's probably about as good as we can do.
Yeah, it's a pretty good shot. Thank you, pilot. Little squat lobster above him.
And if you're good, Roland.
Yes.
Frame them.
One thing about these Satarchies is they tend to have more of a convex head shape, and quite a few of the scorpion fish have more of a
Pilot,
concave
are you ready for a ship move?
No,
head shape.
Copy.
Is there anything else you wanted to see here?
No, I think we're pretty good for this spot. Just,
Give us
come up a little bit.
Can you give us a sort of a nice rotate left and right? There's a
quite
crab.
a sheer
I don't know if
drop
they've
off
seen the
wall
crab.
here.
It's the crab, bro.
Lower left.
You want to see this crab over here, Chris?
I don't see him yet. Where is he?
Centered up on the lasers now.
Yeah, I got a pretty good view of him. Sure, let's take a
come
look.
on in.
Alright, so my best guess is it's could be something like the Progerion moose that we identified earlier, but that's a much better look at him. Could be wrong, but it's a
come on in.
what do they call that group of
This
crabs?
guy has both his pincher still.
Yeah.
And still no antlers.
Maxim.
These are
It's about the best I can do.
no, it's not that family. Let's see
come on out.
here.
Yeah. All right, let's move like 20 meters or 15, 20 meters in this at this heading.
Copy that, pilot. 20 meters, bearing 320.
Yeah.
Copy.
That'd be fine. I will.
Bridge, this is
There's
now.
some more gold colonies overgrowing bamboos. This is really pretty, right on the edge.
I'd like to put in a ship move 20 meters, bearing 320 at 0.2
looks like a
knots.
barracks again, hiding amongst the corals.
So I put this in.
Good copy.
Go ahead, video.
Can you come up a little bit and see what's out ahead of us?
Thank you.
All right, so I got 20 meters from where I am, so
I
you'll
just
be
put
good.
this in the chat too. I think that we're looking at is this is much steeper, much more sticking out than what we've been looking at before, and so maybe the surface here has just been exposed much longer than some of the other carbonates we've been looking at that were much closer to all the sediment, all the sand coming down.
Beautiful
Nav
scene.
copies.
The rocks are really
Yeah.
attractive with the corals. They're really well, they are. I like sort of the whole composition here is really, really amazing.
Nice little snapshot for your computer background.
Yeah. Gonna need to figure out what type of bamboo coral that is, and some point it's got a lot of nodes like that tall one sticking up. It sure has nodes at very high frequency along the stem.
And this is the gold coral here?
Yeah, that's a gold coral. Used to be one of these.
Yeah. Well, that's why I was asking.
Yeah.
It kind of looks like it has a similar pattern, just
Yeah.
fatter.
Yeah, that's it.
Come on in, Roland.
There it is, overgrowing, and then
Yeah, sitting right on a stalk.
Maxim.
Looks like that one's actually started to secrete maybe a little bit of its own matrix, giving it a little bit of a darker tone underneath the tissue. The gold coral will actually have a very dark matrix that
If you
it
go back
produces.
down that stalk, I saw
There's
a
a
cephalopod
paper for those who are
eye.
interested by a guy named
I can
Sininger.
barely just
and
hold this position, Roland.
Okay.
He described the species, revised the names. Used to go by
That's
a
a
couple different
shrimp.
names. This used to be Gerardia, and then it went to Savalia, and then
Yeah, there's
the
something
final
on that coral right there. in the center.
Final decision was to put it in its own genus, Culomonomana, because it's so unique among the parazoanthids.
Getting pushed away.
All right, Abby is weighing in on the bamboo, and she thinks it could be the same clade as Jasonisis. Clearly see that there are these small branches coming out of the nodes along there, quite a lot of them actually. Yeah, outstanding. Did somebody just join the chat? Not yet, maybe.
Ready to pull out for partial?
Quite a lot of current there, it looks like, Pilot.
Yeah. It's taking a bit of concentration to hold this.
Yeah.
It's a nice view there. That's partial.
Kill the lasers.
Something's hiding right there. I think that might be another scorpion fish, maybe.
Yeah, it could be.
You want to look there?
Hello? Who we got?
Hi, Chris, it's Abby.
Hey, Abby, you've come to the rescue. So, what do you think? What are we looking at?
Hey! So, that bamboo, it looks like in the same, it's probably not a Jasonisis, but in that same clade, those all branch at the node, and they look pretty similar. So I can't really, I'm not as familiar with those, and I can't really tell, but I'm thinking it's probably somewhere close to that, to Jasonisis.
Good, Bridge.
Uh-huh. So have you seen this before, Abby? Are you familiar with what this could be or not? It's, you know, it's very planar in general, although not perfectly planar, but it's got, you know, very short internodes, I guess is the way to put it. Do you have any idea?
Right, well,
Go ahead.
One of the, like, it doesn't look like a Jasonisis necessarily, but one of the things about Jasonisis, some of the main characters are that it has, you know, very short internodes, and it branches up the nodes. I mean, that's about,
Bridge needs
that's
to make
the
a
closest
45-degree
that I
heading
can think of what it
adjustment,
is,
so
close to at least.
we'll be standing by in this location until they're done.
So what would be the difference between Jasonisis and, say, Acanella? How would you differentiate those two, given that both of those genera branch from the nodes? Is it that internodal distance or some other thing you're looking at?
Another thing about Jasonisis, and I couldn't really tell from the photos, but the fact that this zoanthid is, you know, overgrowing it like crazy. Jasonisis is known by this really thick, like, seeding time. This really thick tegument all around it. So that's one, you know, unique character. And then also you can't, one really defining thing about Jasonisis that we can't see from these photos is that it has a lot of scales for the sclerae. So
I see. Well, I'll tell you what.
Yeah, unfortunately, from these images, I can't
Tilt down to
tell
a
which
pirouette
one,
would be
but
nice.
it's up. It's one of those Acanella or Jasonisis area. We can probably rule it out to those two. Probably not. I mean, maybe not the same genus as Jasonisis, but something close to it.
Yeah, thanks, Abby. Well, when they're in a good position, maybe I'll ask them to do another close-up. Of this particular coral. That might be helpful. We can certainly do one more because it's obviously a very important species at this location. They're all over the place, and you know.
Yeah, and that's amazing. The zoanthid just completely overgrowing it.
Yeah. So this is actually very in the process of being converted to a gold coral bed from a bamboo bed.
Right.
Watch lead, Nav.
Roger.
Well,
yeah, thank you, Abby. Thank you so much for calling in. I appreciate it.
Thanks.
Chris Asako was asking if it's worthwhile collecting a branch that we get maybe the bamboo coral and a little bit of the gold coral zoanthids together to get some verification of these identifications. He's saying it's a dominant organism and it's something you're on the lookout for. So maybe a collection's worthwhile if you can get a 2-for-1 thing.
here.
Yeah, absolutely, Bruce. I was thinking the same thing myself. And just as a fish that you will definitely be interested in just swam off the screen.
Watch lead, Nav.
Roger, Nav, go ahead.
Ship has to make a heading adjustment of about 45 degrees, so we'll be standing by this location for a few minutes.
Well,
you can live with that.
well, I guess, I don't know, pilot, if you ever heard the conversation about the possibility of collecting a combo sample that had some of the gold and some of the bamboo that is a branch in the process of being overgrown.
That might be tough.
Yeah.
Just a sample in general at the moment.
Right.
I'm using almost 100% thrust if I want a lateral. So the odds of me being able to hold a position long enough are pretty low. Let me take a look around and see if I see something I can perch on, but
yeah,
I don't think I can do it at this point.
That's fine, Josh, and yeah, just something to keep in mind because several people had mentioned this on, and Bruce just mentioned it, passing the message from Osako, and I also was thinking the exact same thing. But yeah, if there's an opportunity that you think is where it's possible to do that, maybe that could be our first biologic. So, Bruce, did you see that fish?
Yeah.
Yeah, if we have time, I have a couple of comments about fish. First, I want to mention that while we were talking about the corals and all, a deep-water snapper swam past. I didn't get a good look at it, but it was in the subfamily Attillinae. So it was one of the commercially important species that you're on the lookout for. It might have been a Randall snapper, Randallicles, but I really couldn't see it because it just zipped past in the background. Second, the roundish fish with the huge pelvic fins, the fins on the bottom that we saw, I think is in the genus Stephopristis. There's a dory-related fish. It's related to the tinselfish we've been seeing. The barred, kind of oblique bars on the body seem to be a characteristic color pattern for Stephopristis. There is another genus that's closely related called Cytopsis that also has those big pelvic fins. When we saw a fish like that in the Marianas, I said, well, I think it's Cytopsis, but then I did some checking, and it looks like the Stephopristis. And if that's the case, that's very interesting because Stephopristis is only known from the Hawaiian Islands and some seamounts to the southeast, way past French Polynesia, called the Nazca Sala y Gomez Ridge Seamount. So if we are actually seeing Stephopristis here at Wake and over in the Marianas, that's a nice range extension for that genus. So keep your eye out for deepwater snappers. And we just saw another fish that's a new record for Wake Atoll. It's called the spike fish. It looked like it was in the genus Holardia, but had a little different color pattern. So we're seeing lots of interesting fish right here at this single site.
Yeah, Bruce, and I concur with both of your IDs there. I am in agreement that was a Randallicles that swam through. I did get a pretty good look at him, and we obviously can't tell until we freeze that image, but I definitely think that you're right on that one.
Right. We need to
and also I agree with the Stephopristis too. That's what I thought it was when I first saw it, but again, the still grab later on will confirm that.
And it's shifted on.
And the Holardia is a little different than the one we have in Hawaii, but that was certainly a Holardia. And the interesting thing about Holardia is that we see them quite often in Hawaii. Guess what? Associated with gold coral. So no surprise there that we saw one.
So how many degrees per minute did you say? I'm sorry.
Okay.
Copy that, Bridge.
Looks like there's a
pilot's. Bridge will be able to get.
I always get mixed up. I think it's a cell.
2 minutes.
Not a siphonophore, but I get a little mixed up on
bearing
those.
of 330, 20 meters.
Just swam by in the current?
At least. Yeah.
Could do 30.
Yes, that's up. Well, it's probably up and over. Yeah, that would be fine.
Okay, I'll put that in. We'll bump it up to 30 meters and.
So the Randallicles that Bruce was pointing out, and he kind of alluded to it, is this was a very important observation because these are commercially valuable bottom fish, and I just wasn't thinking we were going to see any on this particular dive. And
Yeah,
sure
I'm
enough,
not sure that we're
we
clear
got
for 30.
one. So we know that that's
20
one
is better,
species
yes.
they have here at Wake.
Copy that,
We've
co-pilot.
got a different type of bamboo coral down here that's got orange-colored branches, and I don't know.
Am I okay to come down here?
I don't know if it'd be possible to take
Yes.
a peek at
Yeah,
that
that's
one.
fine. I'll follow you down.
This looks more like the something we call Acanella. The genus
Here, come on in,
Acanella,
Roland.
just from the color. Maybe it's the same thing. I don't know.
Cambridge,
Abby
like
is
to do 20
paying
meters,
attention
bearing
right now,
330
and maybe she
degrees
can
and
evaluate whether
0.2 knots.
she thinks the lighter colored one and the oranger colored one are different, or whether they're just different color morphs of the same
Good copy.
species.
Copy that. Pilot's move initiated.
All right.
And my tether's being blown off to my starboard, right?
Behind you to starboard, yes.
Yeah, okay.
And turn to starboard myself. Actually, it looks like it's mostly going straight up from you right now.
That's partial, pilot.
Yeah, copy.
I'll notice that the colonies are growing sideways to the slope here, so the current's presumably running along this face, which I guess is not a surprise. And their corals are lining up to try to optimally filter the water. There are also some little plexaurids that are mixed in, and we've been kind of ignoring. Hopefully they're not mad at us, but we've been so enamored with these bamboos and the golds and fish and things, ignored some of the other inhabitants of this community.
Would you like for me to pull out wide?
So Asako's pitching in that she thinks she saw different colored branches yesterday too.
Okay. Very weird crinoids on that plexaurid. Look at all the tangle of arms they've got.
Top of the screen, yeah.
Top of the screen.
So this move takes me right up and over what you're looking at now.
Those are
Pull
right
up a little
there.
bit.
Yeah, come on in, roll.
They've got them all kind of curled in a ball. Wow, that's a lot of arms.
Group hug.
What on earth is that? I'm not sure we've ever seen a crinoid quite like that before.
So is that the plexaurid that it's on?
Yeah, I think so. Yeah, it looks like it's a plexaurid.
Can't even see the disc.
Yeah, they're battling this current, which is raging here. That's why the image is moving around. You can just see the way the corals are moving in this current. How do you even count how many arms that is? That's got to have 15, 20 arms in there, I think. Very cool. Thank you very much, pilot and video.
There's a couple more right here.
There's a big mushroom coral. Yeah, that red one.
Let me get my toe on.
Oh yeah, there's a couple of the crinoids just sitting on this dead.
You want to raise my port swing arm, please?
Yes.
Dead branch.
Hey, come on in, Roland.
Stowing port lower swing arm. Full stow.
And take my camera.
Excellent.
I have your camera.
This will be a real
what you'd like me to do.
interesting one to send to Charles Messing.
Lost my toe.
He's a deep water crinoid, shallow water crinoid expert
Try
as
that
well,
again.
and there's
So
some other
don't
folks
move my camera without
too
telling
that
me.
are very
Yes.
familiar with this group of echinoderms.
It's just, I think you already know that, but
Can share these images with to try to get an idea of what this could be.
all right, come on in. and you can tilt.
Up or down? What are we looking
There's
for?
another type of zoantha overgrowing
tilt as you need.
that particular bamboo branch.
Maxim.
Holding.
Wow, that's a great shot. Little crab there, right between the two crinoids.
All right, come on out.
Beautiful, thank you.
All right, you can take my camera back.
You have it back?
All right, pushing out ahead.
And I'm
Ship
coming
move
over
completed.
this.
Hey, can we brighten this up, Roland?
As you rotate, it's going to light it up nicely.
So we're on a little bit of a promontory there, and we're kind of looking back at the main slope a bit. Oh, look at all the barracks that are hiding down in the little cave there to the right.
Pretty much in the middle there, yeah.
That's pretty cool. I wonder if we could get a quick snap zoom of that. That would be...
Can you keep me in view?
Yes. Turning to follow
Hey, come on in, Roland. Let's get lasers.
Yeah, the laser would be great to get a... There they are. There's pretty big fish.
Do you want that swinger
You
back
can barely
out
see
or
the
not?
lasers.
Yeah, you can put it back up.
They're like 40 centimeters.
Yeah, I think so. Maybe some bigger than
Maybe
that
a
even.
little bit bigger, yeah. That's a beefy fish.
Come on out, Roland.
Got some a little debris slide here or something, some boulders and other things sticking out.
Pilot set moves completed. Do you want to keep moving in that same direction, 330?
Yeah, I think it'd be nice to, once we zoom in again, look at that contact between the rock and the sand again to see if we can still see that transition from light yellowish to black, to confirm that we're still looking at coated carbonate.
I'm still up pretty high because I just passed
That
over
last
it.
one was
20 meter move,
That's fine.
bearing 330
I'm going to follow you back to
degrees at 0.2 knots.
So, Chris, I just checked some more on the maximum size for Barrex Dactylus. I said 30
Good
centimeters before from one reference that I had. I see the Jack Randall was 60 centimeters over 24 inches, over
That was a
2
cool
feet
feature.
for Barrex Dactylus maximum
back
size
roughly
in his
in our
book on
direction.
Hawaiian fishes. So they're going to be pretty good sized fish.
A little less current back here, closer to the
Yeah,
wall.
thank you, Bruce. And those of you who
looks like you might have one of those
received
gold
the
corals
dive
right
plan
ahead.
last night, we just passed waypoint 2. We're a little bit beyond it right now. I'll give you a little bit of
Fish, bottom of the screen.
your bearings on
Two of them.
on the dive track and where we are in this
Snap. Good
feature.
copy.
See
So
snap.
actually, the rest of this dive is going to be pretty much
Ship
like
move
this.
underway.
We're going to be on quite steep terrain, hopefully seeing a lot more corals and more really cool fish. And
A lot of sea pins.
There's another fish on the right.
I'm going to start coming down again.
Oh,
a
and one
little
on the left too.
tinsel fish.
On the right was something else.
That was a Barracks,
No, it
another
was
one of the
another one of those.
Alfonsinos, little sea star.
Yeah, come on out.
A couple small plexaurid corals in there. And there's a gold tree up there. It's,
Is there gold in them there hills?
there's gold there. Years ago, when harvesting was still occurring in Hawaii, these trees were quite valuable, and that's sort of the problem. They're quite valuable also, but they're extremely old animals, and there's certainly a big question as to whether or not something like this should be harvested at all when you have these animals that could be very, very informative, not only for the deep sea ecology, but for understanding past climate change and whether we should be harvesting animals that are 1,000 years old in the first place.
But could we zoom in on the contact between some of these rocks and the sand just to confirm that this is a coated carbonate and not something else?
Absolutely. Let's try to get a zoom from here, Roland.
Yep, it looks like we still have carbonate with a coating. Yeah, and the little ones too. Thank you.
You're welcome. Come on up.
Following you down a little more.
Are we clear of that pillar? I think you are.
This is it.
Okay.
So yes, but I don't want to drop down below it.
Let's get this fish real
There's
quick.
another big one up there. Is that another tinsel one?
Yeah, another tinsel fish. A lot of tinsel around here, isn't there?
This must be Tinsel Town. Oh, he probably liked that red laser in his eye.
He probably doesn't see it because
Yeah,
a lot of these deep water fish don't have sensitivity in this red range.
Okay, come on up. Let's see.
Some rock pens, and I don't quite understand how these rock pens are perched. Oh yeah, see this clear, transparent, branchy thing
About
upper
5 seconds
center?
for my iris to react.
This is actually a soft coral and alcyonation. And Sako's with us, and I'm certainly hoping that she can give a hand with this thing.
Being photobombed again from the side.
Yeah.
There's a big rock well off to your port. I think it's sheltering you here.
Yeah, come on in, Roland.
Pilot ship move completed.
Thank you.
That's different.
Yeah, so the only one I know of, because there's only one species of soft coral in Hawaii, and this is not it, is Siphono gorgia. That's the only genus I know, and I'm hoping that perhaps Sako or somebody else, Tina perhaps, could render a guess even to what family this soft coral is in.
So you're saying this doesn't really occur in Hawaii?
No, this one does not occur in Hawaii that we're aware of. So Asako says she can't see the exact sclerites in it. There's a few that are running along the main stem, I think, Asako.
You get any tighter, or is that max?
And then there's a few sclerites you can see in the stalks of these polyps, or the branches of the polyps. Okay, she can see it now.
Come out just to hear. Yeah.
Partial.
These are just beautiful animals. Just look at this.
So a little bit more, we can frame both of these.
A few sclerites not arranged in a row. So she's working through the ID right now, just noting different things that are important.
You want to kill lasers?
laser's coming off.
All right, so Asako's going to work on this a bit and then get back to us on it. See if she can
rotate left, get this
dredge up an
yellow.
identification. Thank you, Asako. And we got some single polyp scleric tinians, cup corals that are around it as well. Looks like some plexaurids. Yeah, there's a, I think it's a plexaurid. This isn't a gold. This is one of the soft corals. A little sponge too.
Oh, you got a sponge today.
Well, I had one earlier too.
Oh, you did?
Yeah. So two types of plexaurids, or these Aguchi somnia, this yellow. Cup corals, sort of cup corals, multi-cup corals, I guess.
Come out just a little bit.
Another rock pen or two? Oh, another rock pen or 10.
Partial.
Let's get a frame of this guy.
Yeah, big gold coral colony right here.
Losing my toe.
So if I understand it, while he's framing this up, these rock pens on here, the way I understand it is their foot, the big blob down at the bottom of them.
Right, coming out.
It's sort of like a plunger, and that they can create suction on it, and that's how they can attach to the rocks. So some of those that are oriented vertically, they're not just sitting there. They've created a vacuum kind of underneath that thing. Well, not really a vacuum, but that's how they're managing to perch the way they do.
Better than the suction cups on soap dishes that you get that always fall down.
That's right. So it is kind of functioning. That's the word I was thinking of. It's functioning as a suction cup.
Pilots.
That's why you don't see them just floating away in this strong of a current.
Here a little bit, and then we can, because he's only 25 meters away from it. If you look at the Sirius sonar.
See, these rock pens are all over the bottom here.
Move. Yeah, but that's going to bring him 5 meters
to be above
from
it first.
So
Copy that.
I don't know how
move
steep
up
it
a
is
bit
or how tall it is.
Good copy.
He's going to spin around and get another look at this gold coral here in a moment.
Did you want to see the gold coral again?
No, that's okay. I just assumed that that's what you were doing. If we're coming on up, that's no problem.
There's a little urchin here in the center.
That's Cenopadina. We've seen that one already. Let's see if we got maybe the Anthomastus, that red mushroom coral, over to the left center.
Okay.
I don't think we've got a really good view of one of these yet.
That rock.
There's also an anemone in the top right. Come on in, Rowan.
Yeah, we saw that anemone. Yep. This might give Tina a chance to get a better look on a possible ID on this one. It's Tina Molodtsova, who's in our chat room right now.
And there's a little one of the rock pens next to it.
Yeah, so it's kind of forming a little suction disc. There's a kind of a good view of that base of the rock pen to the upper left that I was talking about, where they kind of form a suction cup. That's how they're hanging onto the rock.
Okay, let's come in tight. Do you have it? That's
Max zoom.
Yeah.
Gorgeous corals, these mushroom corals. Diva is going with the genus Anthomastus for this mushroom coral, Diva Mon.
All right, when you're good, Rowan. Big anemone center.
We could probably pass that one by. We've got a couple zooms of that thing already.
Look how clear that layer is at the bottom of that big rock.
Are
You
the
can see
lasers back on?
nice, nice obvious yellowish tan color of the carbonate.
Yeah, I think you're going to zoom in on this and get a good look at it. Yeah, look at that.
So it's clearly carbonate color. You can even see the shell fragments and pieces in there.
Oh yeah.
All the little coral bits, and then that black coating starts a little bit above it. So I think we're still in the same kind of rock material. We're just, for some reason, this is all coated in this black stuff, which more than likely is manganese. Although we're not sure why it should be here at this depth.
Oh, maybe the current flow that has accelerated the manganese deposition.
Maybe.
Higher current.
And so we have effectively still the same underground, but coated in manganese, and maybe that's because this is so steep that and rocky that it doesn't move as much as the sandy slopes we started on.
Down to the right of those red things at the bottom.
Yeah, another sea star up there.
Is this a sponge?
Now immediately.
Yeah, there's
lower
more of
left
these
where
ribbon
lasers
sponges.
should
That's
be.
really the only sponge we've seen today are these ribbon sponges
Now
that
center
that I think are post-Alastra.
You sound a little disappointed.
Not really. I am. You never know what the site's going to turn out to be.
What is that blue stuff?
It's blue stuff. It is. Roland, you no doubt saw this, and you're zooming in on it right now. So now you got to identify it.
This is what we like to refer to as blue biomaterial.
Oh, okay. So Roland has come up with an ID. It is blue biomaterial. I don't know what we're looking at. Maybe somebody does in the chat. These don't look like polyps. Whether these are foraminifera or bryozoans.
Could this be possibly a new sponge species?
I don't think it's a sponge the way it's moving around, but it could be a sponge, maybe a demasponge. They can encrust on rocks like this.
The blue blob.
And so Deborah's wondering what the blue stuff is too.
Do you have any more zoom? Or is that it?
That's max zoom.
I don't think it's a sponge. Where's Shirley Pomponi when you need her? She's on a Harbor Branch, and she's a demasponge expert.
Is that a little fish sticking out, or is that, no, that's just an optical illusion.
Yeah, it's a little tunicate, I think. Those are two little. Shirley's on vacation. Well, that's no excuse, Deb. No excuse whatsoever. That's Deb Blixen, who works with Shirley at Harbor Branch.
So could these be retracted stoloniferans? Do they retract their polyps?
I don't think so. I don't think they can retract like this. Yeah, Bruce, I have no clue what this stuff is.
Those rocks might be loose if you want to grab it, but...
That's right. We could do that. And then we have a little piece of carbonate, too.
Well, maybe zoom out. Maybe we can take a look at the rock. What do you want to try? I mean,
why not?
Why not?
Yeah.
We're doing pretty good on time from what I can see, and
it's going to be small.
Yeah,
that's okay. They're
well,
not.
They'll be good. They'll go in the bio box.
There you go.
Yeah.
And they're not volcanic anyway. There's not going to be much volcanic stuff here, so we can. We're not going to get a volcanic age out of these.
Now I just have to set up without blowing away.
This is a lucky rock. Normally they're not allowed to write in the bio box, but because this one actually has a relationship with some biology, it's going to get. It's going to get
the plushy ride.
Yeah, it's going to get first class here.
Normally they have to pay for that, huh? But he's got a blue passenger for you.
Yep.
Hydraulics up.
Bringing them up. Hydraulics are up.
So our field ID is, let's see, what did Roland say? Blue biological material? Is that what he said?
So this is going to be one bio.
Okay.
That would be, the blue biomaterial would be the engineering description of it.
Yes.
Blue biology material also works for the biological description of it.
This is going to be the port inboard bio box pilot?
I'm going to try that. I'm going to try...
All right, if I take your Zeus?
Yeah,
Zeus,
go ahead.
rather?
Grip force.
I think this is the smallest rock that I've ever heard a geologist ask for.
That's because it's more about the biomaterial and just getting a few pieces of the carbonate fragments inside for whoever's interested in that. Plus, it doesn't count. Remember,
yeah,
Bruce, it's going in the bio box.
rolling.
It doesn't count for my count of 2.
Yeah. So Sako doesn't think this is an octocoral, and I agree. I don't think it's an octocoral either.
Which one do we want?
The bigger the better.
Left or right?
The right one's probably easier.
Left or right?
The right one's probably easier, right, Josh?
I can get either. The left one looks like it has a little more.
Yeah, okay. If you can grab the left one, that would be good.
All right, come on out a little bit, Roland.
Partial.
It's tiny.
I'm thinking maybe what we should do over here is pick up a bigger rock too, so that if there are any sedimentologists that want to look at these at some point, we have a specimen.
Looks like it's cemented.
Is it?
Yeah.
Yeah, it's not budging.
That darn manganese crust. Or carbonate.
Josh, if I zoom out, there's also a rock on the left edge of the screen.
Yeah, these are all cemented in. All right, come on out, Roland. What are you talking about?
Just on the very left edge of the screen, directly left of lasers.
Oh, yeah.
Just a little blue patch.
There's more blue stuff on the rock that's all the way on the left edge of the screen, too.
I can't tell it's any more or less cemented.
Probably just the cemented.
Where are you seeing that, Roland?
Immediately left edge of the main HD.
It's sort of blending out in your screen, Josh, but it's the very middle left rock. There's some blue material on the bottom side of that one.
And I can't get to it. Well, it's worth the try. I mean,
well,
oh boy.
You know, the way I figure it is, it's good that we're leaving a few mysteries.
He's going to try and pluck it off one more time. Well, there it is. Oh, but the blue material came loose.
There's still blue on it.
There's still blue on it?
A little bit on it still.
Oh, yeah, I see it. Yeah. Yeah. We're good.
Using your left toe.
If a lot of it went floating off, does this count as a biological sample too?
Yeah, yes.
It
Still,
okay.
We're still collecting it because of the blue stuff.
Okay.
Zoom in, Roland. Tilt up on my camera.
Taking your main.
Yeah, see, it's still there.
There's something on there.
Holding.
Not much.
I think it's going to be a bryozoan of some kind, I guess. I don't know.
Oh, there's a fair bit on the other side.
On this side.
Right. So
DNA and everything, Chris.
Yeah. See, you
right,
probably
come
would.
on out, Roland.
Nav, do we have both bio boxes available?
That's correct, Copilot. We have no samples currently.
Thank you, Nav. Where would you like this, Josh?
Well, let's try to open the drawer if we can.
Octopus, top right.
So I should.
Oh yeah,
try both
how
bio
about
boxes.
that?
Yeah.
Okay,
right there.
Got it.
Uh-oh, Bruce.
And there's a Friday octopus.
Yeah, they'll get the rock. They'll get the rock secured, but there's an octopus right on the upper right corner of the screen right now.
About a third of the way from the arm to the edge.
A really tiny octopus.
That goes with Octopus Friday.
No, I think I'm.
Oh, and he's moving down into
moving
the.
down. He's coming
into
down
into the view. Yeah, there he is.
Now.
Oh.
Yeah, Hold on.
That's for Mike Vecchione. That's his his thing.
We don't want to open that box until we're stable.
Okay.
Just because if I run into something.
Should I close?
And you're driving Mike crazy right now because you can't zoom in on the octopus.
Well, he's settling in, so maybe he'll hang out a bit while we get this rock secured.
Biosample.
Right. Biosample, excuse me. Had to come off the bottom a bit in order to even open up the boxes to put the secure this.
Faster, please.
Full open.
Close.
Okay, the blue biological material is secured. So before we move off, maybe we could see if we can find that octopus down on the bottom of the screen somewhere.
You're clear to secure hydraulics.
Okay, I'm giving
and
you
can take your camera back.
Well.
Yep.
I'm taking my screen.
He went straight towards the bottom of the camera.
Where'd he go?
Hydraulics secure.
I believe he's under the corner of that rock.
That rock?
The big giant rock.
There he is. He's in the middle on the bottom, maybe? Oh, no, wait, that's something else. No, that's not him.
Can I zoom?
Zoom on what, Roland?
You could zoom in. Maybe he's cryptic right now, or we can't really see him from
You
this
see him
far.
in there?
I think
No, but I'm just wondering whether it's... If you zoom in right now, yeah, maybe that's him. No?
Nope, coming up.
He was headed in that general direction down the diagonal of that rock. From upper left to lower right.
There's a little fish there, too.
Okay, well, we did get an octopus for Octopus Friday. Oh, there's a big colony of something on that rock out there.
What is this?
Those are, I mean, some of these are sea pens.
Oh, that octopus is good at blending in. I guess they generally are.
We could probably move on, Josh. We're probably not going to find him again. He's going to be a tough one.
Is that a sponge? Quick zoom.
Yeah, another one of these ribbon sponges. Yep.
Right, coming up.
Good. I think it's in the genus Poecilastra. Current's even whipping some of the sediment up here.
That was my props.
That was your props, okay.
Oh, he went back up. Yeah.
Okay, we're moving on here. See if we can find some other stuff.
Blue stuff, green stuff.
Yeah, we've
octopus stuff.
We've got one rock sample already.
Cookie.
You're gonna let that go, aren't you, Jasper? Okay. Looks like a crinoid over there on something.
Well, it's just nice when someone brings this up earlier in the chat that they're hoping for Octopus Friday and the octopus is actually willing to show up. I think we're doing good then.
Lots of sea pens all over the place. These rock pens we think are either Calibelemnon or Anthoptylum.
There's more of this blue stuff on this rock down here.
Yeah. I think we're over the blue stuff.
20 meters in that direction.
Yeah, we burned one bio collection on it. I think now we're going to look for corals.
About 330.
That's a little weird, Josh, that very leggy coral right in front of us, the white one.
Like
I wonder if you could take a quick zoom on that.
20 meters, 330 degrees, 0.2 knots.
Is it just a bamboo? Oh, it's a bamboo from an odd angle. I got it. Okay.
Good copy.
Yeah, that should be sufficient. Thanks.
Copy. Come on up.
Not a tinsel one?
Copy that.
Tinsel one, yeah, maybe. Okay, here's something different. You see this wavering white forked type coral there? Pretty sure that's different.
It's just the wind.
Come on in, roll.
No, it's different, all right.
Huge polyps.
Yeah.
Those are translucent, aren't they?
Yeah, and you see how flexible it is too.
That, and I can see where the current's coming from.
So Scott would probably suggest Anthothylidae, based on the size of the polyps. I'm waiting to see if Tina or Asako have an opinion on this one.
Something on that branch. See that roll?
So like a baby brittle star.
Come in tight.
Going on the upper branch or the lower one?
Right
it's
of
like
center.
a translucent brittle star.
Plexaurid, maybe? I'm not really sure.
There's two of them there.
Or an anthothelid. Yeah, I'm not seeing anyone throwing out any IDs on that. That little ophiuroid on it, brittle star on the top. So I guess that's good enough. Everyone's got a good look at it, but we haven't seen a whole lot of suggestions yet.
Is that to the right? Is that a
I think it's a
squat lobster?
Crab in a shell.
Yeah, come on in.
Go ahead, watch it. I'm sorry, I didn't hear you.
Squat lobster there.
All right, come on out.
It's only going to take us about 15 minutes to get to 50 meters, so somewhere around 4:10, 4:15.
Full wide?
So just question asked whether or what time we have left, and so we're leaving the seafloor around 4:10, roughly. So that is not quite 3 hours, about 2.5, 3/4 that we have left of bottom time to look around.
And Nick, it's good
that's
to
pretty good.
give us
Yeah.
an extra 5 minutes or so to get set up.
No, that's another fish hanging out.
Yeah, the other Grammacolepus tinsel
I did
fish
co-pilot.
again.
It takes 15
I
minutes.
heard that.
Oh, you heard? Oh,
Yep.
Good copy.
Trying to see
Coming,
if there's
coming right at us. No.
to see what we might not have already imaged here. There's a fair amount of stuff on the seafloor.
See now on the chat, Bruce is correcting GSH to star FSH. That's the correction, or does he mean it's a starfish?
No, it's Asterixoid.
An asterixoid.
There's a nice gold colony right next to. Go ahead, Bruce.
We all have our favorites, and from my perspective, I would say the fishes are the stars of the video.
Quick zoom here. Snap.
Oh, look at that one branch, yeah.
Yeah.
There's a sea pen.
Oh, sea pen did become detached. Look at that.
But I'm going to sit here.
Let's see if you'll actually be able to make a landing.
Want me to zoom back in?
Didn't even look like you blew it.
Go ahead.
Oh, yeah. Oh,
there he is.
He did it. He did it. Now, is he going to stick himself to the surface? Can he do it that quick? No.
Nope.
Denied.
He might be able to straighten himself out, though, given a little bit of time.
See how the base is curled up?
Are you suggesting this was voluntary, Roland?
Suggesting he is waiting to reattach.
Okay. Yeah, he does look like the very edge of his feet is sort of rolled up over.
Pilot ship move completed.
Thanks, Snap.
Snap zoom here.
Oh, one of those guys.
Oh, you've seen
there's Cenopadina Pocella again.
Come on out.
These are really pretty urchins.
They are.
Yeah.
Another full gold here. Coming tight.
So yeah, I want to see what that brown mass is on it. Some more of the gold.
Yeah, come on out.
There was just another species of zoanthid on the left of it. Yeah. No, it's one of those crinoids.
Massive balls of crinoid.
Yeah, coming in on the crinoid.
Maxim.
Almost like a ball of yarn.
Yeah, it may not be that many arms. It's just the way they're curled makes it look like there's a lot more than there really is. I don't know, maybe when the current settles down, they unravel those arms more because maybe they just can't, they're too delicate. Maybe they can't feed effectively when there's a strong current like there is now. I don't know.
It's this windy day mode.
When you're good rolling.
Full specimen? Video's clear.
Right, coming up.
So we're spinning slightly to the north here to look down along this ledge that we've been sidling against for a while. Yeah, no, I'm looking at that. What is that?
Looks like a fish.
I think it is too.
You see it, Josh, just right in the middle?
Yeah, it could be a scorpion fish, but it could be different. Yeah, it's a different species of scorpion fish.
Hold on 1 second. All right. Just get tight on the head.
Maxim.
So yeah, Chris, it is a different species of scorpion fish from the shape of the head and all. I almost think it's a pontinus that does not have the long cirri above the eye. I can see short cirrus above each eye. But it's got the kind of concave head, the fairly large head that this large-headed scorpionfish has. On the other hand, it looks like it might be a fairly small fish, so could be a different kind of scorpionfish.
Any chance it'd be in that genus, Bruce? or do you just want to potentially leave it as Pontinus? I don't know how relevant the shape of the head and the size of the head are to going to genus.
Yeah, I should probably err on the side of caution. It could be, a juvenile Pontinus, but it could be something different. Scorpionfish are a lot of species, and they're very difficult to identify without the specimen because some of the first characters have to do with the tooth patterns inside of the mouth, things that we can't see from the videos from the ROV.
Yeah, thank you, Bruce. And he's nestled right up against a Bathypathes, a black coral. So that fish is being coral bombed.
Yeah, come on now, Rowan.
Coral didn't even have to move too much for that.
No.
Now, the other thing I would say is that the color looks a little different than the Pontinus that we see. The color and color pattern seems different, so could be something else.
Okay. Those are just, sorry to interrupt there, those are just the Alfonsinos again.
Curious fish.
Yeah.
Okay, come on.
As we noticed earlier when they swam right up into the camera.
Yeah. There are a ton of these rock pens along the face of these rocks. They are all over the place. Something just went shooting through, almost looked like a sternoptychid, but
another rockfish here, snapping.
Yeah.
Yeah, it could have been that could be the rear end of the fish that we just were looking at. Not sure. There's a little sponge to the left. Possibly a small bamboo coral or something right next to the scorpion fish.
All right, you're good, Roland.
We're still keeping our eye out for a possible gold bamboo collection, a combo collection where we get some gold and some bamboo. It'd be a colony of a bamboo that isn't completely overgrown. Haven't seen any.
That's pretty stretched out. Would you like to keep moving in that direction?
Since the suggestion was made to try for that.
Since the insurmountable cliff.
Yeah.
Copy that, co-pilot 20 meters bearing. You want to do 330 or 320?
330 is fine. It keeps us off the.
Let's come up a little bit.
Copy that.
Oh, let's see.
Coming up.
Bridge is net. I'd like to put another ship move in. 20 meters bearing 330 at 0.2 knots.
A little epigonid. One of the deepwater cardinal fish.
Okay, come on out, Roland.
I wonder if could you pan just slightly to the left, Josh? And there was a. Yeah.
Copy that, Bridge.
I guess these are corals just lined up along the edge of this rock, but I wonder if we could just take a peek at these.
Sure.
There's something that likes to crawl along the edges of rocks like that. I just want to confirm it's not one of those.
Okay, come on in, Roland.
These all look like they're plexaurids to me, I think. See if Asako has any differing opinion. I think she called what could be the same thing earlier a plexaurid. A lot of the polyps are closed, sort of some are closed, some aren't. Yeah, thank you, Josh. That should work.
Right. When you're ready, Roland.
Couple mushroom corals on the rock and
our friendly neighborhood fish.
So we're seeing some relief on the west side of this feature as well as the right side.
Come up a little, Dave.
Yep.
Still have these massive blocks, and you can see that it's made up of all these little different size pieces.
Oh, look at the spire or whatever, this tower right in front of us.
And this is why I keep thinking this is this weathering pattern. This is a large-scale sort of weathering process that made some significant headway into whatever reef deposits were there before.
It's a cool shot from Sirius.
Yeah.
Oh, yeah, look at that. If those of you who have camera 2, you can switch to that. You're going to
feel free to come in
see
if
quite
you'd
a
like.
fascinating picture of the ROV in front of this little pinnacle here. Wow, that's gorgeous.
Is that some purple coral along the edges?
Pilot's move completed.
Would you like to look at these purple corals?
Yes, please. I
Roland spotted that.
Yeah, I was so engrossed by the Sirius. Glad you pointed those out, Josh. Appreciate it.
Oh, that was Roland.
Oh, it was Roland. Okay, thank you.
Come on in, Roland.
Very large batch on the top left.
Yeah, hanging off the edge of the rock.
That's what I'd call a precious coral.
Looks like lavender.
Sure does.
Very large. Shrimp with claws? Squat lobster?
Yeah. He's poking at something.
Is it an amphipod or something? What is he doing?
He's eating something off of there.
Cleaning what looks to be a... Maybe he's got his antennae stuck?
No, he was trying to put it in his mouth, I thought.
On Sirius. Thank you.
Well, let's see if he eats another one.
Appears to be very sticky. It looked like it's stuck to his claw.
Oh boy. Sako or Tina, do either of you have, or Abby, any of you have any clue what type of coral we're looking at here? Almost looks like it even could be a scleractinian of some kind, but I can't see that. I've never seen
better on the upper left.
There is a purple type of Enelopsamia. It's called Pusillus, or Pusilla, I think, but it doesn't quite look like this, and I'm not seeing the branching pattern, in which case it, what are our other candidates here? Wonder if we could look at another one of these colonies,
Sure. Come on out, Roland.
Josh, and see if we can get a little more insight as to what it is. This could be some type of amphiphelid, maybe. Sako's working on this too, trying to figure out what these things are. beautiful species, obviously. Asako suggests it could be a Xeniidae. I've never heard of that family.
Come on in, Roland.
Would you like to Google Xeniidae? Maybe we could see if
we can look.
Yeah, actually,
You've got Google in your hands too.
Yeah, I forgot. I've got my iPad here that I could look it up in.
It's already gone. I look more. Oh, here's it. This could be.
Like that one there.
Yeah, maybe.
There's a barnacle growing on its head. Come on out just a little bit, Roland.
Look at that. This guy's doing the same thing. He's trying to poke at the little polyps.
Yeah. It's a bit different. Previous purple one could be the one. I think they're the same thing because of that white branching with the purple polyps coming off it, and there's not a whole lot of grooves or ridges or anything else on the branches.
Only if you attach a lot of rock to it.
You know, I'm certainly tempted, Roland, to consider this, but
if nobody knows what it is, then you should take a piece.
Josh, there's also a worm on the right edge of the purple.
So barnacle and squat lobster.
As it goes into the rock.
Yeah, so Sako's saying that this is not a Xeniidae, assuming it's the same colony. Again, it could be an anthothellid, I guess, is one possibility.
The attachment of this to the rock also looks fairly unique, doesn't it? It's very distributed.
It's definitely an octocoral for sure. That's as far as we can go at the moment.
Not this one. It's not very clear.
There it is.
It's still.
So what is
it? Not helpful. A white-branched octocoral with stunning purple polyps.
Okay, now we know. So Asako thought, anthothellid is a possibility for this. I don't know, is there any desire to consider this collection versus the gold and the bamboo, or I'm open on that. I know the gold and bamboo would be at least as important. But this is a pretty interesting coral that we've got in front of us right now. Anyone want to weigh in on this? Abby, perhaps? Asako, Tina? Any of you? Bruce, any of you care to render an opinion? Should we hold off and go for what we talked about before, or?
You already know my opinion.
Go ahead, Bridge.
Should we go for full specimen?
Yep, come on in.
So there's a fish swimming by.
Whoa. That was little photobomb.
Copy that, Bridge. Yeah, go ahead. Adjust your heading as you see
Just
fit.
we'll move on.
And,
We didn't see a whole lot of support for collecting this.
Copy
So
that, Bridge.
Can still keep in mind that
heading.
gold coral
Pilots,
bamboo
Bridge
combo.
is making a 20-degree heading change about the
Yeah,
front.
Abby, of course,
Copy, just
she
for current
wasn't
wind.
weighing in because she's totally biased.
I think
Just
you guys
joking,
all
Abby.
are.
Yeah, we all are. You know,
In fact, the geologist should decide what to collect because you guys are all just interested in your own thing.
Yeah, I'm holding out for a sponge.
Huh.
No, I'm
How
kidding.
about this?
No sponges. It's this one.
On the NOAA Ocean Explorer website.
Yeah, it's still delicate purple octocoral polyps, which is the same thing. Anyway, the moment has passed. We'll keep in mind the bamboo and the gold coral. I think that's actually a more valuable collection in some senses. So
looks like your potential chance
whoa,
down there.
some big coral trees coming up. Excuse me, could we zoom in on this red fish that's directly in front of us before he takes off?
Come on in, Roland.
I'm sorry, he's just a Decadactylus. I thought he could be one of the other bottom fish. I mean, he's a barracks. Excuse me.
Right, coming up.
Okay, thank you. Sorry.
Let me ask that person.
Yeah, I don't know. Everybody's describing the purple octocoral. That's really pretty. But Masako is sort of agreeing with our decision to move on and hold off for the bamboo and.
Let's get this fish here.
No, let's get this fish actually. Okay, it's a beard fish. Sorry, thank you.
All right,
might be.
Come on out, Roland.
The polymixia.
That coral there, is it the white and gold?
Yeah, they're looking for a branch that has the white and the gold on it.
Yeah, I know. That's.
And that branch is just all gold. Coming
Right.
partial.
You have to.
I mean, I could take a branch of the gold and then go up and take a separate piece.
Well, let's
Gold
look a
appears to be
little
the
bit
base.
further. I think we're going to see these all along this wall as we head
Yeah,
toward
copy that.
waypoint 3.
Come on out, Roland. How's the ship doing there, Ned?
It's got about another 10 degrees for the heading change. A few more minutes.
Start at 120, didn't we?
Is that co-pilot?
Yeah, didn't we start at 120? Should be
The
about,
ship? Yeah.
Should be about done with that.
It's still coming a little bit, I think.
Copy.
They said the current and the wind coming out of 150.
I misheard.
And they're at 140, almost 140 now. Go ahead, Bridge. Copy that. Pilots, bridge is all settled out, ready for a ship move.
Hey, what do you think, co-pilot?
Do you want to try to move up this, or
Yeah,
continue
we're gonna-
along it?
Translucent fish in your center screen.
The little guy?
Yeah.
How about up to the upper left?
Come on in.
So you.
Okay. Is this
Yeah,
what you were
Bruce
talking
is. This is what I
Yeah.
thought could be. I think they're called sternoptychids, but I've forgotten what the name of these transparent fish are.
Nav, it looks like about 315 would follow the contour. If we want to go upslope, it would have to be more to west.
Is the name of these
Not
things?
sure exactly what we want to do yet, so just stand by.
Well, I'm getting some screen freeze here, so I didn't get a good look at it. It looked like it had clusters or two of photophores along the ventral part of the body, so they could be cernoptychids.
Okay, maybe
some possibilities would be
275.
something called argyropnus. Another one would be Morolicus.
Okay, I'll turn
but
to that.
honestly, I didn't get a good enough look at it.
Is there something you wanted to see here, Chris?
Even if it was a cernoptychid, it's a good possibility.
Yeah, pilot, you see that one colony that's up on the right in the center right now that's got some gold on it? I wonder if that would be a possible.
Now let's do 300 even.
Yeah. And
copy that, co-pilot.
I think it'd be fun, real challenging for you to get in there and collect it. That's why I mentioned it. Thanks, Bruce. So, cernoptychid is one possibility.
How's my tether? Good.
It's
trying
good.
to see
Put
the
a ship
gallery
move in,
here.
20 meters,
you.
bearing
Oh,
300
fine.
at 0.2 knots.
Let's show you the tape.
Good copy.
Yeah, my screen throws up at just the wrong time for that one, so yeah, didn't see it.
Possible good specimen.
Yeah, I
lower
can't find
center.
a picture of it in the older Hurl gallery. Yeah, that one. I was just wondering if there's any, or even the one that's beyond it, the center.
That one I don't think I can get to. That's looking at Sirius. That's too far down.
Well, would it be possible? It's a sort of the same colony. Would it be possible to snip some of the gold and then snip a bit of the bamboo from the same colony?
Yep.
Maybe that's the solution to it.
How do you feel with me being down here?
We're sort of on opposite sides of this peak.
You're comfortable with it? Should we move the ship?
I'm far enough from everything. I'm just worried if there's a shift, the tether might end up in it. Other than that, I'm okay.
The ship is heading where you're looking, or
yes,
more
it
to
is.
the starboard?
No, directly where I'm looking.
290.
Oh, 300.
300.
About here.
So that should put you in a better position.
Okay.
Put you more on top of me.
Yes, it will.
Okay. So I'll get set up here.
Do we maybe want to stop that move early?
How long was it?
It's 20 meters.
No, that's good.
Good.
Looking through that coral, Josh, I think I see a yellow one behind this through the
Yeah,
white.
I can't get back behind it.
Yeah, Chris, go into the Hurl guide and look under gonostomatidae. You have the name misspelled as goniostomatidae in the Hurl guide, but there's a picture in there of an argyropus that is very similar to what we saw.
Yeah, actually, Bruce, I just found that picture. I've been looking for it for the past few minutes. So that's what I think is a better fit for it. The goniostomatid, I agree, the argyropus.
Yeah, so as it happens, you've got the family wrong in the Hurl guide. Argyropus is a cernoptychid.
Oh, it is? Okay. Like right or before or after this, we're going to get a bit of a watch change here.
I think if he clips it here, you'll get both on one branch.
Yeah, it's whether or not that's too big for the basket or the box.
Yeah, I don't know.
The problem.
Come on.
I see.
All right. hydraulics up.
Spinning them up. You should be good.
Oh man, this is just.
Ship move completed, pilots.
Yeah, copy.
Yeah, I'm going to end up directly over you, which is all right.
Another one of those
tether.
little fish that could be the Argyropnis just swam through the scene, Bruce, but it was not as good a view. But hopefully your video won't freeze right at that moment.
Yep, I saw it. It didn't freeze, but you're right. It wasn't as good a view. It's a possibility. There are a couple of other sternoptychids too that could be, but Argyropnis seems like a good bet. That's another species in a family that's usually thought of as in water, but this particular genus Argyropnis is more bottom associated than a lot of the other genera in the family. And most of the specimens that have been collected with bottom crawls instead of midwater crawls.
Thank you, Bruce. So they're lining up for this collection. I'll do my little general announcement. You're looking at a live feed from the NOAA ship Okeanos Explorer. We're on the east side of Wake Island right now. We're currently at a depth of 459 meters. We're about 10 degrees centigrade, dissolved oxygen's about 5 milligrams per liter. And we're inside the wake unit of the Pacific Remote Islands Marine National Monument. And obviously, just like yesterday, we found a pretty interesting spot here. In this case, we're in much shallower water than we were yesterday, and we found precious coral bed here, specifically gold coral. We haven't seen any of the corallids.
Should look at the base to be sure.
That we saw yesterday?
That we saw yesterday. So every place is different, but this is gold coral that you're looking at right now, and it's overgrowing a bamboo. And
So if you look at the base here, Chris?
Yeah.
So, I mean, are we comfortable with me grabbing... I was going to try to get two branches to both parts at the same time, but if you look at the base, I'm not sure if these are two separate corals or what. See where the gold is versus where the other ones are?
Right, they could be connected or they could be different. I'm not really sure. Well, can you snip off just one branch of the gold and then just get a branch of whatever the bamboo is here, even if it's a...
Yeah, I can do that. Come on out, Roland.
Tilt up, we can get a good image of that.
Up?
Yes.
Prior to the gut.
Coming up. Holding. Do you want any other camera views?
No.
Probably should go two separate grabs.
I can't do that, Roland.
Maybe just snip this first or something, and then...
I can't do that.
Oh, you can't? I see.
Because I'd have to...
We can't open the box
right here. Can't open the box right here.
That's a point. That's right.
Yeah.
So,
I think you need to find a trickier place for him to get a sample.
I think he's
getting there.
He's almost there. And he did it.
Great. Very nice.
Great job, Josh. I didn't realize it was quite that difficult. I suggested.
All right. Oh, no.
No, no.
Oh.
Did someone write that down early?
Should have just put it in the box.
Can't believe I did that. That was a user error. Oh, man.
Well, we could try to find another spot if that's... Cause
try one more attempt.
You want to try one more? Okay.
Yeah. Oh, man, I can't believe I did that.
If this works, you're going to end up with a lot bigger sample.
Yeah, this is actually going to work better. She got it?
I don't know.
Yeah, he's got it. Beautiful. Well, it's close. Barely hanging on there. That bamboo. Good enough.
It's hanging by a polyp.
Slipping out.
There it goes.
You know what, let's just go ahead with the gold and put that in the box, because that's a valuable collection.
Might be able to pick up the other piece later. Yeah, it's sitting right there.
Yeah, maybe.
Oh, man. All right.
I think I see a lot of mucus on this. It might be sticky.
Yeah, bring up Mini Zeus and open the drawer. We'll do starboard side.
Copy. That's the wrong one. All right.
So this is a particularly gooey coral too. Just to warn you.
Lovely.
Okay, opening starboard bio box.
And you have the rock sample on the starboard side as well.
We don't have a rock sample.
Import, isn't it? The blue stuff.
Yeah, we do.
No.
Yeah, sure we do.
Sorry,
no, it's a biosample. It has a rock commensal.
Okay, extending drawer.
That's right.
I'm sorry, I did that backwards.
Just leave it. Just leave it. Just open it.
Okay.
It's like it's going to go into port outer, right?
And look what I had chosen.
You had chosen it. That's amazing.
That's actually a standard setting. I hadn't actually chosen anything.
Oh, okay.
If you'd like, I can zoom
There
in
it
with
is. It's in the
HD2 and
box.
show that it's got the other coral also with it.
Do you want me to press collect now or wait until
Close
the
the
other one?
box.
Okay.
Press
There's
collect
close
now.
up. Sorry about that, Josh.
Yeah, we got to figure out the box and the pins.
Yes.
before I'm going to go put it in the box.
That was my fault too, co-pilot. I thought we had.
Oh, okay. Thanks, Roland. Roland was just saying that he thought he saw a small piece drop in the box anyway. You can see the piece sitting right against the rock. I just don't know how difficult this would be to grab, but I think Josh is going to go for it. Couple closed mushroom corals right above that
They just
branch.
saw what happened above their heads, and they're like, not me.
So you can clearly see with that shot that that those anthomastus can actually retract their polyps totally right down into the ball.
Yeah.
Josh has got the branch. Oh, and the squat lobster took off. That's okay. That's a smart squat lobster.
Oh, there's a little hermit crab running around on the bottom there. That was fun running off the black box in the corner too. Yeah.
Oh, really?
We're going to put this in the same box.
Okay.
Right, go ahead.
Opening drawer.
No.
Closing drawer. I'm sorry, I've got myself confused.
Is it starting to come out or something?
The lid wasn't opening with, or
the lid
stayed
wasn't-
closed.
Oh, okay.
Full open.
Clear.
Nice.
That's
done.
terrific, Josh. Thank you very much for that.
Yeah, sorry
That's
that
a great.
took so long.
Oh no, that's terrific. You got it.
You made Chris's day.
Yeah, well, he's going to make some other people's day too. That's a really good specimen. Yep. Okay, we're waiting here. We're having a bit of a front row change out right now. Josh was hanging in there to finish that collection before they switched the front row. Yeah, it does count as one. We think it was the same colony, just two parts of the same colony, and so it's actually really nice to be able to get a sample of the gold coral as well as its host bamboo coral. So
Good afternoon, Watch Leads.
good afternoon. We got Chris.
Chris.
How you doing, Chris?
Good, how are you?
I'm doing well, particularly after that. That was a bit of an effort, but it was all worth it in the end there.
Interesting collection.
Yeah. So Pilot and Nav, there's a question in the chat about how much more time we've got today.
I can answer that in 1 second.
I think it's probably about an hour and a half, maybe 2 hours. I'm not really sure.
Oh yeah, almost 2 hours. Yeah, watch lead, almost exactly 2 hours.
Roger that. So we've got 2 more hours, Tina, that will be on the bottom.
What's our next direction here? Is that to be determined?
That's affirmative. Waypoint 3 from Sirius, 295.
He's talking to me right now, Rowan.
Yeah, so Sako, yeah,
Go
the
ahead,
collection counted as 1, and
So
then
the
we also got
situation
that blue
is
material, so we've
Sirius
actually
to
finished our
waypoint
biological
3,
collections
relatively
today.
northerly, about 295, 300.
Okay.
Hindsight, I wish that we would have seen that beautiful purple
Yeah.
coral
So, you guys wanna
first, because we
yeah,
would
we
have
could
skipped
get
the
one.
blue
Acquire
stuff and
ridge, or just go
probably
down
gone for
acquire
that
ridge?
purple coral, but maybe
Let's go down, try to acquire.
not sure we're going
I'm way
to
off the bottom now.
see
Maybe we
that
can,
coral again, because we're not
get a
going to be
little north
diving
move
at these
going,
depths
just
again.
see how that pans out with sonar. Or a little west of north, what do you think?
Yeah,
Yeah, I was thinking a little west of north.
Sako, we
but
tried
depending on
hard
what you
to
see
have
here.
that blue one count as a geo collection,
You're
but
looking
there was
west.
Yeah,
a little
I think that'll
resistance
change.
from
See.
our geologist. There's one thing, you know what? So Jasper isn't here right now. He stepped out to get a quick drink. Can I may go consult
maybe like
with
a
Brian?
315.
Given that we
Yeah,
are
that
not
sounds good.
going to be collecting any rocks probably today, I wonder if we could get a
30
third
meters,
biologic.
okay?
And
I
I
think
will
that's good.
consult with him to see if that's possible.
I'll call it in.
Copy that.
Ready to draw of your nav. Ready for a ship move. 30 meters, bearing 315
Yep, I can
degrees,
rotate to
decimal
315.
2
I'm at
knots.
300 right now.
Okay, copy that.
It's a good copy, Bridge.
Watch Lee, this is Pilot.
Roger, pilot, go ahead.
Hey, we're just getting another ship move in. As you can see, kind of next to the spire, there's a little divot goes down. There's not much to see down there, looks like, so I'll check this out while we're waiting on the ship to move.
Yeah, if you see any of this sort of purple
Move initiated,
polyps
good
with
copy,
white
thank you, Bridge.
branches, if there's any still on here, I may duck out of the room briefly and talk to Brian Kennedy a bit. We saw
Okay.
a very unusual coral that nobody seems to know what it is, and
Okay. Well, we just got a ship move in to leave this, so.
yeah, well, I think we'll probably see some more of these
Okay.
things. So
Copy. Go tilt down for me, co-pilot.
Yeah. Tilting down.
Did you see their locations before, pilot? The purple stuff?
I did.
Okay.
Yeah.
Yeah, probably just be aware that puts you right underneath me.
Yep, go back the other way.
To boldly go.
And you can just tilt out as I kind of come this way. Get going in the direction of the ship. I don't want to go down in there. Like down where the spire is, because I'm a little worried about the tether following me down and getting stuck on the spire.
Snag on something.
Yeah.
So I just spoke with Brian Kennedy, the expedition coordinator, and Jasper's back too.
Howdy.
And so if the question was, if we're not going to collect 2 rock samples at the site, is there a possibility of subbing one biology for one of the rocks on there? And he said that he did not have any problem with that. We're not exceeding a 4 sample total collection here. So as long as you were comfortable with that, I'm not sure if how many rocks you were thinking
I
of,
was
but
just thinking we would get one if we see a nice big piece.
Okay.
And then we'll keep that trade in mind some other
Okay. Sako thought she saw that purple coral near the top of that spire, but I'm not sure I saw it. We sort of pulled off that, and we're moving right now. I think we'll see some more of these rocks.
Yeah, if we think we'll see more, we won't stop that ship, but we already are underway just to keep things moving.
Yeah, that's fine.
Okay.
I appreciate it, Chris.
Sure.
315.
315, aye.
Yeah, I think Sirius is moving.
Copy. You can lose the skids rolling as I kind of look down. I don't want to just look into nothing.
What's interesting here is that we're now all of a sudden back in the terrain that has just these light colored carbonates. We sort of lost the black coating.
So would manganese be so patchy in its deposition? It seems odd that you'd have it in one area and then just a few.
Ship has about 5 meters left in the move. Pilots.
Okay.
So I can call another.
Bruce, I think that you can sort of see there's a few peaks here that are a little darker.
Can we tack on
I have
another
a feeling that it
30
has to
meters
do
to
with
that
the
move,
steepness of
bearing
the slope. The
315?
really steep, tall structures don't have all this sand coming over the top. I think the sand, it doesn't look like it's moving, but on geologic timescales, this stuff is moving down slope. And so I think it has something to do with how much of the sand sort of scours the top off and keeps it clean, essentially.
Yeah, that makes sense. Thank you.
Roland, you can really see the water moving past there.
Good copy. Thanks, Bridge.
Is that me or?
Nope, that's
Nah,
the
there's been a
warm
pretty strong current.
Okay.
At some point, Josh is going almost 100% just to stay in the same place.
Okay. We're just seeing what?
Seeing the shimmering?
Yeah, the shimmering in the water. Not from a...
It's really bad when we zoom in. It really...
Oh, okay. Okay.
Bad.
That's good enough.
Pilots have called in another additional 30 to that move. 315.
Copy that.
So yeah, it's moved 30, so it'll have another 30 to go.
Yeah, Nasako in the chat room is remarking that there's a lot of seep ends here in the sand. Here we're getting back to some of the bigger, taller rocks sticking out a little bit more, a little bit darker.
Yeah, it looks like up ahead there's a lot more rocks here.
Yep.
Come full wide.
This looks more like the
Thank you.
little area we just came from.
Any of these?
Those are some huge blocks.
There's a fish. Do we want to look fish?
I think we've seen about 30 of these at this point. Bruce, correct me if I'm wrong.
Curious guy.
Yep, this is another one of the Alfonso and the Barracks, and we have seen quite a lot of them, so we can keep on looking. Looking for other fish, I meant.
Yeah. Whoa, this is a big overhang here or something.
Yeah, big boulder. You can see the end of it in the manip cams, but there's a significant overhang there.
That is huge.
An overhang or just a boulder that's...
Chris is going for a boulder. I said overhang. I think it looks kind of curved there underneath it, but... Either way, it's giant.
Yeah. This is more barracks, I think, barracks decadactylus that I've ever seen in one place before. They like this terrain, clearly.
What do they eat? Little fish?
They
It's
eat,
like ship has maybe
Bruce,
9
maybe you know,
meters
they
left
might
of the
eat crustaceans.
Copy that.
Maybe little fish.
You guys still
some
thinking
type of predator.
315 or maybe a little bit northerly? Or
I think 315 looks pretty good.
A little bit more
Yes,
northerly.
Tina's asking about these things are on top.
That also sounds good.
Push us, yeah. Just push us a little closer to that.
Pilot, could we take a quick peek at those little wispy corals on the left side of this boulder?
Wispy corals.
Yeah, they're
yes.
sticking off of it.
Sure. Come back down to him here. Got a fish.
The fish is
Playing
going to
chicken with us.
photobomb you.
Kind of pretty terrain with
you
all
should
of these.
turn, buddy.
I think he's
rock
going to swim
pens.
into us again.
Yeah, he is.
And these ones just dead ahead, Chris?
Yeah, that's
Okay.
correct.
Come on in,
There's
Roland.
a request from the chat to take a look. I think these are bamboos. Got a funny yellow base to them, maybe in Keratoisis. Yeah, that isn't gold, some other type of parazoanthid.
Trying to push in a little.
Yeah, they're bamboos, all right. You can see the nodes. There's a nice shot of that kind of slightly discolored
Bottom of the
bottom there.
stem.
I think they're internodal branching. And Yasaka was remarking, like I did, of the huge number of sea pens behind the boulder. Just amazing view. I don't know, Abby, if you're still watching, let's see, I'm not sure she's on anymore. She might have slipped off. Let's see, yeah.
Yeah, you can really see that rolling. That's weird.
Yeah. Really strong current, or I think it's, I don't know.
Yeah, thank you very much.
Or
That should
temperature
work.
difference.
Yeah, the current's really not.
Some funny kind of bamboo that's different than the one that the gold was attacking.
Maybe walk down towards the base, towards the right.
Sure.
See if we can see the color change.
Yeah, look at that.
Oh, is that what they were talking about?
Yeah, that yellow color right there at the
Gotcha.
base.
Yep.
That's a great shot of it. Whatever that means. I don't know. Yeah, Asako thinks it looks different than the bamboo that the gold coral was overgrowing, and I would agree. I think these are branching at the inner nodes, but I'm not positive.
Yeah. Yep.
Okay,
Doesn't
copy.
Yeah, science is good, pilot, whenever video is.
I'm thinking
Can we look?
Clear.
Let me know,
Can we
Roland.
look slightly behind this thing up on the rock? I thought I saw something right behind it.
Sure.
It's kind of small. Never mind.
thinking 340 pilot.
Video switching out.
Copy. Jasper, what was the item?
Nah, it was tiny. I don't think it was anything.
Okay.
This is a really amazing scene we're looking at. It's like there's hundreds of these rock pens all over the seafloor here, in amongst with the Decadactylus, and there's a Holardia, I'm pretty sure. reminds me of that volcanic pinnacle on the southwest side of the island of Niihau that we imaged last year, where they had these rock pens all over the place.
And here we have a nice eclectic collection of black rocks that are probably, again, manganese coated slightly, and a few lighter colored ones that came rolling down later.
Another one of these tinsel fish. They've almost been as common as the
Is that
barracks.
a fish back there?
Oh, excuse me. That striped fish under the boulder?
Fish.
Yeah.
Go ahead, Tara. Let's see if we can get it before he
This is a good
fails.
When he bolts. Ooh, that's a pretty one.
Hold
oh,
there.
there's another one. Oh, no, that's a shrimp or something. He's going to go behind that little rock there.
Yeah, so that's a little sea bass called Plectrantheus. There was also a nice shrimp above it. That, well, I'll leave the shrimp first, but anyway. So now we're starting to see some of the fish, or at least one of the fish that's a member of family, more often thought of as a coral reef fish family. But this Plectrantheus is one of the deeper living genera in the family of the basslets that are seen around coral reefs.
Bruce, do you think this is the same one as that undescribed species in the Mariana or anything like that, or is this something quite different? I can't quite remember what that other one looked like.
Yeah, I really can't say. This one looks like it's a little different from what we saw in the Marianas. And I don't know, when they were talking about the undescribed species, that it might have been in a different genus, Odontanthius, but we would have seen Plectrantheus there too. I think more than one species. There are almost 50 species of these things worldwide, so it's going to take me some work to figure out what we're looking at here.
Thank you, Bruce. And that really clear anemone-looking thing could have been a Corallomorpharian on the wall. Really
That's what Tina was saying, too.
Oh, was she saying that? Yeah. Maybe just a quick pan up. See this kind of clear thing with the white tips?
Oh, yeah.
I think this could be a Corallomorpharian.
Okay, go ahead, video. Stand by. Copy.
Like there's foraminifera all over that wall. Yeah, my guess would be Corallomorpharian. I'm wondering if Tina's going to hold to the same ID now that she's getting a better look at it. So right above it, right on top of this Corallomorpharian, if you go pan up just a hair, what is that? It's got legs, doesn't it? Right there.
It's a sponge with legs.
It's like a crab with a sponge on top.
Above and to the left.
Yep.
Is that
see it? Stand by. That's Maxime, yeah?
It is.
Copy.
It's a crab, and he's probably carrying a piece of sponge. It could be one of these small homolid crab.
Well, actually, I can't quite get any closer because of this inconvenient rock here.
Okay. No, thank you. It's good.
Coming on.
Succo saw a cup coral in there, too. Another one of those sponges, the demasponges, ribbon sponges.
So yeah, Pilot, what would you say the most?
So this field here, we should also look around to see if there is a rock we want to take.
We kind of want to end up
close
to waypoint 3.
Okay, yeah, you can start cheating that way, maybe.
Yeah.
Yeah, that sounds good.
Okay, like a 350 or something.
Well, I didn't get a chance to answer the
I think it'll be
question
fine.
earlier about what the barracks feed on, what the alphonsines feed on. There was something else going on that kind of took me away. but they are carnivores. They feed on small animals, and they tend to feed more up in the water column than off the bottom. So they'd be feeding on a lot of the little things that we saw in the water as the ROV was approaching the bottom, including some of his little fish like the sternoptychids, small individuals of those, and myctophas.
Did you want to look out at 350
particularly
before I call
on
it in?
small crustaceans.
Yeah,
any
pilot.
little squids
You comfortable if I rotate?
that would be in that,
So pick it up on sonar, see what you see.
Yeah, let me come your way a little.
Copy that.
They might also feed on
I'll rotate with you.
zooplankton, but we don't really know that.
Yeah, as long as we
so they're
follow this.
carnivores, and they forage
I just don't
more
want to
in the
come off
water
the feature,
than
you know?
on the bottom. And I commented at the
Yeah, I
beginning
mean, I
of the
think
dive
it'll-
that we were seeing so many
think it'll,
small animals
in the end, follow the
that would be a
position.
good food base for some of the carnivorous fishes. And sure enough, we're seeing
I was
a
thinking
lot of these
350.
berries here. So that kind of matches up with my initial thoughts.
Yeah, thank you, Bruce.
So that's 3-4.
You know, we in all of the fishing surveys we
about
did many years ago, we
350.
never caught one of those barracks, so we never had
yeah.
any idea
Bridge
of
over
their
now.
stomach contents, what they could be eating.
Hey, Pilot, just so you know,
either.
your starboard light is
Ready
out a little
for
bit.
a ship move.
30 meters.
Your light bar?
Bearing
I
350
didn't know if you knew that or not.
degrees, 0
I
decimal 2
did not
knots.
know that.
Do you want
Okay.
it in, or
More
the other one's
gold on
symmetrical,
that
or
rock,
happy as is?
big bamboo on
It's
the
okay
top.
where it is.
Alright.
And
That is
Good
correct.
to know that. Thank you.
about 100 or so of these sea pins in view right now, all over the bottom.
Come back down the bottom here. Sorry, watch these. We were figuring out the correct way of travel here.
Move initiated. Good copy.
Any
Thank you,
rocks
Bruce.
here, Jasper?
I'm looking. It'd be nice if we could pick one up.
Yeah, but I was kind of hoping for 4 biologicals.
Yeah, that's what I figured. So if we do that, then I get to have 4 samples some other day.
Yeah, sure.
Right.
Right.
Maybe to the left. Oh, no, that's a little big. Maybe to the left a little.
There is an
One. Oh, is that? What is that up there?
yeah, it's a fish.
Yeah, that's good.
Probably.
You want to go check them out?
Yeah, I think the rock will stay there for a minute.
We do have a ship move underway. I think we could probably come back to these if we.
There's more around here.
Okay.
We're just looking for a representative sample of one of these coral-laden rocks.
Understood.
Yeah, let's get a look at this. Could be the Cryanema, but I'm not really sure.
Okay, come on in,
Doesn't
video.
quite look right.
Hold
Yeah,
there.
it is. It's the Cryanema Christ series. We got
Hold
some
there.
And the
good
blue
video.
stuff again.
The blue stuff again.
Not sure what his plan is here.
It might involve escape.
That's just a guess. There's a lot of that blue stuff around here, yeah.
Okay, come on in, video, if you got any more.
That's Max.
Copy.
It's a slightly different perspective, Bruce, than the one we got earlier, like this one. Maybe this would help with nailing down whether this could be
Once you come up a little
Cryanema
bit,
Christ
Copilot.
series or some other type
Copy
of
that.
Cryanema. Yeah, there you are. He's saying it's Cryanema Christ series, named because of those green-yellow patches on his back, presumably.
Yeah, and this nicely shows the fins that distinguish this from the green eyes. This one's got 2 separate dorsal fins, the fins on the back. The first short one is composed of held up by spines, while the second one has soft rays. The green eyes have a single short dorsal fin in the middle of the back, and another difference is those fins under the throat Oh, and there's the 12 big fins. They're up forward again under the throat. In the green ice, those fins are actually back, about halfway back on the body, on the abdomen. So there's some differences
an hour and a half left.
in morphology. They're similar in their habits and their appearance from a distance, but they're totally unrelated fish. This is the spiny rayed fish, and the green eyes are soft rayed fish.
Okay.
Ken Sulek had commented that the
ship has
reflective
moved 18 meters.
from
You got
the
about
lining
12
on
meters
the
left
eyes
of
of
movement.
these, and particularly the ones that give green eyes their common name,
Copy that, Nav.
is an adaptation that filters out some of the wavelengths of light and lets them see
Pilot, you're
the
about
blue light
8-0
of
in my
a little
cam.
better in their hunt for prey.
Thank you, Bruce.
Are we needing to move on?
The ship is close to a stop. We can maybe survey this and adjust if you really find one you're in love with here.
I mean, that doesn't look that big. That one over to the left, that kind of, left of the lasers. It's a little bit further over, like right there. And up.
A little up, yeah.
Yeah, that one up a little bit, right above the lasers now. I mean, up and slightly to the right.
That's 15 centimeters. That's not actually that big. That's right.
Yeah, the one to the right of the lasers now is one we're looking at, the darker colored one. Think that's a possibility, Chris? Or
I think so. Is the ship stopped?
Ready to drive nav? Can we hold current position?
What do you think, co-pilot? What are you looking at? 85.
Yeah.
You now have two biologists that want to collect a rock with a C-pin on it.
Okay.
Yeah. Do you want me to spin now?
Yeah. What is that?
That's fine.
Copy that.
Okay, watch these. Which of these?
That one or the one behind it, whatever one is loose.
Yeah. That one's good.
Yeah, because it has biology. Maybe I should pick the one behind it.
Double bragging.
No.
Biology isn't going to be.
Both of them could be stuck.
Yeah.
This is weathered limestone, so this could just be a piece sticking out of a wall.
Copy.
C-pens look a lot like Calibolemnon. It's
easy for you to say.
Yeah, I'm trying to see if it's in the same, which family. I forget which family is which on the C-pens.
You want to spin me up, co-pilot?
Yeah, if you're ready. Hydraulics up.
Copy. Let's keep your view up.
Well, Calibolemnon is a scleroptilid. I don't know. Maybe not. Maybe Anthoptylan.
Oh, you meant just up on the screen, not rotate camera up, right?
Yeah.
Okay.
Just up on the screen.
Yeah.
Keep pushing a little bit, video. And just hold there.
Sure thing.
There's
So
a crab on there too.
the one lasers is on, or the one behind it? Which one do we want to try
Whichever
first?
one is a little loose.
Okay.
I think they're both potentially stuck.
Okay. Well, we can certainly find out.
The fact that they have that black dusting on them suggests they may have been in place for a long time.
Can you come live, video? There's a
The
crab
crab's
on that
getting wise.
That's pretty stuck. That is also stuck.
All right. 0 for 2.
The crab is bailing. Is there any other ones we want to try here?
You can try the one with the starfish on it.
Yeah,
To
all
the
the way
left.
over to the left.
Where?
Just left of your lasers.
Yeah, little white starfish on it.
I think all these are going to be similar. They're all going to be stuck.
Copy.
These are pieces of carbonate. The stuff around it has been weathered out. Unless
Yeah,
it happens to pop out of the bottom.
Yeah, that's pretty stuck.
Yeah. Let's look for a lighter colored one.
Okay.
Better chance of it coming out.
Okay.
Copy that.
Yeah, a nice fossil in it too.
Well, all these probably will.
Yeah.
That fish is still sitting in the same spot.
Yeah.
I think he's sunbathing.
No, he just doesn't think we can see it.
Mm.
And if we were under ambient conditions here, we wouldn't be able to see it because we wouldn't have any of this light.
And then, see what we got. Let's switch.
And he doesn't know any better.
350.
I tried.
Oh,
shark.
Yeah.
There's a shark.
Do you wanna?
Where is it?
There, yeah, right above. Oh, yeah.
Oh, this is a 6-gill. Hexanchus griseus. I think 1, 2, 3, 4, 5, 6. Got 6 gills. Sorry, Bruce. I should have let you comment on that. Had my glasses off. I didn't know how long he would be here.
Got a fill frame video.
All right, yeah. I got into this business from studying fish larvae, and sharks are live bearers that have young that are basically like miniature versions of the adults. They don't have larvae, so generally I don't work with sharks. So go for it.
Yeah, so this has got 6 gills. You can count them really easily just by taking a look at it. So it's a 6-gill shark.
Can we turn off the
Dorsal
lasers?
fin is well back on the body. These are characteristics of it. The very heterocercal tail, the uneven upper lobe and bottom lobe of the tail fin there. These are other clues. And also just the head shape and the size of the shark. Are the lasers on? I just wonder how big this thing is.
We got them. Yeah.
Oh, just for getting the, and it's a male too. So the 1 6-gill we're all familiar with is Hexanchus griseus. I don't know how many 6-gill sharks are out there that look like this, but do you have any idea?
There is another species, Hexanchus nakamura'i. I'll have to check and see what the differences are. I think it's something about the snout length in relationship to the head, and then some things about tooth morphology. Odds are that there's Hexanchus griseus. So,
you have any idea how big this is, Pilot?
We got lasers on him earlier.
And
if you want to throw him back on, you can.
You can see him in Sirius view. He's almost as wide as Sirius. Or as D2. Maybe even a little wider.
And the D2 is how wide? I'm not sure.
About a meter and a half?
2 meters?
Lasers
yeah, about 8 feet.
About 8 feet?
Yeah.
Okay, so he's probably at least 8 feet long.
Yeah.
We might have to let him go from here.
Okay.
There they are.
There they are.
Can I take one last zoom?
Nice
Okay.
place. Yeah, thank you.
Go ahead.
Go ahead, Bridge.
The one character that tells those two species apart is the length of the dorsal fin, that fin on the back, versus the length of the gap between the dorsal fin and the tail fin. If it's about equal,
yeah, confirm center of rotation is A-frame.
If the gap is noticeably
Thanks,
longer
Bridge. Yeah, go ahead.
than the length of the dorsal fin, it's the other species, nakamura'i. So
Ship's going to change heading, pilots.
Take a look at those relative
Come up to starboard.
Looks to me like it's the same, Bruce, in which case that would make it griseus, if I understood you correctly, Curve. Is that right?
That's right, and that's what it looks like to me, too.
Okay, let's let him go video.
Yep.
Well, that was a nice
Let him leave the frame.
shark bombing.
And then I'll come wide.
Copy.
Nice.
Why doesn't he just eat some big fish for us right in front of the camera?
These little guys are pretty scared, I bet.
There's plenty of fish for him to eat.
Yeah, that's true. It's kind of slow. I don't know what happens when he's actually... He may be a scavenger more than a predator.
Yes, I rotated
really
almost
sure.
90 degrees.
Copy.
So I can just follow you back in.
I'm just astonished by the sheer number of these rock pens at this site. There are thousands and thousands of these things here.
Yeah, you can just follow me back.
Now, those sharks are both predators and
Probably not going to be able to move it
so
shift changes
that
heading.
would
So
eat these fish and probably would like to have a tasty alphonsin every now and again. But why that one wasn't eating anything? That's a basic law of ecology.
Go ahead,
You have
Rich.
a complete knowledge of the biology of a species and the exact specification of environmental conditions. Any individual animal is just going to do whatever it pleases.
Thank
All right,
you.
that's better.
So there's a bunch of gold corals on the top of that little pinnacle. Wow.
Copy that.
Think they spotted another fish maybe or not? Fantastic terrain here.
Lose the skids video as I come down. Thank you.
Gladly.
So a bunch of gold corals in the background at this place, right here on this drop-off.
Okay, Sean. Ready? We're going to do a quick pilot change here. Watch, please.
Okay. And while they're doing that, I'll just give you an update. Those of you who are not on the chat room, we're at 447 meters. And our temperature is 10.3 degrees centigrade. And our dissolved oxygen is just over 5 milligrams per liter.
We've come up a lot in temperature.
Yeah, we have. Well, dissolved oxygen, too. We started out at 2, as you
That's
remember.
right.
Somewhere
That's
around
right. Yeah,
600. Now we're up to 5.
Hey, pilot,
kind of in a depth range where you
direction
get
think
very
next?
significant
The ship is still
changes
changing
in
heading.
temperature and
So,
dissolved oxygen before it
We'll let 'em do that,
stabilizes
but,
out maybe around 800 to
just ride the
1,000
ridge,
meters.
so.
And then oxygen starts coming back up again.
How much more of a
Temperature
heading change
just kind of
do they have?
flattens out a bit.
I think they need like at least 10 more degrees.
Okay.
Yeah, Nav, I would just
work around a little.
Keep towards waypoint 3. I think it's fine.
What you're looking at all over the bottom, in case you're seeing this at the Maui
Copy
Ocean
that,
Center or the Waikiki Aquarium, you're looking at
10-meter contours.
a huge field of rock pens.
With all these,
They're sea pens, but they've
all these weird
adapted
formations.
to living on rocks.
Kind of hard to.
Interspersed between them are a few sponges, some
Yeah, I think that's
Gorgonian
fine.
corals, including
Isn't that like 350
precious
anyway?
gold coral, the ones that are on that little pinnacle off to the right.
Yeah, okay.
It's perfect.
This is very old carbonate.
Right, it's sitting on top of a very old carbonate bottom made of pieces of shells and pieces of coral that all were debris coming off of an old reef that was building here essentially, that the current reef is basically the end product of. And that material slides downhill, makes layers that get turned into rock, and that roundish shapes that we're seeing are probably the result of weathering in the open air. So this was at some point at sea level.
So who you got piloting now? I think it's Sean's head. Is that right? It is.
Yep,
okay.
Sean. Me and Chris
We're
just
getting
did
better
a
at
quick
that.
exchange.
Getting better at recognizing the backs of heads.
So we've got a nice little stand of gold coral up ahead. So it makes for a real pretty scene. This is pretty good colony there on the left.
Do you want to zoom on that, Watchley?
Or even just get a little bit closer.
Copy
We've
that.
zoomed a lot on these golds and even collected them, but maybe we can get a little closer just to image the group as a whole.
Yeah, absolutely.
This is a massive gold colony. You can see how it may even be merged. Could be even 3 colonies merged together. And some people are astonished about the age of these things, but right now a colony like that, if I had to speculate, is probably at least 2,000 years old or older. And then there's one behind it that looks pretty darn big as well.
That's a long time ago.
Yeah, some beautiful big gold coral trees, and that's their host These are a parazoanthid. They're a parasitic coral,
Yeah, ship
and
has
they
about 5 more degrees.
overgrow other species of coral, and in particular, we
Go ahead,
think
Rich.
these have been overgrowing these white bamboo corals behind it. And the ones behind it just haven't been parasitized yet. These gold corals are extremely slow growers, and
Perfect.
they're
Thank you, Bridge.
extremely long-lived.
Pod's ready for a move. Due north, 355, basically.
Yeah.
Bridge,
really
are we now?
Fascinating life history. And
All right, we're ready for a ship move. 30 meters, bearing 355 degrees, decimal 2.
On these groups. This group, and it's got all types of stories about them.
Good copy.
Kind of a really nice scenic as we're cruising in this little depression here.
Move initiated, good copy. Thank you, Bridge.
Yeah, it's quite busy with life here.
Yeah.
Let's see, we are at about 447 meters.
Yeah, I'm not moving. Try not to move forward anymore. I'm just kind of laterally.
Yeah, that's perfect.
Nice drop off.
Yeah, pretty scene. Look at this.
Yeah, that last move is 355. Copy that now. 355.
I can turn there.
The ship's starting to move, so.
And this is
Copy that.
Interesting look.
355.
Hard to tell the sand from the weathered limestone here almost.
Pretty
Yeah,
much
that's
the same
okay.
color.
Yeah.
This doesn't have as much crusting on it.
Yeah.
I'm
Kind
actually
of
going
a
to come
nice,
up a little bit
look back
so I can see a
over
little bit
this,
more of your tether.
pretty,
Bridge, are we now?
dramatic
As I come
terrain.
this way.
Just confirm bailout 090. Thank you, Bridge.
We are on the east side of Wake Island, and we're in the Wake unit of the Pacific Remote Islands Marine National Monument. This is one of the shallower dives we're doing on this cruise. This particular ROV has sort of a minimum depth rating of around 250 meters, and we're pretty close to that already. We're at 450, and it has a maximum depth range of 6,000 meters. So we're usually
Quite a ways.
Yeah, we're usually working in deeper water, but we needed to do a dive in these depths to look for these precious corals, like these gold corals you're seeing in front of us right now, these yellow ones.
Yeah, that'll be interesting to see where that ship move takes us. Not quite sure what's on the end of this.
Yeah.
An update on yesterday.
But I think that's the right way.
So we're about 200 meters from our end waypoint right now, and we've still got about
an hour and a few minutes.
Yeah, a little hour and 15 minutes maybe. There's a little holarity. I wonder if we could zoom in on that little fish again. We haven't got a
Video, you want to come in?
gotten a kind of a faraway image, but not a close-up of them.
Gonna take a little more zoom.
Yep, go for it.
That's max.
Looks like the ship has about 5 meters
Let's
left.
turn off the lasers.
It looks a little different from the ones in Hawaii because we see those as red or pink in color. We also saw reddish ones over in the Marianas. The species in Hawaii was thought to be an endemic species found only in the Hawaiian Islands, but these haven't been reported from other islands in the Pacific. So our records from the Okeanos Explorer in the Marianas and here at Wake Atoll are totally new. We have seen these in the Line
Looking
Islands.
right down at you, pilot. You look okay?
Well, which leaves us with the question, are they all the same species? Is the Hawaiian species much more widespread than previously thought? Or are there a couple or more related species that look similar when we see them in these videos? And without specimens, we won't know the answers to those questions.
Well, one thing is that we definitely know that this genus is here in Wake now, and that knowledge did not exist before today.
All right, video, want to come on out? So
come here.
That's a start.
Thank you.
Full wide.
Looking back at you. Just dead ahead is where you want to go.
Video, you can lose these skids if you'd like.
And that is quite the drop-off.
Indeed it is.
Uh-oh.
What?
We forgot something down the hill.
All right, let's push out a little, pilot.
Copy that.
It's one of those things you do when you're doing field work as a
pure
geologist. Oops, I forgot my hammer at the bottom of the hill. Gotta go back and get it.
Again?
So I think this might be the last thing you can see for a little while. So if you want to pirouette or something, that's totally fine.
Before, I wonder if his arms are a little extended here. I wonder if you could take a snap zoom on this brown thing on the coral. The currents have settled down a little
center,
bit.
watch lead?
Dead center?
Yeah, dead center.
All right, video, come on in quickly.
It doesn't look like he's the fuzzball that we've been looking at.
Yeah, he is. He's got some of his arms out now, so we can actually get a better look at him, because I don't think the current is quite as intense. Definitely has prehensile cirri. Those are the way it's grasped onto the branch.
Pilots.
And prehensile in that they can wrap it around the branch several times.
It's this little white spaghettis that you're seeing around the bamboo branch.
Yeah. So I'm trying to think. So what is the Latin term for bad hair day?
Got a bit more zoom.
Well, if that would be
I don't think anybody's going to come back with an answer to that one.
No, but that's actually the way I look when I wake up in the morning. Pretty much that same thing.
Right.
There's some clumps of hair just sticking straight up out of nowhere, and other parts are just sort of curled off to the side. So I relate. I really do. It's crinoid. It's a feather star. Definitely has an awful lot of arms, clearly. I don't know if this is more than one individual, but I can only see one set of cirri on the bottom.
I think it's two.
It may be two. Yeah, it could be, because that's a lot of arms. Wonder if this could be something like in the family Chirida-metra or something.
Here's one. There's the other one.
You see another.
This seems to center right on this branch.
Oh, you're
here.
right. I can see the cirri right here. Yeah, so there are two there, one on top of the other. At least two.
Come out for a slightly wider view.
Thank you, pilot. That's great.
Yeah, video whenever you're ready.
Clear.
All right. Let's go
Zooming to lose the skids.
Copy.
Looks like I've settled out a little bit. So once you push out past that kind of rock there, maybe we'll get another ship move in.
Copy that.
Or we can go ahead and do that in anticipation.
Yeah.
355.
355. 5.
Bridge, ROV nav. Ready for another move. Same as last. 30 meters, bearing 355 degrees, decimal 2 knots. Good copy.
We're finding a lot of these corals kind of on these little promontories that are sticking out from the main wall.
Particularly the ones that have the little dark coating. The ones that don't seem to have a lot. They have some of the rock bands, but not much else.
Come down with me slowly then?
Well,
just get to the bottom? Or?
I can't come down until I'm clear of that.
Good little bit of manganese coating may stabilize.
I think maybe if you pushed out.
Frankly, the carbon.
I'm looking right down at you. So if you push,
I still think that it
might be able to acquire just
show that it hasn't
really
had a lot of sand flowing over the top of it in density currents, and so the ones that are clean
I came down a little bit.
are the ones that have had sand moving
You're not
by
even close to
recently.
that yet.
So I just don't think it's quite as stable of an environment.
Gotcha. Yeah, another altitude, 13 meters. So.
Yeah.
A lot of fish down here.
Yeah, there's a bunch of barracks down there. Same ones we've been seeing all day. Trying to see if there's any different fish.
Yeah, watch it. So we're just going to head down here and acquire bottom again.
Okay.
And go from there.
I would head out more than down. You can't come down quite yet. Because
you're still on top?
Yeah, your tether will scrape over that feature that we just went on.
This would be a good place to look for a loose rock. Some of those rocks look loose and they're yellow, so they're not all encrusted potentially.
Yeah, this would be a good spot. Actually,
question is how big.
Once I get out there.
Once we get down there.
Yeah, and there's a little white sea star on the one boulder.
Yeah, I see him. Yeah.
Okay, I think you can start to come down.
There.
Yeah.
Feel good?
Yeah, feel better.
Copy that.
So would you want a light-colored rock or a darker one?
I think we're going to have a better chance if it's a light-colored rock that's sitting on top. I suspect the black ones are all basically have been in place for a long time. Although this hillside may be steep enough.
Another one of those tinsel fish, and that sea star is the tree master. Now that I get a better look at it, these sort of disc-shaped sea stars that we imaged earlier on.
Like the pin cushion?
Yeah, like the pin cushion. What about those rocks over to the right? You think any of those might be?
Let's wait and see when they're happy and down as far as they want to be. But yeah, I think most of the stuff that's sitting at the bottom here is all these are loose probably.
Could we get the lasers on, Sean?
Yeah. Lasers coming on. Got a port rail cam rider there.
That's a big one.
Wow, that's really big.
I think we're looking at this size, or this one.
Yeah, maybe. They look, well, yeah, the ones below the laser look like they could be possibles.
Yeah, watch it. Do you see any?
Yeah, if you go down a little bit,
look
straight down. There was a couple of very light-colored rocks that looked like about the right size. Keep going down a little further.
Let's
and
tilt
it's right
down.
on the sand there. Those two kind of overlapping each other. Yeah, one of those to the left of the lasers, or even the right. What you want to?
I don't.
Huge. I think that's
that might be too big.
Yeah. What about one of those?
Yeah, that would be good. I think.
Those two look loose. Would one of those work?
Yeah.
Yeah.
Copy that.
I mean, we're doing this for posterity, right?
That's right.
Neither of us are gonna do much else than look at the rock and go, "Ooh, pretty fossils."
And Tina is watching right now, and it would be kind of good to do the mushroom corals after we grab your rock, because there's about 3 of them all lined up in a row over to the left.
I think maybe
before we leave here,
pushing me out another 15 meters or 10 meters would be good. I'm looking back at him and pushing me out to the middle of the kind of hole.
Okay, you got 5 meters left in this move.
Oh, okay.
So
we might be okay then.
Yeah, if you
Yeah.
Yeah.
You should be able to come down, I think, a little bit.
No. That feature is right under me.
Oh,
He just went over.
Okay, I thought we were past it.
No. So, but I think we're stable here. So, if you want to get set for a collection, I'll spin you up.
Copy that.
Okay.
You can sort of see the fossil coral structure in here. Looks like a relatively large coral head.
I'm going to turn to face you.
The rock right behind the ones we're looking at.
There's a bit of current down here. You can just see by the way the sea fans are. Maybe it's the
I don't
thrusters.
think that's me either.
Oh, you don't think it's you?
I don't have a lot of thruster on right now. It's possible, but
It's been windy all day.
I don't think it's all me.
Underwater windy, I mean.
So trade currents? No, I guess you wouldn't. call them that way,
They're
would you?
coming from the right, from starboard side, which is from the northwest.
No.
Here.
Well,
Okay, I came around to look at you.
That's the
Mainly
northwest.
just to
pretty
see the
much.
tether, and
I
I
guess
feel
so,
a lot
yeah.
better.
Feel good?
Have we
Yeah.
gone through a whole full tidal cycle at this point? Weren't they coming from the south earlier?
let's see what my notes have here. The current along ridge from west is what I had. So that makes
Northwest west.
must have been northwest.
Ship's in position.
So Washley, do you have a preference on right or left right here?
The harder one.
Harder one? Copy that.
No, I don't have a preference.
Copy that. I'll give them a squeeze and see what we got.
No, you don't need to squeeze it. They're fine.
Oh, they're fine? All right.
Yeah.
Video, do you want a quick zoom? Probably just do the one dead center.
Sure thing.
Before I collect it. Copy that.
Maybe the bottom one is nicer looking. I don't know.
Okay.
Bottom one,
the bigger one.
Yeah. More
Yeah.
chance of more different pieces of things in there.
Copy that.
Sure.
All right, Copilot, you want to bring port wing, port wing inboard?
Sure.
Shouldn't hit. I can go for the lid first.
Is there is something in the starboard one?
Oh, there's something in there. Oh.
I don't know if there's something in the starboard one.
I thought there wasn't. Nav, do we have a rock in port? Or I'm sorry, did I say starboard or? Do we have rocks?
There is no rocks in any of the rock boxes. They're all in the bio boxes.
We did have one rock with a biological.
You mean your blue rock?
That's not my blue rock. That's your blue stuff. There happens to be a rock attached to it.
Attached to it. You got the box? Okay.
Yeah.
It's a commensal rock.
Well, it's actually going to wind up as a geo specimen, so it will be a second geo. Because that's really the only way we can deal with it in the database.
It would go above the skid, right?
Treat it as a separate rock.
I still think we should just have it be a commensal rock. They're having us fill out the habitat for the rocks. We might as well have a commensal rock at that point.
Yeah, there you go.
Ooh.
There's the clock.
He's going to actually put it in the port box?
Well, it's not huge. I think
No, but we're not planning to pick up a huge one.
Marshall, you said
the one on the right, Sean.
If you can get the bottom one.
Yeah, but if that's too difficult, the one on the left is okay.
To be sure.
They're sitting on the sand. I think it'll be fine.
Yeah. Yep.
Yep. They're loose. Little epigonid, deepwater cardinal fish flitting around on the right. Those are really common in the steppe range. So it's just about everywhere. Marianas, Hawaii. There it is.
Yeah, they're actually found worldwide. The epigonus are found worldwide in all of the oceans.
Yeah, thanks, Bruce. Bruce is over at the Exploration Command Center at the Inouye Regional Center on Ford Island. He's with the NOAA Fisheries Science Center, as I mentioned before.
All right, Vida, you want to come on in?
Yeah, we've had a few visitors from the public come through here today. We had our last group just about 5 minutes ago, and they've all been very impressed by what they've seen on this dive. They think it's beautiful down there.
Oh, thank you, Bruce. That's good to hear.
Yeah, keep going.
It's like it's got little forams on it and sort of flock stuff. That's pretty typical. It's got possibly some little barnacles, and it's got a little coral, maybe a cup coral on the bottom side too. Yeah, it's going to fit in that box very nicely. You ready to punch collect on it?
I am. Once he drops it. Or carefully puts this down.
It's in.
It's in the box.
So,
all right. Do you want to try coming out? See if word shots?
2-3
Sure.
Geo.
Mm-hmm.
Wonderful.
Auto stow.
No, and then you wanted to look at those pink enthymanthus,
packages,
or whatever they're called.
Yeah, Anthomastus.
Amastus.
Yeah,
sorry.
or Pseudoanthomastus, depending
Package
on
secure. Confirm.
Species they are. So, pilot, before we leave here, if you could just swing the camera to the left and just get a quick zoom on these red mushroom corals on the left.
Copy that. Arm is off.
Copy. Spinning you down.
The pink guys with their hands up.
Yep.
They're all cheering. Yay! They're all cheering for our pilot. You did it! Yay!
Thank you. So, Washington, I'm sorry. What are we zooming in on?
If you could pan to the left just a little bit, and you'll see sort of a row about 3 of these red mushroom corals. There's one of them showing right now.
They're more pink than red, I'd say.
They're pink, I guess.
Top left of your screen.
Copy that. Okay. Let's see.
Yeah, we're stationary, so you got some time.
There they are. We could get a zoom on that.
There's a lot of current right here, so just give me 1 second.
And now you see why I said they were all cheering for you.
Yeah, and the reason Tina was wondering, she just joined very actively now, and she's asking whether we image one of these, so I wanted to give her a chance to look at it while she's watching.
I think that means you'd like a zoom in.
Yeah.
Sorry, Washley, it's give me 1 more second. Slipped off my toe hold.
Yeah, I don't think that's as a result of your thruster. Look at the way those mushroom corals are blowing around in that current.
Yeah, I'm fighting pretty good.
Yeah, the current definitely picked up, so we've seen a whole cycle here, I think.
Yeah, I wasn't doing that when I was flying.
Even when just when I got down here.
Yeah.
given the change in temperature and salinity and
let me know if you want me to take it.
oxygen, we may also be in a completely different stratified layer.
Nice.
I mean, we have twice as much oxygen here as we did when we started the dive.
Video, you want to come
Right, and we're not that much deeper.
No. I think we came up from, what was it, 600 and some meters? We came up 200 meters maybe.
You know, these are different than what we were looking at earlier. I think there's two kinds of these mushroom corals that are down here. I'm pretty sure this is different.
I'm going to go for a bit tighter.
Yep.
Yeah, the ones facing the currents are almost bending backwards completely.
Video.
Yep.
Yeah,
still pushed off again.
Yeah, and so Sako also agrees it looks different than the Anthomastus that we imaged before.
Washes, do you want another view on that? I slipped off. I can go back in if you want.
Sort of waiting to see if Tina's going to weigh in on this.
She may have fallen back asleep. Oh no, there she is. This one is Anthomastus.
This is Anthomastus, yeah, okay.
She just said she was sleeping at 6 a.m. there.
Oh, really?
She is.
Okay.
So I guess she just woke up from take a little nap.
Yeah. No, I think we're good, pilot.
Okay.
Thanks very much. Yeah.
Copy that. Leave these be then.
I'm wondering if those other ones that we saw were Pseudanthomastus. I sometimes get them mixed up. Yeah, I actually got Pseudanthomastus
So turned way around
down as earlier on in my notes.
But if you want to come back to 350,
it was Tina last year instructed me what the difference, what type of things to look for that are.
Yeah.
That looks like a tinsel fish again.
Yeah,
Picks
there's
up in
a
that
tinsel
hole,
fish,
maybe.
right? Could have seen those all day long. I've seen probably more of these fish than I've seen on anywhere else.
I'm going to keep turning.
Still more? Copy that.
So we're a little bit up on the flats here. We've come a little ways off of the really sharp ridge. And
okay.
I don't know,
I'm just about there.
I don't know if it'd be possible to get a little bit closer to that ridge.
Yeah, Washington, we can adjust the next ship move. We weren't really sure what's out here before, but we can adjust. It will go off course from waypoint 3 a little bit, though. Is that fine? Would you rather stay along the ridge?
Yeah, no, that would. Well, the ridge would be a little bit better, I think. We just follow the ridge for as long as we can.
Okay.
Because we are seeing more corals and things.
Yeah. So, Copilot, how do you feel if we?
Yeah, we can do
turn
that.
Back a little and then put a ship move towards the ridge.
I would just go northeast.
Northeast.
Yeah.
Copy that.
What do you think?
They can require this.
Yeah.
Situation.
Yeah, reacquire the.
So now we're really at the base of that big carbonate cliff, and
that way.
we're seeing how it transforms into this
Maybe
much
take a
less
couple moves to get over to that
landscape
ridge,
of just sand with ripples and a few blocks here and there.
Probably.
Nah.
It's a very different area.
Definitely the wrong way.
That's a
We're
local.
on a little sort of pinnacle feature.
You're looking at
a really sharp drop off is still to our starboard.
I think it's 050, Sean.
We're kind of peeking over the
so
edge
we'll
here.
kind of go downhill a little bit.
It drops off on this side too.
Situation. Well, I think there's more than one step in this topography. We're not seeing all of that in the mapping data.
050, okay, Sean?
And you'll notice that the C-pens have really dropped off here.
Yeah, we can try that.
Yeah.
And if you see a ridge on the way, we can ice that. But
yeah.
Yeah, I think
maybe
there's
this is just too much sand for them.
There's a lot of this local up and down, but the actual real sharp ridge is to our northeast or east.
Okay. Bridge over now. Ready for a ship move. 30 meters, bearing 050, decimal 2 knots.
Which is right there.
Okay. All right. Thank you.
Might be quite a ways.
Not that far. Just looking at the contours here. They're about 150 meters away from waypoint 3, so 30 or 40 meters to the ridge, probably.
Okay, I'm just about there, Sean.
Copy that.
That way it looks pretty clear on sonar, so I'm going to come down a little bit, give you some leash.
Yeah,
it's good copy, Bridge.
Nice and flat here.
Yeah, Tina's noting there was a few C-pins where there's a few rocks laying around.
But it's gone.
Like right here in the
Sorry,
bottom.
just you said.
So these truly are rock pens. They are preferring to
transparent
attach themselves to the rocks, not to the sand.
Okay.
Most sea pens, the reason I'm remarking on that is most sea pens actually prefer to anchor themselves in sediment. So this is a very different type of a penatulation.
Yes, I'm actually pushing back this entire time as I go forward.
Oh yeah, it's coming at you?
Yeah, right from rear.
East to west?
Pretty much, yeah. Yeah.
Oh no, west to east.
Other way around. Yeah. Oh, sorry, yeah, west to east. Towards the east.
Pushing back, I'll take a
pretty
second
close
look.
to the drop off. There's no sign of it yet, but it's there. It just goes straight out.
Yeah, you can kind of tell
out there.
everything's a lot more.
Maybe we're starting to see a trace of something. No. My imagination is a little murky. The water here.
I thought the same thing.
It might, with the high currents, it might just be kicking up a lot of sediment.
I see.
Yeah, I couldn't find the tether in the Sirius Titan, so it is pretty murky. Yeah.
Oh, okay.
Well, that one is sitting right in the middle of the sand. There's no rocks around him.
Yeah,
gonna hold 050. Let me know if you want to.
There's a little starfish next to
turn
him.
Back over.
Give me a little tug there.
Yeah.
What is this, Chris?
What's that? I don't know. Could we look at, I think these are urchins. Could we take a look at this green cluster of things on the right-hand side?
Green cluster, yes.
Might have to wait a little bit.
Good spot. I had actually turned away from the screen.
I'll come down a little bit.
Yeah, let me know if I...
You're tugging me now.
Oh, maybe these are corals, or it could be some more of these Aguchi Somnia.
Video, come on in. Green.
Yeah, I think that's what they are.
We've got a little bit more leash.
Yep, just your clusters of
they
these
are
Aguchi
stuck
Somnia.
to the littlest rocks, aren't they?
Yeah.
Holding partial.
We got some good images of these before. Thank you, pilot.
Copy that.
These are
video whenever you're ready. It's not much of an interest.
Almost slightly different species, almost. Maybe just hang here just for another second.
Oh, hang here?
Yeah. Well, yeah. I mean, just keep with the zoom just for a moment. There's a lot more in each of these clusters.
These guys are some sclerotinia.
Yeah, that's Aguchi Somnia's in that group.
Oh, okay.
Sclerotinia. Just could be a slightly different species.
There's a tiny little thing in there too.
Yeah, a little plexaurid there. Yeah, that's. Yeah, now he's got a little bit of light on it. Now this is. I think these are different.
A bit more zoom, I think. Yep.
Oh, come on, Chris, commit.
Well, the other one, the other one had a little more of a funnel shape to these.
I thought they were
these
darker
more,
yellow too.
Yeah, they were darker yellow, and these are almost a little more tubular. If you look at this one. Yeah, I think we've got another animal here, but I think we're good whenever video is too. Thank you.
We stop?
Copy that.
No, it's just moving.
Video whenever you're ready.
A couple more seconds.
4 meters. It's got 6 meters to go.
Okay. Once he's done, you keep it going. Sonar looks good. This looks pretty flat over to the ridge, I think.
Copy. Yeah, it should be.
We can go more east if you want to just acquire ridge and adjust. Whatever you think.
Thank you.
Yeah, I think keep going 050.
Okay, yeah, that's what I was thinking, and then kind of that way we're going towards waypoint 3 too. Yeah.
Exactly.
Tina says Steve will not tell the species without specimens in hand.
Yeah, I came over a little.
Yeah, when I come back.
Yeah, copy that now.
I'll add 30 meters to that move.
You see a whole pit behind us thing from the current digging.
Unusual
He
fish.
hates guessing, Tina, but if
add
you wait him out,
30
he'll
meters to
eventually
that
look
move
at your
at
photos.
050 degrees?
Where do you see it?
Oh, never
she's
mind.
talking
I think it's
about
just
Steve Cairns
I think
at
it's
the
just a C-pen.
Smithsonian.
Ignore me.
Copy that.
He doesn't like guessing, but he's willing to do it, just to try to be as helpful as he can be, because he's such a great guy.
Okay, let's
quite
keep coming.
an expert.
I came over.
Yeah, coming.
On scleractinians and primnoid corals and all types of things.
Okay, I'm back to ship's heading.
Copy that.
Ship's bearing.
Yeah, we're definitely getting close to the, there could be the edge right there.
It does seem to drop off a little bit there, doesn't it?
A sandy drop.
Should have some leash if you want to go check it out. I'm looking at you, 45.
Yeah, copy that.
Definitely the currents are running along this ridge. You can see by the bed forms or the ripples right now and the orientation of the ROV. The prevailing current is just right along this face.
Well, if you look at the map we have up, basically we're coming off sort of a knob, and there's another knob north of us, and this is sort of a smooth slope all the way up. This is basically where the sand is being transported, and it shoots off the edge here probably.
Dead ahead.
Jumps the cliff.
A really nice sort of possible rock formation.
That's
almost dead ahead there.
That's why
we're probably going to peer over the end and go, ooh, that's deep.
You guys agree?
Agreed, yeah.
So once Sirius gets over top of it, then I'll just stop the ship probably.
Yeah, and then go.
Yeah.
It might put us right there, I mean.
Seems to be have picked up considerably now that we're on harder terrain. It's closer to the
closer to the edge.
Getting a little ways out there.
They're trying to climb out of there.
Come down a little bit for you, maybe.
They're all bent over in the current.
So is ship moving?
Yeah.
Oh, it is. Okay. And that, what's the direction?
050.
Nice.
Yeah, Tina noticed the asteroid, and Tina, those are Tree Master, Mirabilis. The asteroids, we imaged those a bit earlier in the dive. Sort of rounder, disc-shaped orange and yellow ones.
To east.
I can kind of start to cheat my heading that way.
Let's get a look.
Tether looks good.
Yeah, I didn't see the fish either, Bruce. I was a little distracted too.
Another sea star going out of the picture.
Yeah, that's another one
in the
of
water
these
column.
Oh, what is this jelly?
Is it a jelly up there?
Yeah.
Or is that a Anistenophores again?
Sure. Yeah, that's a good point.
It's going up.
He's coming at you.
Current's going too fast for that guy.
It's okay.
Cannot see him.
Yeah, that looks pretty steep. Very rounded limestones here at the top.
And they're not manganese coated.
Nope.
And it seemed like those corals were preferred the slightly manganese coated, so I wonder if it is stabilizing.
I think this is being overrun by sand. Every time there's a little bit too much sand, it just flushes down.
I'm going to zoom out the skids again.
Copy that.
What is this?
It's just a rock sticking
sure?
out. Yeah. It's kind of... It's just one of these other lighter
two sea
color balls.
stars.
Yeah, I suppose we could take a look at the stars. We looked at these earlier, but we can sort of confirm that they're the same ones.
Copy that.
Maybe the one on the left first, because...
Video quick zoom on sea star.
That's sizable.
Yeah.
That's 10 centimeters.
Yeah, that is a good sized star.
Next time some sand comes by, he's going for a little ride down the hillside.
Holding
This
pressure.
spa is not cold in today, so
I hear you. Yeah.
It's probably celebrating Octopus Friday somewhere else.
Yeah, so the sea star could be something in the genus Apollinaster, but I'm not really sure. And there's another one here.
Those are the cookie cutters.
Cookie cutter stars, one type of cookie cutter star.
Video come out. Yeah.
Very rounded, very smooth carbonate.
Recenter. Alright, coming on the one on the right.
Roger.
Yeah, Tina's pointing out there's too much sand to have crust. I think it's actually the fact that there is so much sand flushing through that's making sure there's no crust precipitating at all.
Partial.
Ship's in position, pilots.
Back away. Ship's good. Alright, but if you want to try to get a quick zoom for
Roger.
more, I'll try to hold it.
What do you think, Nav? Just keep me here and then just face kind of up along the ridge? Like, don't move me west or east at all, but just along the ridge.
That'll be enough for an easy,
Yeah,
I
come
think.
on out.
Nice.
It's possible.
Yeah.
Not absolutely certain, but it's pretty close.
There's a whole bunch, I think.
Yeah.
I see another one now.
Oh, we got
There's one up
35
there.
minutes left on bottom.
Yeah,
I
so
think
this
they're kind of all
cookies-
over the way, over the edge here. So we are.
We're about ready for another move. Copy that. Pilots, you ready for a move north?
Affirmative.
I'll call it in.
Yeah, sounds good.
I'll come north. You're good out there.
Bridge RV now.
Sorry, I'll come back.
No, that's all right.
Alright, trip move 30 meters, due north, 000, at a speed of 0.2 knots.
Tina's
copy, Bridge,
asking
thank
whether
you.
you know if this particular sea star likes to be on carbonate.
I don't know the answer to that.
After this move, the ship's going to want to
it's a
change
shallower water
Change
sea
heading?
star, I
Yeah.
think.
Copy.
There's a couple
Let's come to port.
possibilities for it.
Wings shifted on a little bit.
As long as he rotates about the A-frame, is cool with
And
us.
I'm going to look in my guide.
Is this your handy guide made from previous Okeanos
Yeah, so
expeditions?
I've actually combined in my iPad the Okeanos benthic deepwater animal guide in addition to the Hurl animal guide, also deepwater, and have links to the Mbari animal guide too. So there's several of them, but I have to admit the Okeanos one from last year is really beautiful, and the imagery is so good. That's the thing. Mbari's got a nice guide too, as does the University of Hawaii, but these actually look like
These are the poofier ones.
yeah, I wonder if these could be Mediaster maybe. I'm not really certain. I might put that down as a possibility. And I was going to see how many of those cookie cutters are possible candidates.
Negative.
So it's not Spheria discus, I'm pretty sure. Can rule that out, but there's one other one. I think what it was. I do not think they're Astroceramus. Okay, here's one. Well, they could be Ceramaster Bauer's eye, actually. That's another possibility. But that's known deeper. And
I think you're tugging me a little bit.
The image I have of it was over 1,000 meters.
The current,
so,
yeah.
Yeah, I don't feel comfortable coming down anymore.
Yeah. There's so many of those guys.
This is completely coated in these rock pens. They really seem to like this particular type of area.
Yeah, it's a nice drop off.
Turning to keep you in view.
Yeah, Bruce, I agree. It looks
north.
like the water's kind of funneling down over the edge here. Sort of seems to also be what the sea pins or the rock pins are seeming to be subject to. You can sort of see it in the little white particles in the water column going in that direction as well.
So we got some pretty good images of this rock pen early on in the dive, but because it is so dominant, I'm wondering whether it'd be worth doing one more close-up of one of these pilot whenever there's one that's convenient.
Yeah.
Let's find one where we can get a pretty decent broadside or good image of it. And one of them that looks characteristic, like the one at the very bottom is kind of a weird shape.
Maybe 8 meters
yeah.
left.
Copy
that. Seems like wherever you put down, there'll be a couple in view, though.
Yeah.
Starting to look down at you, pilot.
Copy that. How much is the ship move left?
About 6 meters left.
You're okay.
All right, video, come on in.
Okay, so there's the pen, excuse me, there's the pen that's running right down the main stem, which gives the whole group their name, sea pens.
And look
that's
how full of pieces that thing is underneath, the rock is underneath.
Yeah. You mean
the carbonate?
Carbonate's got all types of stuff in it?
It's just completely, it's all pieces of stuff, of carbonate, of shells and corals. It's completely stuffed.
So I'm wondering if the sea pen is a Calibolemnon or a scleroptilid at least, rather than an anthoptylum. There's an awful lot of polyps on it. I'm gonna.
Washley, is that good, or do you want another?
I think that's good. Yeah, thank you.
For that, all right, co-pilot.
Yeah, let's get out ahead here.
Do a little hop. See what's ahead.
Come around to north, that's the way we're headed?
Yep.
Okay, I'm there.
Bridge RV nav. Looks like ship move is complete. If you wanted to change your heading, you can do so now.
There's the drop off. It's just dramatic the way it just
abruptly falls
off.
It's got to be
30. Can you confirm center of rotation?
A cliff, an old cliff that was
thanks, Bridge.
Eaten away at. Is this going to be one of those big coral heads again?
I don't know what it is. What is that?
It's a chunk.
co-pilots, it's going to take the bridge a couple minutes to change the heading.
There it
They're
is.
coming to
There's a shadow.
130.
So
Copy that.
I use bamboo
Copy.
corals again. That's just
40
sticking
degrees.
off it sideways.
Copy.
Watch it. Do you want to zoom at all on this?
No, I think we can pass this one up. I think
Copy
this is
that.
the same one that we were seeing a lot of earlier on. Thank you.
Little sea star on the bottom again. This really is rock bin and sea star.
Surprised this kind of slope is able to hold the sediment the way it does. It's so steep.
Yeah, but remember we saw the zoom in. We saw how grainy that carbonate was, just full of little pieces. So
right,
they all make little ledges for a few grains of sand to sit on.
Yeah, Tina had noted, yeah, that was a dead icidid. It's got some live branches to it, but part of it was dead.
Got some time here, pilot, if you want to take another close-up on those sea pens.
Copy that. Just want to see what's up. Nothing really, huh?
Yeah, no hard returns on my sonar.
Yep. Sea pens are almost laying down. That's right. There must be pretty strong current here.
It seems to be flowing right downhill, though.
Yeah, it's just
Or
flowing
sort of
right down over that cliff.
See, sort of sideways because the ROV is oriented toward the northwest right now. So
it's pretty much straight down.
yeah.
Pretty much moving, falling straight off of the cliff here.
Yeah, and the Sako was noting it too. The sea pens are almost completely flat against the substrate. The current's so strong here.
The sea stars look pretty flat too.
Yeah, that's true. They're not reason. It's probably why they can exist here.
Yeah, they're the only ones that can hold on without too much trouble. So you get a little density current coming down, and then it's all over for everyone.
Now, when the ship's all set, I think we can get another one in towards waypoint 3.
10-4.
So we're just about 100 meters away from waypoint 3, so we're pretty close to our end point, and we probably have about a half an hour left in the dive, I think.
Yeah,
Just
20
guesstimating.
minutes left. We got
Might be a few minutes less than that.
3:28.
Yeah.
Does 3:28 look alright for you guys, or does north follow the ridge?
Maybe we should make a push for the hillside there. It's right next to that waypoint.
That's fine.
Yeah, watch it. We're just waiting for the ship's doing a quick heading rotation. So
Yeah,
as soon as we're done with that, we'll get another move in.
So are you talking about that little pinnacle?
Yeah,
just to the
basically
west
that's
of the
just
waypoint
to the
3?
left of the waypoint. Yeah, I suspect that we're not going to see much else than this until we get to that little area.
Now, is that possible just instead of waypoint 3 going slightly to the west of it toward that pinnacle feature? That little rise there? Yeah, that's it.
I think that's it. Goes down.
It goes down?
Yeah.
Oh, okay.
It's darker in color.
Okay.
So I think that's
Down?
actually.
Got it.
Yeah.
So it's a hollow.
Yeah.
Okay.
Really?
Yeah.
Yeah, else it would be closer to get lighter, I think, rather than darker.
Doesn't make sense with the shadow on it.
Yeah, I'm wondering whether that's just because you're getting shading from the northwest, and could that just be the hill shade effect?
Yeah, we can check it out and
Yeah.
see what's there. But
Yeah, just take a peek at it because we won't have much time to really spend on it. But
yeah, we can definitely go that way.
okay, thank you.
Are you fighting to come back here? Come back west?
Oh no,
Oh,
I'm just not. I don't have a ton of thrust.
Oh, okay. I thought you were fighting it hard.
No. Just gonna go down a little.
Okay, they want to see this feature, pilots. It's going to be 310.
Let's do it.
That sounds good.
Looks like you're 90.
One of these
1,
stars
2, 3, and there was one right off the screen over in the right.
Well, one in the middle is different. It's not quite the same.
Ship's
That could be the mediaster.
It's just impressive how many. I don't think we've seen this many sea stars in any dive.
If you want to check one of these out, go ahead. Otherwise, don't go any more down.
Yeah.
This rock, copy that. Video, do you want to do a quick zoom?
You think they just
Sure.
slide over the top of one of these sea pens and eat it?
Yeah, possibly.
Sorry.
Yeah, so it does look a little different. That could be a mediaster. Unfortunately, we don't have our premier sea star expert on the line today. This is Chris Ma. He's been such a huge help. And he's actually taken a day off. I can't believe it. The guy's just slacking, I guess, you know. No, he's been a tremendous help, and he's been there almost every single dive. So it's really, really valuable having his assistance. So it'll be interesting to see whether he agrees, whether this is mediaster or something else. It probably is something else, but. I don't think. Oh, that's a really nice shot. Wow.
It's a little tiny snail there.
Yeah, I know that, but I don't think the star is mediaster after all. I don't recall those little pseudopodia or whatever they're called coming out of the back being on mediaster. Coming out of the back,
video.
making some notes.
That's pretty good.
Nice.
In anticipation of that move, why don't you come off the cliff there?
Ship has about 4 degrees left.
Copy. It will be, what, 310?
Copy.
Yeah.
What's that?
Bruce is typing on the
that's where I
chat.
am.
This is like a lion moving in on its prey. We see the sea stars slowly moving in on their sea pens to eat. Just like lion prides go off on their herds.
I'll just recenter. What's this?
Copy that. You ready for a ship move if I give it to you right now?
Center with your co-pilot.
Alright. 30 meters, bearing 310 degrees, decimal 2 knots.
Tina's asking, they're eating sea pens? No, we are not sure if they're actually eating sea pens, but there's so many of them, and that's the only thing around here.
Yeah, but Bruce may remember this too, and Tina as well. When we were on the southwest side of Niihau last year, there was a lot of what we thought were Hipposteria, which is a known coral predator, and they are mixed in with the sea pen forest there that we saw. So I'm trying to bring up the pictures right now, and we were all speculating whether or not those sea stars were eating the sea pens. Where is that? Trying to find the pictures. I may not have included them. Oh, here's the field. And we did decide it was probably Calyptrophora symmetricum. But yeah, there were sea stars mixed in with them, and that we suspected they could be eating them.
So
when you find a good spot,
the alternative is that all these things are basically being flushed down by the sediment, and for some reason the sea stars are
blown away.
Okay at holding on while the rest of the material is sliding by them.
Just going to autos.
Yeah.
Actually, the ship is moving towards that little feature there.
Okay.
So we're on our way there. We're just going to do one more pilot change and then
call it a day.
Going to push out.
Yeah. See the little knoll and call it a day.
Sure.
Great.
Just want to let you know that we're not waiting on anything. The ship's moving. So.
Okay. Thank you for that. I appreciate it.
Sure.
Got Levi in the driver's seat right now. Sean's taking over for navigator, and I think that's Chris, that's you and co-pilot, right?
Yes, sir.
And is it Tara? I think it's Tara over there at video. Art's clipping in the back row with Jasper Contour and myself, Chris Kelly.
We're not clipping, we're just talking.
Yep.
After we spend talking most of the day, we go and play with the samples and then
write some reports.
And write some reports for the rest of the evening. Yeah. And we should stay further away so we can pretend that that's a giant crab, but it's not.
Your starboard swing arm is out, lower is slightly out. See that?
So what we're doing, Tina and Asako and Bruce, is we're deviating slightly from waypoint 3, and we're going just slightly upslope, and it's actually just to the west of waypoint 3, just before it. There is a rise that looks like a little pinnacle feature, and we're seeing if we can get back onto that really hard substrate where all the corals were located right at the end of the dive here, at least to take a look.
Because this seems pretty barren.
Yeah, Dab, I think for us to make it there in time, we're going to have to keep tacking 30 meters on.
Yeah, I can stack.
Yeah, just keep them coming.
That feature's pretty close. It's only about 60 or so meters away.
Sounds good.
Bridge, is that?
Is that a crab?
Hey, can I add another 30 meters to the current move?
We can skip this, Levi. This is the whole area that we imaged a little while ago. Sorry, Bruce. I didn't mean to interrupt. What was that again?
Well, I was just saying, right before the Holardia appeared, that it was interesting that the fish seemed to have disappeared. I would have expected to see the green eyes and the cryonemia.
Good copy.
Here,
thank you,
but
Bridge.
there's almost nothing in terms of fish.
That's a good point. We're not even getting any of the sediment dwellers.
No, it's a tiny little guy.
It looks like a salp or something.
That's a lot smaller than I thought it was.
Or that could be one of those.
Oh, there is a little swimmer there.
Just as you said that, Bruce, chlorophthalmus just showed up.
All the way on the bottom of your screen.
Yeah, and we can pass on this one too. We got really great images of that little fish.
Copy that.
There's a couple coral trees that are in the distance, so maybe we are getting closer to a pinnacle feature.
Urchin down there?
Yeah, what was that?
Yeah, another one of those bamboos. And we're directly aimed right at the pinnacle right now. And this sea urchin we've seen before. This is a Cenopadina pulchella. Levi, you're getting the dregs here. We've imaged most everything here.
Yeah, copy that.
I know you're desperately trying to image something. Maybe we'll... How about whatever is on this sort of dead bamboo? We can take a look at that.
All right.
It's right in front of us. You blame Sean and Josh and the others for getting all the good close-ups, leaving you nothing.
Couldn't you have just skipped on something?
This is the same urchin, but he's on the coral, I think.
You think that's an urchin?
Well, no.
It seems really big.
I'm not sure it is, but...
Maybe it is. Oh, yeah.
Yeah, it's climbed up the coral.
How does an urchin like that climb up a coral?
How did the urchin
carefully? I don't know.
Ow.
He's got 2 feet.
Yeah.
He's got, I mean, it's a dead coral, so I don't think the urchin killed the coral.
Could he have just gotten washed downhill and got caught in there by chance?
It's possible.
Tina says he can do it slowly.
Yeah. That's it. I mean, he can. They certainly can move around.
Yeah.
So, and there are corals that predate on. Excuse me, there are urchins that predate on corals that crawl up on them.
Oh, really?
So yeah. We suspect that something called Histosideris actually is a coral predator, but we're not positive about it.
All right.
Thank you, Levi.
Yeah.
Clear.
I put it back on, okay. There's, what is that? Oh no, those are sort of sea pens again.
Roland's been digging around on the web about that blue mass of stuff that we did collect on that rock, trying to see if he can find anything that looked like it, and he thinks it could be a possible tunicate, but it could be something brand new. He couldn't find anything that looked like it anywhere on the web. So we'll get a better peek at it tonight when we're prepping the samples. Pinnacle, it should be coming up pretty soon. We're actually about almost 40 meters from the high point.
15 meters left.
Really disappointed.
And then, so I think D2 should be able to stretch out and just see it. And I can add on a little more. Copy that.
Now we're still seeing pieces of rock in the sand. These are all pieces of, they look like at least pieces of this coral reef debris. Bigger pieces rolling down the hill. And lots of ripples in the sand from the water that's flowing over the top of this.
You got a few boulders too. A big anemone on that boulder. And then now I'm starting to see something up ahead finally, I think.
If we get off at 412, I'll give you 4 minutes
poking
to
up.
set up.
And I think I see a coral on it too.
So 8 minutes.
I see a coral at least. Thought I saw the train coming up, but now I don't. This is another one of these icidid colonies that we were seeing earlier in the dive.
Alright, pilots, 15 more meters was added. And D2 should just be maybe 10 meters away.
Where is our high point? Inquiring minds want to know.
Maybe it was a dimple after all.
It could have been, because we seem to be going down, not up.
Shadow. So, 10 to 20.
I'm not ready to yield yet.
What's that?
Yeah, it's one of those crinoids, I think.
You think so?
Well, maybe.
I think it's overgrowing it. Oh, look at these.
What's that?
Look at all those.
These are the mushroom corals, most of which are closed up.
Oh, sorry, I thought that was neat.
I'm at like 25% thrust to stay off the ground.
So Levi, we should have listened to you. I have a feeling you said it was a depression, and we went, nah. And it looks like you were right. I'm not seeing any type of a pinnacle here.
Ooh, what's this?
Chief Nav.
It's not a pinnacle.
It's not a pinnacle, but it's still something funny looking. Yeah. Another gold coral.
Yeah, it could have a little bit of gold tissue on it. Maybe we could take a zoom at this pilot. It looks like it's in the early phases of being overgrown.
Yeah, we'll get in there.
Yeah, Washley, so we have about 5 minutes left before we have to start setting up.
All right, so...
I don't... we should have seen it by now.
Yeah, we're pretty much at that now that we know depression.
It's to the left, actually, I think.
Yeah, I think we haven't quite seen it yet.
I don't know.
Alright, video, you can frame it up.
That's interesting. I think this guy came along with the rock.
Current's blowing me a little bit.
Gastropod, a little pagurid in a gastropod shell down below it.
Well, it's something that's just about done. You want 10 more meters?
Yeah.
Finish up last 5 minutes.
Sure. Yeah.
So that block that they're on is also clearly a coral piece.
Okay to take a bit more zoom?
Yeah. Feel free to come in. Get this gold guy.
Yeah, and it's definitely a nodal brancher. You can see with that close-up exactly where the branches are coming out, right at the nodes.
It's full.
Okay.
The little hydroids that are growing on the dead part of the colony over on the left. So if you've been watching this broadcast, we're almost finished. We just have a few more minutes. And it's been a live feed from the NOAA ship Okeanos Explorer. We are on the east side of Wake Island in the Wake unit of the Pacific Remote Islands
Once you're done
Marine
here,
National
Pilot,
Monument.
just give it one more push towards that feature.
We certainly hope you enjoyed the dive. We certainly have. We saw a lot of stuff. Really cool stuff.
See if we can't
Sharks, beautiful corals, gold corals.
Check it out.
Lots of fish, actually.
Lots of fish, anemones, mushroom, corals, urchins, tons and tons of sea pens. I'm just doing sort of a little wrap-up here.
Yep. Interesting landforms, too.
Bruce, do you want to have a few final words here about the fish that we saw today?
Shark.
Well, it was interesting. We got up into shallow water where we started to see some of the spiny-rayed fish. We even saw one fish that's of a family that's typically thought of as a reef, coral reef species.
So we're stretched out.
So it was nice to see the diversity. Also, some hint of, well, actually more than a hint, the commercially important
to your port,
that we didn't know were at Wake Atoll. This is a marine national monument, so there'll be protection for those.
Yeah.
We saw pontoons and we saw one deep water snapper. And I just want to say I really appreciate the work of the videographers and the pilots to get great images of those fish and to zoom in on certain features that we wanted to see when we asked for it. So it was a really good dive.
So those images are better than the pictures of the dead fish in a tray in formalin? Is that right, Bruce? Is that what you're saying?
Well, yeah. We still use specimens for further identification. We can't get away from that. But, yeah, it's remarkable to see these things live in their natural habitat and also some aspects of their behavior. So this is really valuable.
Yeah, thank you so much, Bruce. That's the voice of Bruce Mundy at the NOAA Fishery Science Center on Ford Island. And we still also have Asako Matsumoto with us at Chiba Institute in Japan, and she's been invaluable on all of these cruises, and she was with us the whole day today, enjoying the corals along with us. And Tina Molitsova woke up early and got to see the last half of the dive here, because it's really early morning for her.
I think she went to bed late too.
Yeah. What have we got? What is going on with what's up ahead?
Is that the leech I got? Copilot?
Yeah.
I'll just sit down.
The ship just stopped.
It's just the
Alright, video, come in. I'll try to.
What is that? Okay. Oh, it's just coral branches. Okay, we were seeing things.
Just coral branches.
Just coral branches.
Just coral.
But anyway, we thank all the help that we've gotten on this dive and some of the other dives. Those of you who'd like,
how do you want to
can
set
hang
up
on
here,
for a
pilot?
post-dive discussion for tomorrow's dive site.
Copy that.
Lindsay is here, and she's setting up the display, and we're going to switch over, I think, any second. It's all ready to go. I think they're lifting off the bottom now.
See that? Oh, yeah.
Yep, watch these just lifted off bottom.
Great.
Thank you.
Okay, thank you, pilots. Terrific job today. It's a fantastic dive. So, let's see what we've got here.
You want to steal my cigars?
Sure. Let me know what else you want.
Go ahead, Nap.
Yeah, Dive Soup and I talked about it. We're comfortable where we're at right now.
All right, auto head in.
Okay.
Copy that.
Depth in.
Standing by.
Okay.
You can come up a little bit, and then I'll back up a little bit.
Copy. You want me to start turning?
You got enough tether, right?
Yeah.
Yeah? Okay. Yeah, definitely. Okay. You can look down and flip her around.
Yeah, you don't need to come back anymore.
Copy. Push it forward. All right. Okay, you're flipped around.
Okay, now you can push out. And actually we should come up together.
Okay, coming up, pushing out.
Got you in fisheye.
Okay, pushing out. Coming up.
Keep pushing out.
Okay.
And keep coming up.
Okay.
Delta 28,
Oh,
so
yeah.
you won't see yourself for a while.
Yeah, pilots, I might have you come up 100 meters before having the ship move ahead. Besides that, there's nothing in our way. We're just going to deeper water.
Yep. Start to see it in your tether can there.
All right, I see you. 16 meter delta, still coming up.